Okay, Ashwin, I'm going to start it and just let people start coming in.
Sounds good. I'll go on mute.
All right, welcome everyone. We're going to get started shortly. I'm the co-organizer of the event, Todd Dragan, assistant professor at Rice University here in Houston, Texas. I'm joined by my co-organizer and co-lead of Covert, Dr. Ashwin Beheshti. Ashwin, want to introduce yourself?
Yeah, I was on mute. Yeah, hi, I'm Dr. Afshin Beheshti. I'm currently as Todd said, a co-lead of Covert, the COVID-19 International Research Team. And I'm a scientist, a principal investigator at NASA Ames at KBR at NASA Ames. So we'll be having a good discussion today of all the people in Covert and various topics.
Yeah, so we'll maybe just give everyone one more minute to arrive before we get started. So in the meantime, I will share my the updated agenda on the screen. So bear with us for maybe one more minute before we get started. Okay, for those just joining us, I'm Dr. Todd Triangle. I'm an assistant professor at Rice University here in Houston, Texas, co-lead of Covert. We're going to just go ahead and get started. We have a packed schedule for today, 8 hours of content, mostly highlighting research updates from the COVID-19 research team over the last year, and also some exciting keynote speakers, which I'll get into. But for now, I'd like to pass it over to our co-lead and who will provide some perspective on what Covert is, and again, and then I'll jump in after Dr. Beheshti finishes and go over some specific agenda items for today. So, Ashvin, please take it away.
Oh, thanks, Todd. Yeah, you guys could hear me okay, right? I saw someone in the chat saying we were on mute, but if not, yeah, let me know. Okay, so yeah, as Todd said, I'm Afshin Beheshti. This is our symposium. Todd will go over it. Also, and you guys know the agenda. I'm sure people have signed up. So the whole point, I'm going to briefly give introduction to Covert and how Todd and I formed it, and the history, and the whole idea of the COVID-19 International Research Team is open, as its title is, open and collaborative science in age of pandemic. So this is the concept we have, basically open science and as inclusive as possible to have as many people involved to get us out of this pandemic. So a little history of Covert. Initially, as I said, I'm at NASA Ames Research Center, KBR at NASA Ames Research Center, and in NASA we have this platform called NASA Gene Lab, which is basically where all the omics for space biology ends up, all the bioinformatic type data, it ends up there. And just to briefly on that, we have these analysis working groups, which Todd was part of, and some other members of Covert were part of. And back in March, when the pandemic was really starting to take off in the US, several members who have bioinformatics and computational skills were already working, like Todd, were working on COVID-related research. So we kind of spawned off that, and because a lot of scientists at the time, which maybe many of you here, wanted this to help somehow. And you don't necessarily have to be a biologist, have virus background, but you have computational or other skills in biology that you can apply. And this is how it evolved. So it was basically a group of 10 to 20 of us. And now we've evolved to 200 plus members from all around the world. And we became a nonprofit on last July. And now today, we already have 2 symposiums that we have done. And we have many different subgroups and many different types of topics that people are trying to work on within Covert. And also, all this work that we've done the past year, we have currently 17 papers prepared for submission soon, hopefully one of them probably this week or next week, and to follow many more. And also we collaborate with other COVID type groups like the National COVID Cohort Collaborative, that's NIH NCATS backed thing. So just to briefly, we have many subgroups in Covert you see here, and people who are part of Covert are participate as much as they want in any of these subgroups to cover different aspects of which you'll hear today about COVID research, from modeling to machine learning to inflammation aspects of COVID to mitochondrial aspects and so on. So this is what we're going to hear from today from all the great work people have done. And the whole goal of Covert is to enhance public knowledge and the data sets through collection of sequencing data, also determine possible optimal targets for therapeutics, create new deliveries, because not only in Covert when we formed was having computational people, but we also have virologists, epidemiologists, clinicians, biologists, and so on. There's a whole slew of backgrounds that we could actually tap into and work together as a community to potentially develop new therapeutics, which is one of the topics today. We'll be talking about that for an antiviral, and then hopefully push it to clinical trials. So that's the whole goal. And not only to do that, but also figure out population dynamics, create new tools, which people will talk about today, for free for people to use. There's two great tools that people that we're going to hear about later today about that. And then also develop like multi-institutional collaborations for future pandemic preparedness. Just a big announcement we want to make is that we recently have this other COVID group, which was formed by Dr. Yvonne Kago. As you say, she's a former astronaut, retired Air Force colonel, and then also flight surgeon, space surgeon. So she has an impressive CV of many different accomplishments, but she had formed back in April, which you'll hear later on in our panel session, the Breathing Space Accelerator, which is basically a large consortium of large tech companies working together to, again, prepare people to, through these different methods to actually get people back to work, maybe get people to travel, and then using AI and other kind of tech, other data platforms, also create ways to help governments, and which we'll get into in the panel session at the end of the day. So this is a big announcement. They're joining Covert, and the whole goal is, and the whole goal is to actually be able to securely share data, have ways to actually help countries. Like she has an initiative with Thailand. This group BSA has an initiative Thailand to help them also develop centralize the patient databases for COVID, so the public can access their own data and figure out what maybe certain hotspots, so they don't know, okay, this region has a certain hotspot, how to tackle that. And then so the public can actually use this. And then also using the blockchain technology, the data is private, so then you can also give access to scientists to properly and in a private manner get access to EHR data that then we can use for research. So there's a lot of great things to come with this new collaboration, which we'll talk about later in our panel session of the day. But also, as we progress, we'll announce more and more on our website, and you probably hear more about us, how we're working with BSA and Dr. Cagle to push forward and help the communities. And these are some of the things that they're working on that will work with them. And then, as I said, there's basically two tools we'll talk about today that people should go to our website and download and use. One is the COVID exposure assessment tool, which Brian Schmoller will talk about, that he created. Basically a very nice tool, interactive PDF that we're going to eventually develop to a web app and a phone app that people could access the group exposure that might occur. He'll discuss it. And then our mask check app in collaboration with Real Networks that we put out. It's a free mask check app. You can download it, use it. Riza Razul will talk about this later, but it's a great resource and tool to basically, for schools or anywhere, public transportation. Anyone can use this tool to actually enforce mask rules, because as we know, this is, we still need to be vigilant with wearing masks in public places. And then also the mini subgroups are going to talk about all their nice, great work that we're doing. And one brief thing we'll briefly mention as we go on, we're also, we as I mentioned, we're not only doing research, we're not only trying to push forward the science and make things available to public. This through the initiative really driven by Dr. Nydia Travoa from the NIH Fogarty International Center, very talented postdoc there. She has a lot of collaborators out in different countries, like for example, Dr. Talavera. She's in Mexico, the University of Autónoma de Yucatan, and she in certain regions and certain countries, like for example, in Mexico, they're poorly underrepresented for the viral sequencing that they've done. So then if you want to actually monitor different new variants popping up, things like that, it is really important for us to try to figure out, like, for example, the UK does a great job. The US is starting to do a good job, and so on. What representations of different viral variants of this popping up, so we know what new mutations might happen that people should be concerned about. So in collaboration, Nydia's collaborator, Dr. Talavera, She's actually collected many samples since March. And now with Covert collaborators, Dr. Chris Mason, Dr. Michael Zodi from the New York Genome Center, they've agreed to sequence 500 some of these samples from the Yucatan region, and then we'll also do some microRNA sequencing, and then some serological assays on top of that. So this will be available once they sequence it back to the public, because it'll go back to GISAID, where the public viral sequences are. So now we can start mapping places like Yucatan region, and then also Dr. Nia Tribi from University of Ghana. He's also providing samples to sequence, again driven by NIDIA. So we're going to be able to get additional things and provide that back to the public. And then we're, since we have collaborators and resources that possibly can do this, we want to reach out to other places that might have samples that need underrepresented sequences from places that need to have, understand what kind of viral sequences are out there. So, and of course we haven't done this all on our own, because the amazing thing about Covert and the community that comes together, there's many different resources that people have provided. One was the law firm Wiley Law Firm, which set us up generously set us up as a nonprofit pro bono. Very great people there that did that, and also all these other computational resources and other resources that are brought in to help us out since we started, because everyone here is doing this basically from the kindness of their heart in a grassroots movement, as opposed to anything else. So I guess I'll stop sharing and pass it over to Todd now.
Yeah, great. Thanks, Afshin. I'm going to share my screen now. Just go over some additional information for today. So welcome again to our third symposium. This is the third symposium we've held. Covert has held in less than a year. Very excited to be here with everyone today. Once again, I'm an assistant professor at Rice University here in Houston, Texas, and I'll just get into it. Okay, so just some background. I said this is the third. Well, we held our first symposium April 17th, 2020. It was a really nice first event with really great attendance here, and then followed up with a second symposium back in July, and this brings us to our current symposium here. Okay. And just to highlight a few things here, we have over 300 participants today with us from over 17 countries, which is amazing to see. Some high-level summaries of who's attending today. As we've enjoyed for all three of our events, great representation from industry, academia, and government. So it's wonderful to see that. The international participation from various sectors, all with us today to hear all about this. A lot of Covert specific research updates, but also with some really wonderful keynote speakers. And with that, I'll go briefly over today's agenda. Right after, as soon as I stop speaking, I'm going to hand it over to for a couple workshop recaps from workshop leads that we held prior to this event. And after that, we'll hear from Dr. Duncan McCannell for his all the great work he's doing at CDC and Spheres. We'll go into our lightning talk session, have our second keynote, Dr. Dinesh Agarwal, who is replacing our keynote, Dr. Sharon Peacock, who unfortunately wasn't able to join us today for personal emergencies. So, but we're really excited for Dinesh's talk. Then we have two lightning talk sessions after a break. We'll have two more keynotes in a lightning talk session, and we'll end this whole event with a panel discussion, which should be a lot of fun, where we'll cover past, present, and future of Covert. With that, just bookkeeping items for everyone today. We have 21 speakers, 4 keynotes. This is a webinar format. Please, there's a Q&A session for every talk. Please submit your questions, upvote questions that you have interest in seeing answered. We have a team behind the scenes that's working to make sure that the top voted questions will get answered by the speakers. This is live streamed to Facebook Live. This is on the Covert Facebook page if you want to look at it. There's the link. There's the Twitter hashtag for this event if you'd like to tweet. It's hashtag Covert 2021. And a link to the event recording will be emailed to all registered participants on about a day after the event concludes. So with that, I just want to say thank you to all of the organizers and sponsors, Ken Kennedy Institute, Executive Director Angela Wilkins, Rice Computer Science, Heather Ferreira, Covert Outreach Team, Eliza, Cecil, Sigrid, and Eve, and of course, all of the speakers today that made this event possible. And with that, I would like to hand it over to Professor Eve Ritelli, professor at Iowa State University, to give us a quick recap of the RNA-seq analysis workshop that she led, and that will be followed by Dr. Fritz Sedlacek, who will give us an update on the SARS-CoV-2 genomic analysis workshop that he led. So let me stop my share here and hand it over to them. So Eve,
Hello, everyone. I'm hoping this is sharing. So I wanted to just quickly go over our, oops, sorry, I can't see this, shoot. I'll just leave this in this view because it's easier. Can everybody see that? Okay, so can people hear me and see this? Okay, so we had a hands-on workshop basically on analyzing RNA-seq data, and we were using COVID-19 data as an example in an HPC environment. And the organizer is Arminder Singh, my graduate student, who has just got he's graduating, got a great job offer, which he's negotiating about, so I can't talk about it. The basic premise is that we went from RNA-seq, multi-study raw RNA-seq data to visualization. And darn, I'm so sorry. Let's see. And the first workshop was Jeff Haltom, and he gave a basic Python refresher workshop for people that weren't up on it, because Python was what we were using. Then Arminder went in and talked about the beautiful program he's developed, PyRPipe, that automates and makes efficient and makes reproducible analysis of data, going from the RNA-seq data in SRA to a full analysis of it. And finally, Priyanka Bandri talked about our program MetaOwnGraph, which was developed by Arminder Singh and a number of others. And she gave a really nice workshop on that. So that's for visualization and analysis of the data once it's been taken through PyRPipe. So that, in a nutshell, is our workshop. And it went very well.
Thanks so much, Eve. That's great for the thanks for the recap here. And just and these are, again, kind of workshops that either were co-organized or affiliated with Covert members and happened just again earlier in the week. And I'd like to pass it now over to Fritz to give us an update on the SARS-CoV-2 genomic workshop.
Yeah, happy to do that. Welcome, everyone. I hope you can see my slides. So this was a workshop that we organized between Todd at Rice University, myself at Baylor, together with the CMMR and together with Ben Busby on DNA Nexus for sponsoring compute. So overall, we had like 54 participants that we accepted from all over the world, as you can see, ranging really from the Philippines to the West Coast of the US. This was really important for us to really kind of update everyone on best practices on how to process this RNA-seq data sets for SARS-CoV-2 directly and identify the important variances in all of their own samples, also from Africa up to really like North Europe. The team was amazing, I would say. Like, we had great support on the back side from Meredith, Michelle, Angela, and also from Gerald from Rice and Baylor. And then, as I said before, DNA Nexus really sponsored our compute. Ben Busby did an amazing job in bringing everyone up to date and how to connect to DNA Nexus and leverage the cloud for each of the participants, enabling them to compute their own variants and dissect their own samples later on. And then module 1, Sarah kicked us off from Baylor with a detailed analysis of QCing reads and then how to filter reads. And then I was taking over module 2 in terms of mapping and variant calling. And then we did a happy hour at the end of day 1 where everyone was contributing one slide to introduce themselves, and it was a nice networking session. Module 3, Todd was leading that from Rice University, obviously, about assembly and deeper details on low frequency variant calling and media. Module 4 really dived into phylogenetics of these variants. Since this was a virtual workshop, we relied a lot on different platforms, as I said, for computing and Nexus, which is kind of a middle layer between the cloud and us. Remo was our primary platform for communications and sharing lectures. GitHub, where we put all the instructions and slides of the workshop, and this is also available publicly for everyone. And I think we're also going to post the videos of the lectures soon, at least this week or next. And then we use Slack to really build a community, to have the Slack channel up also for later times, so people can communicate with each other and network further or raise questions. We started to get some feedback over a survey in the end. Again, the workshop just ended yesterday evening, so right now we just have 9 people that answered this survey. But overall, it's extremely satisfying and extremely nice to see. So as you can see at the top right is the content. People are really enthusiastic and happy about the content. Lower right is the difficulty level. So you could see that some of them said like extremely easy to follow. Some of them were neutral. This was kind of expected since we had a really mixed level of participants. Some of them are kind of novel to terminal, some of them are not. And then using the Nexus was also kind of split a little bit. Again, this is always the hardest part of a hands-on workshop on how to connect to the cloud instances and get that going right in the beginning. But as you can see, no one kind of completely failed. So this is really thanks to our team here that really picked everyone up that struggled a little bit and showed how it was going. So to bring it to end, I think this was a really successful workshop that we had. We might want to think of in the future to extend it by 1 day. We're also thinking about, not sure if we are going to do that, to include nanopore data set into this workshop, since we know like nanopore is also one of the other sequencing technology for COVID sequencing. And we are probably looking out for another workshop since we had more as double as registered people than participants in 2021. And that's kind of summarizing the entire workshop. I think it was a great success, and I'm really thankful again for the entire team helping, and thanks for Todd who helped me organize this event together.
Yeah, thanks, Fritz. Thanks so much for the summary. Thanks, Eve, as well. And so I'll just kind of go back to the screen share again, and just while I'm here, I just wanted to emphasize that one of the main missions that of Covert is actually outreach and symposium. So we're a symposium organization. So we're really excited that we've collaborated with many on some of these workshop events to get both hands-on training, but then outreach on the scientific side. So I'm going to go back into my screen share here. Let's see. Wanted to highlight one thing on the website. Sorry about this. Let me close that window. So we just got a recap now of the workshops, and then now our first event, and will happen in about 5 minutes. I will be joined by Dr. Duncan McCannell, Chief Science Officer of CDC's Office of Advanced Molecular Detection, and he'll talk to us about bootstrapping national genomic surveillance of SARS-CoV-2. So that's our first keynote speaker here. I'm going to just go and bring up and talk a little bit about our subgroups here while we wait for a keynote speaker to arrive and join us. So bear with me for a second. I'll stop my share here. And yes, so Ashwin, maybe we could talk a little bit while we're waiting for a keynote speaker to arrive to talk a little bit about the subgroups structure. So I think maybe I can just go to the website, our groups.io website, and maybe talk a little bit about our subgroup structure.
Screen, for the, if you want to look at
this.
This.
Yeah. If you maybe you can share the screen. Either one of us can share.
Yeah. Show that. Go back to the diagram while we're waiting for Dr. McConnell. Okay. So here's the subgroups. All right. So you see that, Todd, right? Yeah.
Yep, exactly. So here, again, as Ashwin mentioned, Covert is really reinforced by all the subgroup activity that we have, and the icons might be a little bit small for everyone to see, but the big idea here is we have a collection of various subgroups and different expertises. And the whole point here was to bring together international team of experts with expertise in all of these various topics to work together collaboratively within subgroups, but also between subgroups and across subgroups to bring some innovative solutions either on therapeutics or understanding impact of variants and et cetera. So with that, I think Dr. McCannell is now here, so we'll go ahead and get into our first keynote presentation. We're a little bit ahead of schedule, but I think that's okay. So I think we'll just go ahead and get started with that. Duncan, welcome. If you'd like to share your video, feel free. If not, I'll just go ahead and get started with your introduction and we can get into your presentation. Okay, great. Thanks so much. So welcome, Duncan. Great to have you, and thanks again for agreeing to join us today for our third symposium and anniversary event. So I'd like to just give a little bit of background first before I hand it over to Dr. McCannell here. So Dr. Duncan McCannell is the Chief Science Officer for the CDC's Office of Advanced Molecular Detection, OAMD, where he helps implement, coordinate, and support pathogen genomics, bioinformatics, high-performance computing, and other innovative laboratory technologies across the CDC's 4 infectious disease centers. I've also been fortunate to learn about an initiative that he's started with others called Spheres at CDC, where he's connected just this amazing network of public health lab scientists and resources all together. So it's been really fascinating to learn about that from Dr. McCannell. But without further ado, I'll hand it over to our first keynote speaker. Please take it away.
Thanks, Todd, and thanks to Covert for inviting me to speak this morning. As someone who spends at least part of their career coming up with acronyms and different names for complex programs, I got to say Covert probably takes the cake. It's a great name. So today, what I'd like to talk about a little bit is genomic surveillance within the United States, where we've come from in terms of SARS-CoV-2 surveillance, and perhaps start to get into a little bit of how we hope to move on in the future. But first, it's important to really understand the context in which a lot of this was happening. So as Todd said, I come from a program called the Advanced Molecular Detection Program. It really was born back in 2011 with a bioinformatics blue ribbon panel at CDC, which brought in a panel of external experts and really underscored the need to make concerted investments in advanced laboratory technologies and bioinformatics, and the workforce needed to make that happen. The Amd program was approved in the budget in 2014. It's a $30 million a year public health laboratory innovation program. We work across all of CDC's infectious disease centers. We do things like also manage the high performance computing, scientific computing activities for the agency. And we also do a lot on workforce development, managing fellowship programs and really trying to build out the public health workforce to be able to use these types of technologies to make better use of the data that's there, to be better contributors of the data that we generate, and many of those things. The program itself has 5 key objectives. These are probably fairly self-evident. The first and foremost is pathogen detection and characterization. We want to be able to use a lot of these new technologies to detect and characterize, respond to infectious disease threats. Ideally, you want to translate this into things like the development of new diagnostics, share data to ensure that therapeutics can be maintained, et cetera. But a lot of this requires sustained investment in infrastructure. Building bioinformatics capacity, integrating this with information systems, in many cases across the public health lab system, across the public health system in general, and across the clinical space. There's a lot of gaps there that still need to be addressed. And ideally, we want to be able to use these kinds of approaches, these kinds of technologies to look down the road a little bit. Ideally, generating more data, using it to build better models, and at least be able to predict where we might be in the near future, if not a little bit farther out. So next generation sequencing capacity expanded very rapidly. If you go back to where we were in 2013, only about 6 or 7 state public health labs had next generation sequencing capacity. A lot of this was attached to FDA's genome tracker, foodborne or food surveillance programs. Some of it was attached to PulseNet. Some of it was based on internal state funding, but there really wasn't a huge amount of capacity across the state public health lab system. If we fast forward, oops, going the wrong direction on my slides here. If we fast forward a couple of years, it took until 2018. All right, it's going to really speed up there. 2018 for sequencing capacity really to hit all 50 state labs. So it wasn't exactly an overnight process, but that ramp up was fully accomplished. Once there, many public health labs, foodborne disease detection and foodborne disease surveillance was probably the primary driver in many public health labs, but it certainly wasn't the only one. Once the technology was there, once the capability was there, many state and local public health departments started pivoting and using these approaches to, sorry, just saw the chat. Using these approaches for things like healthcare associated infections, for viral hepatitis, for Legionella, for TB, and many, many local priorities. So it was a gradual sea change. The other important thing is that we realized that there were a lot of shared challenges. Workforce development, particularly around laboratory and bioinformatics, continue to be a challenge. Informatics continue to be a challenge. And trying to figure out how to wrap all this into a quality framework and align it with CLIA and other requirements continue to be an important factor for many public health labs. The worst animated slide ever. So yeah, one of the things that we did try to invest in early was some shared infrastructure and platforms. What you find though is that public health traditionally has been very siloed. A lot of these activities happen in relative isolation, and trying to figure out systems that can actually operate across them takes some time to develop, to socialize, and to get right. They also take sustained investment, which is challenging when you're trying to do it at these kinds of scales across these many different programs. But we have had some success there. So let me take you back in time. Let's switch gears now that I've given you a little bit of context of sequencing, and take you back in time to early April 2020, where the pandemic had started to roll or become evident in the United States. I'm not sure what our case rate was at the time point I've got on the slide, which was at the end of April. But if you look at what sequences had been generated, there were about 4,000, just shy of 4,000 sequences generated for the entire United States for those first 3 or 4 months. The breakdown in this is very telling though. So I essentially just went into GISAID for this. I pulled categorized US submitters into academic and clinical laboratories, public health labs at the state and local level, and then federal labs, and private sector was essentially the other category. And if you look, a few things pop out almost immediately. For one thing, the majority of sequencing really was occurring in academia and the private sector, and oftentimes these were in labs that had some, but not necessarily direct connection to public health, state and local public health, or federal public health. A number of states, federal sequencing was really the only sequencing that was going on. These are these states with orange dots. And you did have a handful of states that had state and local laboratories that were starting to sequence with some regularity. Those are highlighted here in purple. So it was a little bit of a mix, but you also had a lot of uncharted territory out there. Many states had 0 sequences submitted at this point, even though they had cases. So the picture really was that the sequencing across the country was uneven, it was consistent, and in a lot of states, it was driven by local cluster investigations or existing programs or networks that were already set up. So it wasn't necessarily giving a representative view of viruses that were circulating around the country, or how that could, or giving useful information on how that could be used. To address this, we launched an initiative. So the initial emails to this went out, I think, inviting about 30 or 40 labs that had been sequencing across the US, a number of the large public repositories that people were sequencing and submitting data to, a number of other federal players that were involved in sequencing. But what we felt was that we needed to at least try and align all of these efforts. You know, there were many different labs that were starting to sequence, many of whom were doing it relatively independently or in isolation. And it really was incumbent on us to try and figure out how to align these efforts and essentially use them to help coordinate national sequencing efforts as they existed in the moment. This initiative became Spheres, and this is the original announcement from the New York Times. But our goal was really to align academic and private sector sequencing efforts and try and hook them into their local state and local public health labs as much as possible, and tie that into federal efforts as much as possible. We also wanted to maximize the quantity and quality and usefulness of the data that was being generated by many of these labs. There was tremendous enthusiasm for generating sequence, but you know, there wasn't really a standard way of going about it. There wasn't a lot of consensus on protocols. There wasn't a good source of information on how to structure data or metadata or submission processes or best practices. And we felt that that was a way that we could, at the very least, in the absence of some dedicated resources to support large-scale sequencing, we could use the power of convening to start making some improvements. This also allowed us to engage with a number of partners across academia, the private sector, NGOs, and you know, basically bring them into the fold and start having this conversation about what is really needed to scale sequencing. How can we use this data more effectively? How can we ensure that we're getting the information that we need? And it also gave us at least an interface with which we could align our efforts with other global sequencing efforts, many of which are probably familiar to this audience, many of which are pretty sophisticated and far along. So who makes up this Fierce Consortium today? This is probably actually a little out of date. We have nearly all state public health labs on board now, many county and local public health labs. Most of the federal agencies that are directly involved in sequencing are participating pretty readily. But as you can see from the slide, and obviously these are not meant to imply endorsement by CDC or HHS, but just to give you a sense of the diversity of participation, there really is quite a breadth of different groups that are involved. At last census, we have just over 950 scientists that participate in Spheres, either through Slack or through our Zoom calls, and this represents well north of 200 organizations across the US, so it's gotten fairly big fairly quickly. So what does Spheres do exactly? A lot of what we do is just bring together a technical community of practice, you know, that provides a basis for networking. Has fostered many collaborations. It allows for much better engagement with state and local and federal public health. And it really brings together the shared pool of expertise and capability from across the US public sector and private sector. There are a number of different focus areas. So many of the groups that are participating focus on genomic epidemiology. That could be things like outbreak investigation and response. It also means things like large-scale surveillance. A number of university labs, clinical labs, are either working very closely with their state or local partners, or conducting large-scale surveillance in their own right. And having some sort of guidance, some sort of connection to public health is extremely useful, because it can make sure that the data that's being generated can have the greatest impact and ideally the fastest use. It also allows us to work on things like laboratory and bioinformatic strategies standards. So there is a thriving wastewater surveillance community out there. Spheres is engaged with that because many groups are interested in using wastewater surveillance as an early indicator. There's also a lot of groups that are very interested in strategies like pooled testing of large groups. How that interfaces with sequencing, how that data can be used, how we can use that to better inform public health is an ongoing topic of conversation. There's also some of the basics, sequencing workflows, sequencing platforms, protocols, those sorts of things. We have actually all of the major vendors participate in that. So, if there are technical questions, there's essentially a direct line to technical support and a pretty broad community of other users that likely have experienced similar issues. So it's, as a technical resource, it's actually been pretty exceptional. It's also allowed us to target things like standards. Naming conventions for viruses, for example, were one of the early things that we took on. And we've also worked with PHAGE, or the Public Health Alliance for Genomic Epidemiology, to develop some socialize some metadata standards, consistent ways of capturing not only data, but contextual data about the sequence data that we're collecting, not only to improve the quality of individual labs, or the data collected by individual labs, but to ensure that the data that is flowing into public repositories like GISAID and NCBI has consistent and well-documented metadata throughout. It's also been a great forum for technical assistance. So one of the things that CDC has spun up fairly recently has been a team that we call TOAST, or the Technical Outreach and Assistance to States team. I think that's right. But essentially what it does is reaches out to the various state and local public health labs, identifies a lot of their technical or workforce or resource challenges, and starts to figure out how to best either connect them with partners in the community that can help, or identify standard menu of protocols and resources that they can draw on to get sequencing up and running in their jurisdiction. The other advantage is we've been working very closely with many of the sequence repositories directly, as well as many of the tool developers. So, next-strain developers are very, for example, are very closely involved in many of the Spheres conversations. There's a direct feedback loop there, and many of the groups use tools like that as part of their local reporting or to visualize their data. It's also been a way that we've used to, it isn't in itself an explicit funding organization, but as we start to identify resources and bring additional potential funders on board, this is a way that we can not only help target some of those requests or those calls for proposal, but also to help tailor that a little bit. One of the challenges that I think that we have going forward is sustainability. Many of these labs have filled sort of a surveillance role that is traditionally held by public health. We're trying to figure out how to best transition some of that into more of a research domain. But public health doesn't necessarily have all the kinds of tools that NIH or a research granting agency might have. And trying to figure out how to make sure that this is supportable long-term and sustainable is key, because there's tremendous potential here. There's, we've had enormous success, but trying to figure out how to make sure that it is supportable is something that we're working on very, very closely. This also allows us to look at the infrastructure, look at the data that we're putting out as CDC and other partners, and really get feedback on what we should be doing, what we should be prioritizing, what would be useful to the broader research community. And for some of our data resources, actually user acceptability testing. We can put something out and see whether it works or where there are glitches or problems. NCBI, for example, has been using this community as a way to test out a lot of the submission processes and some of the new tools that they've put online. We've had many of the other secondary repositories or resource sites that have been doing a lot of analysis show off their tools and basically work through the community to figure out how to make them more useful. And as I said, a lot of this is conducted over Zoom. We've adapted to virtual life fairly well, but it's also a fairly active Slack workspace, and that has been the two avenues of success. So fast forwarding a little bit, let's look at where we were in mid-November. So at that point, the US sequence submissions to GISAID were at about just over 41,000. As you can see, over half of that, though, was still academic and clinical laboratories. But many of the state and local public health laboratories were starting to generate significant amounts of sequence data. CDC really was filling a role where, and other federal laboratories were really filling a role where they were sequencing in states that, or in regions and jurisdictions where capacity was still fairly limited. But you can see from the chart here that there still were, well, at least one state that was that still had no sequence submitted. So, you know, it wasn't necessarily evenly distributed. The size of the circles here on this chart also give you a sense of the proportion of sequences that were submitted. So you definitely had some states that had submitted a handful and probably weren't very representative, or weren't certainly weren't longitudinal. You also had some states that had more or less saturated the ground with sequencing reagents, and had covered an enormous volume of the cases that they'd seen. So there really was a lot of disparity between. So before I go on, I think it's important to really look at why it is challenging to stand up large-scale surveillance in the United States. And part of that is, well, and this probably applies to many countries as well. Part of this just has to do with the nature of diagnostic testing. If you look across the country, there are literally hundreds of independent test providers. So these are generally private clinical labs, or in some cases public labs that perform primary diagnostic testing in most municipalities. So, you know, if you look at any city, there's probably 2 or 3 different providers that are providing routine community testing. Those providers aren't necessarily, there's no real incentive for them to hang on to those, hang on to those samples, or to refer them to public health or to a sequencing provider. There aren't necessarily ways to transmit the necessary metadata to make those sequences useful. And so really it is a problem with the distribution of testing and the fragmentation of testing. There are some other challenges. Many public health labs are stretched pretty thin. So, basic diagnostic testing, other priorities need to come first. And in many cases, that has meant that genomic sequencing and genomic epidemiology of SARS-CoV-2 has taken a little bit of a backseat. In many public health departments too, you have the other end of the spectrum, the epidemiologists and people that are actually taking these sequence data and turning it into meaningful public health action. Don't necessarily have the molecular background to understand or to translate what the sequences are saying, what the various representations are saying, into something that can actually be used for public health practice. And as we've learned in many cases, in many investigations where groups are interested in using SARS-CoV-2 sequence data, the results aren't always as informative as we'd like. If you're looking at a fairly rapid outbreak in a congregate setting, for example, depending on how many introductions you have, the sequence data might not be as informative as it could be, just because you won't see that many differences with this virus over a very short amount of time during a very high intensity transmission. So following those transmission chains isn't necessarily going to give you additional answers. This piece in the middle here is essentially what we call the Amd Triangle, or well, I'll call it the Amd Triangle. But it essentially is the 3 pillars that we build on: sequencing capacity in the lab, epidemiology, and applying these to integrating the results of laboratory testing with epidemiology, and the bioinformatics needed to actually convert a lot of these raw data into information that we can use. All of those are critical as part of the public health response, and meaningfully, if you're trying to do that in a very short time frame, it's absolutely critical that you have the sequence data and the testing data side by side, essentially very closely integrated, because as we've seen in many countries, if testing, handoff to the sequencing lab, analysis and translation to public health response can easily take 10, 14, even longer days. And that can limit the usefulness of the data. This is you're essentially looking at many of these outbreaks, many of the surveillance data in the rearview mirror. And depending on what your application is, depending on what your goals are, that can be problematic. Encompassing all of this, there are a number of factors you need to consider. Things like turnaround time, how quickly you can get from sequence from the swab to an actual piece of usable information, the cost and sustainability, depending on how you scale it and what you're trying to do and accomplish with the work you're doing. That obviously feeds into like the scale and objectives of your work, but also the actionability of the data. We really encourage public health departments that are bringing sequencing on board for any pathogen, and including SARS-CoV-2, to really consider how they're going to be using the data. How can it be used most effectively? And to really work backwards from there and figure out and use that to not only scale their approach, but also design their entire work and sequencing strategy so that they can actually meet those objectives. And underpinning all of this is logistics and data sharing. That's usually the single most greatest challenge across any large-scale surveillance effort, and it's often extremely underappreciated. So to scale things up, we basically rolled out 2 programs beginning in November of 2020. The first of these was the National SARS-CoV-2 Strain Surveillance Program, or what we call NS3. Initially, the goal of this was to bring in 20 random positive samples per month per jurisdiction, but this rapidly, sorry about the typo on the slide, this rapidly scaled up to about 3,000 sequences or 3,000 samples a month. The advantages of this system, this is essentially built on what we conventionally do for seasonal influenza monitoring. But the advantages are it brings all of these specimens into a central location. So they're all run at CDC. They go through a very similar testing and sequencing pipeline. So the results are very, very consistent from period to period. And more importantly, though, it actually captures the viruses. So they are available, they're banked, they can be cultured, they can be analyzed further, and they can be made available to the broader research community. So this really serves 2 roles. It provides some initial long-term or large-scale surveillance for the country, but it also ensures that those viruses are available for additional characterization. To supplement this, we launched a series of what we call baseline contracts in December of 2020. And really the problem that we were trying to address here was really the sample access problem. As I mentioned earlier, because of the distributed nature of testing in the US, it's difficult sometimes to connect those, it's difficult always to connect those samples with sequencing capacity and connect those to the data with actual public health action. By working with some of the large diagnostic companies, we're able to essentially sample from their diagnostic flow and attach sequencing onto that and use that as an additional source of sequence data. That's been an important increase, and essentially what we did there is work backwards. We looked at total diagnostic test volume across the country, looked at providers that were doing the most testing across the broadest geographic area, and essentially tried to assemble a patchwork, a quilt that would cover the entire country and cover specific populations that we wanted to focus on. So sequencing over the past few months, as a result of some of this, has really scaled up. And the other piece of this is many state and public health departments, especially after the identification of the first variants of concern, began to reprioritize sequencing and started sequencing in fairly large volumes. And I'll show you some of that data in a minute. But if you look at where we were in mid-December, we were at about 50,000 sequences in GISAID, about half of that in GenBank. And just over the past 3 months, that is essentially quadrupled and is continuing to accelerate. So there's a fairly large amount of sequence submitted into both repositories. Spheres itself and CDC's approach in general has been to ensure that we have data in as many formats as openly available as possible. And so we've actually been encouraging groups to submit to multiple repositories if they're in a position to do so. So most CDC and our state and local public health departments all submit almost immediately to GISAID, consensus sequence to GISAID, but we're also submitting to SRA, and we're submitting to GenBank. And part of the reason for that is ensuring that there's a lot of applications of raw sequence data that we want to make sure is available not only for public health but the broader research community. And there are many tools downstream of public health surveillance that are extremely advantageous to the broader research community, advantageous to public health, but for a variety of reasons, they might not be compatible with GISAID usage requirements. And so this ensures that public sequence data from the US is as public as we possibly can. As of March 29th, as I said, there's about 220,000 sequences in GISAID, about 100,000 in GenBank, and we're starting to deposit more and more into SRA. That is at about 21,000. We have a significant backlog to catch up on, and that will hopefully continue to accelerate. If there's interest in looking at this raw data, I've pointed to the Umbrella Bio project here at 615-625. That has about 30 different sequencing projects underneath it, and it will continue to get populated by both U.S. federal efforts as well as a number of the Spheres participants as well. So let's look at the breakdown today. This is actually from about a week and a half ago, so I apologize, it's a little bit out of date. You can see from the orange that federal sequencing efforts, so that's driven largely by NS3, and contract sequencing is starting to take up a fairly large proportion. You can see from the bar graph down here, the approximate proportion of US submissions every week that are made up by those federal submissions. But really what we're trying to do is paint these broad strokes of what is circulating across the country and ensure that not only state and local public health, but the broader research community has important background contextual data, baseline data that they can draw from and really understand what's circulating nationally, what's circulating regionally, and what's circulating ideally at the state level as well. Academic and clinical sequencing continues to play an important role. This is dominated by several large academic and clinical centers, but that is absolutely critical, and we're hopeful that that will continue. And as you can see here, state and local public health has really increased as well. In purple, dark purple here, it represents a significant proportion of sequence submissions every week. And right now is making up, I don't know, what is that, close to a third of total sequence submissions since the beginning. Again, that is a little bit unevenly distributed, but hopefully between state and federal, which now make up about half of sequence submissions in GISAID, we will continue to improve that. So at present, we have basically three different sequencing inputs. There's the national surveillance that I talked about, which really as a goal is to perform large-scale, representative, and consistent data generation for across the country, and to allow for these large-scale phylogenetic assessments, look at viral monitoring, and really conduct that national level risk assessment of emerging variants. State, territorial, local, and tribal we're hoping are going to not only supplement sequencing at the state and local levels, but have the resources to focus in on cluster investigations, specific local and regional transmission patterns, or variants of local concern, or identify them early. The advantages of that are, the national surveillance has a bit of a time delay built in. It takes some time to bring in all these samples. It takes time to get the sequencing done. So, by the time we actually get genomic sequence data out from that, it's probably 10 to 14 days from the sample collection. State and local public health labs have the ability to be more nimble than that and respond much more quickly, and ideally have those data responding in real time. We also want to make sure that we're supporting research innovation and really building this extended network through Spheres. There's an enormous number of really exciting studies now that are starting to make use of not only this data infrastructure that's starting to take shape, but to build in and around the data that's being gathered. And we're hopeful that many of these academic labs that have really been been trying to do a dual role of surveillance, outbreak response, and research, applied research, will be able to focus more on the latter two, and to work very closely in partnership with their state and local public health labs to conduct research that's going to impact their community and nationally. These all fit together. You know, Spheres really covers the broad scope all the way down from the national to the regional level. Right now, this is probably out of date, but I think it's around 6,000 a month. NS3 and baseline, as you'll see, I've got 25,000 sequences a month and ramping here, and that's probably a low estimate. But again, you know, state and local public health departments have been really starting to pick up steam, and we're really excited about the potential there and the collaborations between state and local and academia. So this is from the CDC variant website. I'd encourage you to just search for CDC genomic surveillance or genomic surveillance dashboard, I think are the Google terms that will get you there. This gives you a sense of output. So, you know, nominally we've got about a 7,000 sequence a week target that we set somewhat arbitrarily early on as a stretch goal, just because of where we were in December. Right now, we're easily hitting 10 to 12,000 sequences a week if you combine federal, state and local sequence submissions. We're doing interesting things with that. So one resource I'd like to point you guys at is if you look at nextstrain.org/group/spheres, one thing that we do very weekly, it's usually on Tuesday nights, is build state-focused builds of NextStrain. So for all 50 states, as well as DC and Puerto Rico, we routinely build state-focused builds. So we subsample the state, and actually I'll show you one right here. We subsample the state and compare it to other states in across the United States, but also some global context. And so this gives a sense, at least a local sense of what is happening in each state and the overall genomic epidemiology of viruses that are circulating there. It doesn't have fine-grained geolocation or geographic information, but the date ranges are usually pretty good. This is from California, so you can see that it's a state that's been sequencing pretty regularly. The query state here is always in red. The other U.S. states are in various colors, and gray is the global context. We've also been building, so there's one that actually brings in all of the NS3 viruses or NS3 sequences. We build that one pretty regularly. Here I just was demonstrating that we can look at specific genotypes using the built-in NextStrain tools. But at present, I think this visualization has about 1,500 or so viruses that were specifically brought in through NS3. Again, all of those sequences are listed in public repositories. All of those sequences and all of those viruses are available for study. The bold highlighted ones, I think I actually chose, was out here in the Pacific Islands. I can't remember what I was doing, but yeah, some of the beauties of open data. All right, in the time that remains, since I know I need to wrap up pretty soon, I wanted to talk a little bit about variant classification. So again, if you want to look at the CDC webpage on this, there's a lot of detail. We recently updated our list of variants of concern. These definitions were developed in collaboration across the US government, across the US interagency by the SARS-CoV-2 interagency group. We define three broad categories, and these essentially correlate with systems used elsewhere. They're very closely aligned to what WHO uses as well. But we put some specific definitions around variants of interest, which are mostly focused on genetic markers that we know have impacts on things like receptor binding or neutralization or potential diagnostic impact. But it is essentially a genetic definition. Variants of concern are really those where we actually have, they focus on the functional consequences of these mutations. So where there's clear correlation or clear corroborating evidence of an increase in transmissibility or more severe disease. And we we include a number of not only the 3 VOCs that have been identified previously, but we recently escalated 427 and 429, which is the variant first identified in Southern California, to this category, and I'll cover why we did that in a moment. The last category we added was a variant of high consequence. These are viruses that have, where there's clear and definite evidence that prevention measures or medical countermeasures are failing for it. We wanted to make sure that there was a designation on top of variants of concern for those viruses that are potential 5-alarm fires. If you go to the website again, this gives a breakdown of proportions. Again, this is from national surveillance, so the data is likely going to be a little bit lagged. It does give at least what is there in terms of circulating proportions, and it is all in a downloadable format. A lot of these data are being pushed directly into public repositories. So, if there are questions, I'll actually give you a guide of what some of the various CDC and other collections are. If there's interest in actually pulling the sequences yourself and analyzing them, there's definitely opportunity to do so. And we're trying to get as fast as possible in getting those sequences in. One thing to bear in mind, though, is that with many of the variants of concern, we are delaying the submission of those sequences a little bit to public repositories. Whether we'll continue that remains to be seen. But part of the reason for that was that we wanted to make sure that state and local public health departments weren't caught blindsided by the reports of new variants of concern within their jurisdictions. There's obviously a lot of media interest in and around variants, and there was, in many cases, this can be very disruptive to ongoing public health responses if a new variant is identified within a region and if it suddenly makes the news. On the Ryan, I see your comment. I'll address that in a second. On the topic of variants, so again, I direct you to the website. If there's interest, there's a lot of information on how we define these variants and criteria. There's also specific guidance in and around that. And for the main variants of concern, there's actually resources such as BEI links, so you can actually access reference materials if you need to. And I do apologize, Ryan, too. He just reminded me that a lot of the sequencing that's happened in and around Maine has actually been from other federal labs. In a lot of the graphics that I showed earlier, I grouped a lot of federal activities under one umbrella. Jackson Labs, in particular, has been doing a phenomenal job of supporting state and local public health efforts across the region. I probably should have broken that out separately or put it into the state category, Ryan. But this was what happens when you generalize. So I wanted to also give an updated field guide for sequences. There's a number of different sequences starting to pop up in public repositories. If you want to find the NS3 data, those are usually ST, and all of these refers to state or the state of origin. CDC, and then usually a -2- will give you NS3 data. There are a number of NS3 enhanced or special studies, places where the states have sent in additional samples that merit additional characterization. We've included those. They usually have fairly complex looking identifiers. So if you see something that looks a little bit weird, I just showed it in regex format here, but if you see something that looks a little weird, that is what that is. A lot of the baseline surveillance data, they are usually separated out by vendor. Most of these are random selection. For one of these, so LabCorp and Quest are both essentially national reference chains. They pull from a variety of different backgrounds, from large hospitals all the way down to community clinics and everything in between. We also have some providers that essentially provide sequence backend or sequence testing for retail pharmacies across the country. So this is probably going to be a slightly different category of patient, because they're going to be the type of ambulatory patient that can walk into a pharmacy and get tested. But together these do give a very broad view. One advantage, though, is that we're using the exact same identifiers across GISAID, SRA, and other repositories where these are being deposited. So you shouldn't have too much trouble linking them up if there's interest in going through them. So with that, I'm going to switch over again. Actually, this slide highlights it very well. I'm going to switch over and start talking about some of the variants of concern that we've seen, and just give a quick weather report, and then hopefully wrap up and not be too much over time. For this, I just used essentially the state and local submissions to GISAID. Part of the reason I did that is that most states, most federal sequencing activities are conducting essentially unbiased surveillance at this point. So the data is much more likely to be more representative. So this incorporates not only some of the baseline data and NS3 data, but also a lot of the broad scale surveillance sequencing that's happening in states across the country. As you can see, the proportion of, in blue here is B117, that's increasing fairly regularly, as we've seen in a number of other countries. But the overall data set is about 100,000 sequences that I'll be drawing from here. B117, again, I take this last data point with a grain of salt, just because the denominator is probably not as big as it could be, but it's definitely trending upwards in a lot of the states. One thing to note is that it definitely doesn't have the same sorts of kinetics in all different backgrounds, all different environments. Some states we're seeing very rapid expansion of B117. Others are a little bit more gradual. However, it is a fairly aggressive variant, based on what we've seen internationally in other countries that have tracked its entry and emergence. It is certainly following the same type of pattern across the United States. For B1351, the proportions that we're seeing right now are much more limited, and a lot of that, at least initially, was really heavily centered on the East Coast of the United States. We are starting to see more sporadic cases, evidence of community spread across the U.S., and state and local surveillance is starting to pick this up in larger numbers. So, I expect that this chart will change over time. I think we're just on the very flat part of the curve. And on the even flatter part of the curve is the P1 variant, which for the most part has just been reported or deposited sporadically. A lot of that has been fairly recent, but we are seeing it in an increasing number of states. So I think that will change over time. So I did want to spend a moment before we close talking about B1.427.429. There was a decision made to escalate that to a variant of concern, at least within the United States. Part of the reason for that was, there was at least some emerging evidence to suggest a modest increased transmissibility. And it is a virus that we see at fairly high proportions across the United States, especially with a fairly heavy center of gravity towards the Western United States, where in California it's probably close to 50% of the viruses identified or collected, and in a lot of the Western states it's at least 20 or 25%. The decision really, though, was driven by response to monoclonal antibodies. So for 427.429, which has the L452R spike mutation, there was, based on pseudovirus testing, there was at least 1,000-fold reduction in neutralizing titer for bemnifosbuvir, and similar, although less, about an 8-fold reduction for BAM and EDI. Because this was impacting some of the frontline therapeutic agents, because it was fairly widespread and was impacting potential distribution of those therapeutics, the decision was made. It met all the criteria necessary to be designated a variant of concern, but really is just a lot of that is the national circumstance and the way that we approach it in the United States. I should say that we don't see a lot of 427.429 outside the US. There have been a number of cases. There's also different strategies in terms of how monoclonals are deployed. in other countries. So, the factors that drove this decision domestically within the United States probably don't exist in other countries. The last thing I want to bring up in the interest of time is this genomic epidemiology toolkit that we recently put out. As I said, one of the challenges that we have is how to implement or how to make sure that the U.S. public health workforce has the necessary molecular epi skills to apply these types of data to ongoing investigations and to use it to help inform public health strategy and response. This toolkit is a modular, essentially web-based approach. We've brought in a number of wonderful collaborators, mostly through our state and local public health labs. There's some fantastic folks at CDC, including Mike Wiggand and Nancy Chow and Shatavia Morrison, and a number of people that I'm forgetting that all participated on our end to make this happen. But it really is designed to not only give some technical tools on how to analyze data and how to interpret some of the data that's being handed off by bioinformaticians, but some examples, some worked examples of where this can be most useful. I'm just going to end with a few quick takeaways. I realize I'm already over and I see your camera come on. So yes, this Spheres for us really represents a new model, a new public-private model. I don't think we really anticipated just how big it would grow or how incredible a resource it would become over the course of time. It's been a way of engaging academia and in clinical commercial partners with public health in ways that we really haven't ever been able to accomplish across the US public health system before. And it allows us to leverage a lot of the existing investments that we made. What we're hoping, though, is that it's going to allow us to build on traditional laboratory surveillance methods, use this burgeoning research community as really as a source of innovation and focus, and more importantly, to use to help build a stronger and more resilient public healthcare system. As I said, our future plans really blend the strengths of this collaborative model with what we hope is going to be more of a sustainable centers of excellence or accelerator model that we can build on top of that and use this community to really drive forward into the future. With that, I realize I'm at least 5 minutes over, so I will open to questions.
Oh, thanks so much, Duncan. That was wonderful. Really start to finish. Really enjoyed the latest updates that you shared and really great perspectives. Yeah, so we are a little bit over, but in the interest, just to do the fact we have you here, I'm going to maybe ask you 2 quick questions or questions that we have from the audience. One that's been upvoted to the top here, and I'll get right to it. It's a question for you is that, what are the outstanding major needs to improve the tracking of variants of interest and concern in your kind of perspective?
I think a lot of it is at the fine grain. We're starting to get to a point where we're getting the broad strokes surveillance under control. We're getting at least the broad national picture. But really, to me, the more exciting piece is at the other end of the spectrum, where the data is generated very rapidly, where it's tied very closely to on the ground epidemiology, and where it can be applied almost immediately to help guide public health response and infection control. The other piece is, how do we fold in some of these larger surveillance strategies? Wastewater surveillance, pooled testing, especially as we go forward, and we're not necessarily in the midst of the major part of the curve as we're starting to get into more of the flare-ups and ideally the tail. How can we apply these hybrid strategies to do large-scale surveillance, to use these tools to track down outbreaks as they happen?
Perfect, thanks. And then we'll end with one more question here. And this is a broad question, so take it wherever you'd like. It's, where do you see Spheres in the next 12 months, given some of what you presented today here? So March 31st, 2022, where is it?
So ideally, I think what we'd like to be able to do is, as I said, I think we're looking at ways of making this sustainable. Public health does not necessarily have all the tools necessary to work with academic and private sector partners on this scale. We've used some tools at our disposal, like broad agency announcements, but we're not necessarily a granting agency. There are ways that I think having centers of excellence is an easy model to look at, looking at things like accelerators. But I think what we would like to do is transition this into a broader broader technical community that we can use to focus some of these opportunities in the future that we can use to help direct applied research in and around public health, and ideally extend to other pathogens of concern. I realize that we're under unusual mitigating circumstances in the midst of a pandemic, but there's many different public health challenges that are out there that really are ripe for collaboration and innovation.
Well, with that, I will end our first keynote. That was really great. Thank you for your time today. Really enjoyed that, and thanks for answering some of the questions here. For the questions, we'll just move on to our first lightning talk. So let's thank our speaker. Wonderful talk, and we'll move on to our next session. Thanks again, Duncan. Okay, so I'm going to share my screen now and get into our first lightning talk session. Let me share the screen. We're running a little bit behind, but we'll keep going here. Okay, so sorry about that. Sorry about that, everyone. My Zoom crashed. So let me try to get back to my screen share. Okay, so if everyone can hear me fine, we're in our first lightning session. I'm gonna have each speaker, so we have 5 talks to get through. These are focusing on SARS-CoV-2 variants, phylogenetics, and structural analyses. Really great talks here, and we're going to just get into it. So I'm going to hand it right over to our first speaker, Ina. Ina, please take it away. And as a reminder, each speaker in the session will introduce themselves.
Thank you so much, Todd. Let me share my screen. Can you see it?
Yep, it's in. Yep, it looks great.
Great. All right. So my name is Irina Malkovich-Berry. I am from Walter Reed Army Institute of Research, and I lead the viral genomics section within the viral diseases branch. Today, I will be talking about comparative recombination analysis of human coronaviruses and implications for the SARS-CoV-2 pandemic. So recombination is a known evolutionary driver in a range of RNA viruses, and it has been observed for many coronaviruses infecting humans. So with the emergence of SARS-CoV-2, there is of course a renewed interest in identifying the role of recombination in the evolution of this and other human coronaviruses. And this is because recombination patterns in circulating seasonal coronaviruses may predict the evolutionary future of SARS-CoV-2. Further down the line, recombination in SARS-CoV-2 may lead to immune escape and may impact molecular diagnostic performance. And more recently, there has been some reports of recombination in SARS-CoV-2. So for this analysis, we got full genomes from the Viper and GISAID databases of coronaviruses. We restricted this to full genomes and excluded those with long stretches of ambiguous base calls and laboratory construct and undated sequences. For SARS-CoV-2, throughout October 2020, we downloaded all available full genomes from GISAID by then and subsampled those because of the large data set. For recombination analysis, we used RDP4 suite that contains several different methods, for detection of recombination. So our results, before I go into our results, I want to show that the recombination events that we detected by RDP4 screening in the coronaviruses were divided into several different tiers. Recombination events detected by 1 to 3 methods were low confidence events. Those detected by 3 or more methods were moderate confidence events, and those detected by 3 or more methods without any other evolutionary process that could possibly explain the recombination signal were deemed high confidence recombination events. We also noticed recombination events that had identical patterns in several multiple genomes. Those would indicate emergence of a recombinant followed by their spread in the human population. And then those with single genomes, those were unique recombination patterns for a genome. So our results showed that all 4 circulating seasonal coronaviruses had recombination with high confidence detected, and also MERS. The highest ones with the highest number of recombination events were NL63 and OC43. For SARS-CoV-1, we only detected 1 event with low confidence, and this was, excuse me, my screen is kind of going a little bit faster. For SARS-CoV-1, we detected 1 low confidence recombination event, and this was because, most probably because that virus circulated for a very short period of time within the human population. And for SARS-CoV-2, we detected 8 moderate confidence recombination events. We further went to see where the breakpoints for these recombination events existed in these viruses. For NL63, OC43, and MERS virus, we noticed that there was a significant preference for recombination breakpoints to appear in the structural genes of these viruses. For the other 2 viruses, we only had 1 recombination event each, so we couldn't determine the statistics on it, but those were in ORF1 AB. And for SARS-CoV-2, the breakpoints in the moderate confidence were found in, half of the breakpoints events were found in the structural genes, and 1/4 was within the S gene. So then what we did is we wanted to build maximum likelihood phylogenetic trees of all the full genomes, including the recombinant genomes that we found. Here I show OC43. 20.3% of the genomes were recombinants here, and multiple genome taxa with multiple genome events are marked in blue, and those in single genome events are yellow. What we can see here, that some of the single genome events actually fell within the multiple genome events clusters, indicating a 2nd generation recombination, so recombination of an already recombinant virus for OC43. Then what we went and did, we estimated root to tip regression of OC43 of all the genomes. And this is to see if there's any temporal signal in the data. And this is important to have for downstream analysis for estimation of times of the origins of the most recent common ancestors. And when including all the genomes on the top panel on the right here, we see that there was the temporal signal was pretty much non-existent. As the line there is is horizontal for evolutionary rate. However, when we removed the genomes that we estimated to be recombinants, the temporal signal was restored. And this other slide shows the same thing for NL63. Here, however, we had 89.2% of the genomes that had some kind of high confidence recombination signal. And here we also saw second generation recombination. Because so many genomes here were recombinant, here instead we looked for a region that was completely void of recombination breakpoints in most of these genomes, and we found such a region. This is figure on the right top side of the slide, and this region was about 7,000 nucleotides long. So when we did rotative regression for these, when including all the genomes, including recombinants, again, there was no temporal signal in the data. However, when we only use the non-recombinant region, on the bottom panel on the right here, we see that the temporal signal was very nicely restored. These results would indicate that these recombinant genomes might have great impact on temporal signal. Recombination is very well known to affect topologies of viruses. So this is not something new, but here we show that by using our method and detecting the high confidence recombination genomes, and when removing those, then the remaining data can indeed be used as the temporal signal is restored. So this is just to show our data for all 4 seasonal coronaviruses, and it just moved again on me. But briefly, what it showed is that the viruses that were most affected by this were, of course, OC43 and NL63, because those had the most recombination events. We then further went ahead to use Bayesian analysis to estimate TMRCAs of these 4 viruses without any recombinant genomes or recombinant regions. And our results were pretty consistent with what has previously been seen in these viruses, previous analysis that had only covered the S gene of these viruses. For 229E, however, our TMRCA for the full genome analysis was 1989. And 229E was first isolated in 1965, around 1965. So what we did here is we went back and did the MCC tree, so Bayesian time analysis of the S gene 229E only, because there were more genomes available here. And then the TMRCA of this virus was estimated to the early 60s. And this tree also showed that some of these older lineages had not been sampled since. And this is consistent with previous showings that 229E experiences consistent lineage extinction and replacement. So this becomes important to take into account when estimated the emergences of these viruses. But current analyses show that currently sampled sequences of these viruses show TMRCAs that are shown here in this table. And also show that the current variation that exists in the coronaviruses and the amount of recombination that we find has come up only in the last about 70 years. So, in conclusion, we show here that recombination is very common in human coronaviruses, circulating human coronaviruses, and that it has arisen fairly recently within their time of circulation in the human population. We also show that this recombination has preference for the structural genes of these viruses. And this may have impact on the evolutionary behavior of SARS-CoV-2 as it continues to spread in the human population, and points to possible preference for the structural genes of recombination in human coronaviruses. It might happen in SARS-CoV-2. So this becomes important to screen for and continue sequencing more SARS-CoV-2 genomes to monitor for appearance of recombination, especially in the structural genes, as this may impact the diagnostic assays, and it may impact immune responses and escape from immunity. And in the end, I would like to thank all the submitting and publishing labs from the GIS-8 for the SARS-CoV-2 genomes that we got to use, and all our collaborators and all our funders. And thank you. I will take questions.
Wonderful. Thanks, Ina. That was great. And thanks for being on time. So I'll maybe field one question for you here from the audience. And the question is, what kind of evidence goes into the recombination confidence calculations? Is it always clear what combination events are rated as high versus low confidence?
Yes. So the RDP4 method has several different, or suite has several different methods that utilize several different approaches to detect recombination. And what we deemed as high confidence was when we had at least 3 different methods, so 3 different approaches, find recombination, the exact same recombination patterns within a data set, plus that recombination could not be explained by any other evolutionary processes. So that is what we deemed as high confidence. And just the fact that when removing those recombinants, the temporal signals were nicely restored tells us that those most probably indeed were the genomes that were messing up the topology and the temporal signal.
Great, thanks so much. In the interest of time, we'll move on to our next speaker. That was wonderful way to start our session. Let's move on to our next speaker. And again, each speaker will introduce himself. And so, Pili, please go ahead and activate your camera and get your presentation shared. Thank you. Sorry, you're muted.
Sorry, can you see my presentation?
Yes.
Okay, so hi, I'm Pilar Gallego, and first of all, thank you very much for inviting me to speak today. I'm going to present a paper we are working on about intrahost genomic variation in SARS-CoV-2 transmission clusters in northwestern Spain. First, I will briefly introduce myself. I'm a first-year PhD student at the University of Vigo in Spain in the laboratory of David Posada at the Center for Biomedical Research, and I'm working on SARS-CoV-2 genomic epidemiology. Our group's wet lab is centered in NGS and single-cell genomics, and the dry lab is focused on computational evolutionary biology and cancer genomics. The objectives of this study are the delimitation of clusters with no previous epidemiological information about the samples, understand the transmission routes, and estimate the transmission bottleneck size. We started all of this thanks to Trengen's lab, as they study intrahost and interhost variability of SARS-CoV-2 between patients, and saw that intrahost variability was shared and could help understand transmission histories. My supervisor collaborated in a paper where they didn't have epidemiological information of the samples, so we then decided with them to conduct this study using a set of samples for which we had some more information. We initially had 49 samples, of 3 of them that corresponded with the highest CT values failed when sequencing. So we worked with the remaining 46 samples. Those belong to 10 non-epidemiological clusters, and 2 of them came from nursing homes. And so we didn't have information about the first introduction of the virus, while the other 8 belong to families that live together or that had been infected one to another in the same social event. So for example, in the case of cluster D, patient D1 came from a travel and infected some of the other members of the family during a birthday party. And in the case of cluster E, we had 2 possibilities, as it consisted of 2 brothers that came from traveling and infected their mom. Then, for example, cluster I was composed of 2 girls that were infected during a birthday party from the same person and then infected their respective parents. We followed the Arctic sequencing protocol version 3, and we started with nasopharyngeal exudates, from which we extracted the viral RNA, And 2 independent researchers performed retrotranscription from RNA from the same patient in order to obtain technical replicates to work with during the whole process. Apart from working with replicates and in order to work with samples that we were all able to say that hadn't suffered cross-contamination, we added a unique spiking of CMAs to its retrotranscription reaction. And this spiking, it would be a sequence of a gene of maize to which we added a different forward and reverse barcode in its end, so no sample shares the same combination of barcodes with another. So we can then map the reads to original sequences and count how many and to which barcodes they map. And this allows us to confirm that the variability shared between samples was not due to cross-contamination, as we only detected the barcode that corresponded to each sample. And we sequenced the 98 libraries into high output runs on an Illumina Mini-Seq, and the mean depth by sample obtained, taking into account both replicates, was very high, around 6,000x. For the bioinformatic analysis, we used the IBAR pipeline, and variant calling was performed with IBAR for its replicates, and then the results were filtered. So we only consider a mutation that was called and considered true for both replicates. And apart from this, they had to present an allele frequency above 0.02 and not be placed in a known homoplastic position. And we lower the IBAR default allele frequency threshold, considering the high depth of coverage we had per sample. And therefore, we consider intrahost variants, those with an allele frequency between 0.02 and 0.98. And about that threshold, we consider a variant to be fixed. And there are some cases when a variant can be fixed in a patient and intrahost in another. Merging the map trim reads from both replicates, we also obtain the consensus sequence for each sample that has then been passed to next clade and pangolin in order to obtain the clade and lineages. We obtain a high number of shared intrahost mutations between non-related samples, and most of them appear at very low frequencies and were predominantly indels. And some of these mutations appear in samples that were collected with months of differences between them, and we're also calling other datasets available at our laboratory, as well as in some SRA samples, and when doing the variant calling with other callers such as LoFrec. And we are not sure what is the reason these mutations keep being called, and if all of them are due to systematic errors of the pipeline or some of them are hotspots. But we deem them uninformative for the purposes of this article, as we cannot assume they have been transmitted from one sample to another and thus cannot be used to infer transmission. And so we filter those interhost mutations that appear in more than 2 clusters. After this filtering step, our samples present a mean of between 2 and 3 intrahost mutations and a mean of 12 fixed mutations. The easiest way to see if clusters could be differentiated is looking at the phylogenetic tree, and this shows us that in some cases clusters could be in fact differentiated from the rest as they are monophyletic and with high bootstrap, but in others it wouldn't be so easy, and without previous epidemiological information we wouldn't know where to put the limit. In this tree, we can also see the difference between the earlier clusters from March and April. So they belong to the first wave of the disease in our country, clade 28 lineage B.1, and the later ones that runs from August to October and belong to cluster 20 lineage B.1.177, which is very common in Spain. And we follow existing methods of cluster delimitation and transmission history study as the ones included in Warby et al., between which we can differentiate those based exclusively on shared intrahost variability and those reliant on consensus sequences. The ones based on shared intrahost variability were applied to 2 datasets, the original one in the left and the filter one on the right. So we can see that while before filtering the variant, it was impossible to distinguish anything, all samples were related and most of them share a high number of intrahost variants with a lot of other samples. When using the filtered datasets, we do not obtain anything better. And this is due to the fact that most samples do not even present any intrahost variant after filtering, or at least not any share with another sample, and those that do are sometimes from different clusters. On the other hand, when using methods based on genetic distances, meaning differences in variance above the 50% threshold that are going to be included in the consensus sequence, we can observe that the clusters are separated near perfectly. And the only error with respect to our information is that cluster D, that is the largest one with 10 samples, is divided in 2 different clusters, which is explained by the fact that the 3 samples that appear separated from the rest serve to additional SNPs among them. As we can see, using any of those methods to determine the transmission histories inside each cluster proved to be inefficient. And this is due to the fact that sequences are too similar to each other. So most of the time, the logic of assigning the donor as the sample with the highest number of observed variants or the minimum distance to our sample gives us as result multiple arrows that go to different samples and in both directions. And other methods more sophisticated to infer transmission histories based on Bayesian inferences, for example, as Transfilo or others, could also not be applied to our data, as they are too similar and too near in time for these methods. Using these methods based only on genomic information is not enough, so we relied also on the available epidemiological information to establish true transmission pairs. And in some cases, when epidemiological information alone was also not enough, we combined both approaches, and we have, for example, the case of cluster D, where we know that the primary case is D1, but it doesn't mean that this patient infected all the rest of the cluster. So it's clear D1, D3, and D6 are related as they share 2 additional mutations at very high frequencies, fixed or nullly fixed, that do not appear in the rest of the cluster as at such high frequencies or do not appear at all. So we would have pairs D1 to D3 and D1 to D6. Then we could also infer the pair D3 to D2, as D3 presents 2 variants almost fixed that have a very low frequency in D2 and 1 low frequency variant that is above 50% in D2. The situation is strange as they seem to reverse the frequencies, but even if we consider the transmission to go in the other direction, it would be the same problem and also contradicting our epidemiological information. But it seems clear that both samples are connected. And then we would have D2 to D10, as they share 1 variant at a high frequency and another variant with allele frequency 0.88 in D2 and 0.11 in D10. For cluster F, on the other hand, we know the index case came from out of the community, but we lack that sample, so we could only infer the transmission event F1 to F3, as they share 1 fixed and 1 low frequency variant, and F1 has multiple private variants at low frequency, which means it's more likely the donor of the pair, as it would be easier for them to disappear than to generate de novo if F1 was the recipient. And lastly, the bottleneck size, meaning the size of the viral population transferred from the donor to the recipient host, was estimated using a beta binomial model of Sobel, Leonard et al. And assumes that you are estimating using true donor-recipient pairs. This method also assumes that the intrahost variants detected are real and didn't arise de novo in the different patients, and this is why we use our filter set of variants. And we found a very small number of trustful shared intrahost variants between transmission pairs, 1 on average. Because of this, our bottleneck size estimations are also small, as they range from 1 to 17, with confidence intervals ranging from 0 to 45. And these small bottleneck sizes are concordant with previous findings of other authors, although they disagree with the first estimations that are being reviewed. And in conclusion, cluster delimitation is possible when using consensus sequences, but not when using only interhost variability, as it's very reduced, and transmission history study is difficult without previous epidemiological information. And the bottleneck size of chain is very low. And well, finally, we would like to acknowledge the collaboration of Triangle Lab from Rice University, as they collaborated with the computational analysis and microbiology department of TUI that collected the samples and epidemiological information. And thank you very much for your attention.
Yeah, great talk. Thanks so much. And again, thanks for being right on time. So I have one question for you, just so we can keep moving along here. have you tried this with the British variant, and do you think it would be possible, or do you think the bottleneck size would be higher in that case?
We haven't tried yet, but we have some more samples right now from different places in our community. Most of them are from the British variant, that is the most frequent one right now in the region. So we are going to try and compute it to see if there are any differences in the bottleneck size.
Great. I think we'll just leave it at that, just because again we're such a tight session here. Thanks again. Great talk, and let's move on to our next speaker here, and Nydia. So please turn your camera and share your presentation and take it away.
Hello everyone. Let me just share my screen here. Can you see my screen?
Yep, looks great.
Thank you. So good morning, everyone. My name is Nydia Trovão. I'm a molecular epidemiologist at the National Institutes of Health, and I just want to take the opportunity to thank Covert for the invite to speak at the symposium. So in this talk, my aim is to define what is a variant and how they emerge. More specifically, I want to focus on the impact of variants on transmissibility, their epidemiological impact on infection rates, their susceptibility to vaccines, and the mechanisms that might drive their emergence. As we know, SARS-CoV-2 is an RNA virus, and despite its proofreading mechanism, during replication, mutations are generated. Those mutations will be accumulated over time, and when a set of genomes that contains a particular set of mutations starts circulating and growing in frequency in the population as a distinct group of viruses, they are called variants. Fortunately, not all mutations generated during the replication cycle are fixed in the viral population. Mutations appear in the viral population, with some being advantageous and getting fixed, as we can see here in the green line. Some are neutral and vary in their frequency in the population, depending on the different pressures, as we can see by the blue lines. And the deleterious mutations that decrease the fitness of the virus tend to disappear, as depicted in red. So through genomic surveillance and epidemiology, we can gain insight on the circulating viral diversity by looking at different mutations circulating at a particular point in time. For example, in time 20, here, the green, the blue, and the red mutations circulate simultaneously, as we can see. So there is more diversity here than, for example, in time 85, where only the red mutation is circulating. So the viral diversity depends on the selective pressures that the virus is under and what is maintained through time in the population. So these mutations can appear anywhere in the viral genome. However, considering that the spike protein is responsible for the viral attachment and entry in the cell, mutations that happen in the spike are of particular interest because they may have implications on the viral transmissibility, morbidity, and mortality, the ability to evade detection by diagnostic tests, and the ability to evade immunity caused by infections and reinfection, therapeutic antibody neutralizing antibodies, or even vaccines. So a variant of interest pertains to genomes with a lot of mutations comparing to their parent lineages, particularly mutations with known clinical or epidemiological effects. And here we are also looking if there is an increase on their frequency, if the mutations appear to have emerged independently from those described in other locations or settings, what is called convergent evolution, or if they tend to spread in locations with high levels of immunity, meaning they are escaping immunity. So by sampling and sequencing viral genomes over time and space, we are able to reconstruct the virus, if the virus is evolving and how it's spreading around the world. The pandemic started with viruses that were part of lineage A, here in purple in the phylogenetic tree, also called by the next strain nomenclature system as clade 19. However, in early 2020, we observed that viruses with a mutation from aspartate to glycine in position 614 of the spike started to also circulate and increase in their frequency to later become the dominant circulating variant denominated the B1 lineage, or clade 20, depicted here in green in the phylogenetic tree. And further studies demonstrated that the success of lineage B1 was due to the moderate increase in transmissibility conferred by the D614G mutation. Of course, more recently, as we all know, in late 2020, 3 variants of concern have emerged in 3 different continents. The UK reported the circulation of a variant that carried a mutation in position 501, along with other mutations. And of course, these were concerning news, as preliminary studies suggested that this 501 mutation confers substantial increase in viral transmissibility comparatively to the previously detected B1 lineage, because it helps the virus to bind more tightly to human cells. And shortly after, other lineages sharing the same mutation on position 501, along with varied mutation constellations, started to also being reported in South Africa and in Brazil. And besides their increase in infectiousness, these other variants that carry mutations in position 484 in the spike may also help the virus avoid some kinds of antibodies and also appear less susceptible to some vaccines. So, as I was mentioning, genomic epidemiology efforts like those led by the Next Strain team provide enormous help on identifying and tracing the spatial history of such mutations. Here in this tree, the green dots represent the viral co-circulating diversity, whereas the yellow, the orange, and the blue dots represent the variants firstly detected in the UK, South Africa, and Brazil. And these have been classified as different lineages and are now referred as B117, B1351, and P1 lineages, respectively. And what I want to note here is that despite sharing these mutations, these lineages appear to have emerged in different locations in the world. And after meticulous phylogenetic analysis, this is shown by forming different clades in the tree. So this suggests that there is such fitness advantage to these mutations that they were generated and fixed independently through distinct evolutionary pathways. So another commonality across these variants is that they appear to be evolving faster, meaning that the accumulation of mutations is faster than their ancestral, that in their ancestral lineages or those that are currently co-circulating. So when studying the evolutionary history of pathogens, it is important to estimate the rate at which they accumulate mutations, because this gives us an indication of how soon control strategies like vaccines need to be updated. And previous lineages of SARS-CoV-2, here depicted in blue, accumulated mutations slowly at rates comparable to that of seasonal coronaviruses OC43 and 229E. However, the recent variants here in red appear to be acquiring mutations at rates even higher than those of influenza A virus subtype H3 and 2, for which we need to update vaccines every year. However, besides these 3 variants of concern, many other variants have been described. Almost actually simultaneous to the detection of the UK, the Brazil, and the South African variants, the California variant was identified in Southern California, with most cases occurring in LA. And what they noticed was a switch between the predominant circulating lineage to 36% of the cases being infected with these variants. So it has multiple mutations in the spike protein, and so it was considered initially as a variant of interest, but further studies established that it's actually 20% more contagious, and so it was bumped as a variant of concern. In New York, for example, a genomic surveillance effort between late December and mid-February this year showed a steady increase in the detection rate of two independent sublineages carrying mutations that we know can evade some antibody immunity in a part of the country with high levels of immunity already. So that is still under investigation. And just a few days ago, on a large genomic surveillance effort in India, they identified the circulation of the UK, the Brazilian, and the South African variants in the country. But most noteworthy was a variant that had two of the most important mutations for the Brazil and California variants. And this could be possibly evidence of convergent evolution or recombination, given the co-circulation of the precursor lineages in a location with high infection rates. So as we continue to trace the origins and evolution of these variants, we question about the mechanisms that might have led to their emergence. And of course, it is reasonable to assume that in places with high infection rates, there is more opportunity for viruses to generate mutations that might confer increased viral fitness. However, there are other mechanisms that might also promote these situations. For example, in Boston, an immunocompromised patient with COVID-19 was sampled at 4 different times and had his viruses removed. And they observed that the subimmune response, instead of clearing the viruses, drove the generation and selection of a viral population with the same mutations observed in the recently identified variants. So this is a cautionary tale about the importance of not only doing population-wide surveillance and sequencing, of course, but also following the viral profiles of hospitalized patients with long COVID. So to end on a positive note, I just want to mention that all vaccines available have shown excellent efficacy against severe disease and are a bit less potent, but still effective against the variants. And nonetheless, pharma companies are already working on developing vaccine boosters and variant-specific vaccines to better protect against variants of concern. So with that, I'd like to thank the Covert team and you for your attention, and I'd be happy to answer any of your questions.
Wonderful, great, very, very timely topic for sure, and very nicely presented, Nydia. So I'll go to the questions. We may have time for 2. The top voted question that I'll ask is, the question is, what is the mechanism for how the new strains are mutating more rapidly versus parent strains, is the question. And the parent strain was in quote unquote, but I think the general idea here is
the precursor.
Precursor and how the variants are appearing so rapidly.
So we are still not sure. I don't think, or at least no studies have yet shown any changes in the polymerase function. It might just be that these mutations confer such advantageous fitness that they are just more easily fixed in the population. Great.
Okay, and then I think we have time for one more. And then one question just popped up. So one is, I'll go to is the California variant responding to vaccines? And then just the follow up there, and this will be it, is where should we be looking for these new variants of potential interest or concern? So just 2-part question there.
Okay, the California variant does have a mutation that increases in transmissibility, but I don't think there's any study yet confirming that there are any effects on immunity. So even natural infection, escaping our natural infection with other lineages or vaccines. And in terms of where these new variants might be popping up, for sure, as we were just looking in locations with high infection rates. And there is a big need for augmenting and increasing surveillance in locations with these high infection rates that are undersampled. For example, many locations in South America, Africa, and of course, the other mechanism, the other location where these mutations might emerge is in immune compromised patients that have lung disease. And so the suboptimal immune pressure might lead to the accumulation and the emergence of these mutations. So there's a big gap on surveillance on these two systems for sure.
Wonderful. Thanks so much, Nydia. Thanks again for your nice talk. We'll move on to our next speaker just to keep things more or less on time. Mauricio, please go ahead and introduce yourself and share your presentation.
Sure. Thank you, Todd. Can you see my screen?
Yep, looks great.
Yeah. Okay, perfect. So thanks for having me. It's a great honor and pleasure to be here today. My name is Mauricio. I'm a postdoctoral research associate at Covert Lab in Department of Computer Science at Rice University. I will present my work entitled Structural Analysis Tools for Drug Development and Target Identification Against SARS-CoV-2. And I split this work in two different projects. So the first one is about SARS-ARENA. This is a sequence and structure-based identification tool for SARS-derived conserved peptides. And the second project is more in the field of molecular docking. It's called COVID. This is an ensemble docking approach for SARS-CoV-2 drug screening. So in respect to the first project, just a little bit of background. The COVID-19 pandemic is caused by a novel strain of coronavirus with a fast-growing number of new variants and confirmed cases worldwide, as we can see in the presentations. And most vaccine development efforts focus on SARS-CoV-2 envelope proteins. But one setback on that is that these proteins are more susceptible to selection pressure and then more prone to mutations. So one strategy to induce a broader long-term unprotective cellular immunity is to look to proteins that are less susceptible to selection pressure, such as the nucleocapsid protein. So this is just a representation of a work from 2006, where the authors recover immune cells from SARS-recovered patients, and they stimulate the cells with N protein peptides. Then, after that, they observe the immune response triggered by these presentations, and they observed that CD8 and CD4 T cells were responsible for trigger immune response against these peptides. And it was also interesting to observe that depending on the region of the N protein, the immune response was different. The region is represented here for these different peptide pools. So this project one is focused more on the sequence conservation analysis, specifically on the N protein, and on the structural analysis. And on the structural analysis, we are focusing on the presentation of these peptides by a molecule called MHC1 for TCRs in CD8 cells. Just to give you an idea of the structure, this is what we are working with. You can see here in blue and in gray the MHC molecule, and this MHC molecule will present the peptide for a TCR represented here by the green and the yellow colors. And what the TCR will observe is a peptide in a cleft inside a molecule called MHC. And what the TCR will actually observe is a surface with different charges, from negative to positive charges. And going to the literature, we can also observe that some conserved regions in N protein, specifically one immunogenic region that I'm showing here. Here we have two peptide sequences that are very similar to each other, but they have some differences. But one important fact here is that these two sequences, despite being for different viruses, they have hotspots in the surface of the PMHC. And these hotspots are being recognized by the same TCR. So this is a way where we can use this tool to evaluate new targets against SARS-CoV-2. So our idea with this project is to provide to the user two workflows. The first workflow is a pipeline to analyze N protein sequences retrieved from NCBI, and then retrieve the peptides that are in most conserved regions. And these peptides that are in most conserved regions, after we pick them from the workflow one, they can be used in the workflow two in a structure-based analysis, where the user will be able to model these peptides according to a MHC of interest, and then he or she can use these to identify hotspots for T cell response. These two workflows were created using Jupyter Notebook, and also Docker and other softwares that are all inside Docker images, so the user don't need to install each one of these programs. He can use these workflows in a straightforward manner. So just to give you an idea of the workflow one, how we set that, the first part is to fetch proteins from NCBI datasets. Then our algorithm will make a sequence alignment using the muscle algorithm. And then we will compute a conservation score in this alignment. And what the user will have is this graph where he or she can set a peptide length from 8 to 12 residues, and also can set a conservation threshold, in this case 85%. So all the peptides that are above this line are 85% conserved inside our alignment. And the user don't need to be concerned about looking to these peptides in the graph, because we will select these peptides and read in a file. And this file with all the peptides can be used in the next workflow that is more structure-based workflow. And in the workflow 2, the first input will be the peptide sequence. These are the ligands that the user recovered from workflow 1. And the user can also choose which MHC sequence he wants to use to analyze these peptides in the context of this MHC. So the first part, we make a peptide selection, because not all peptides will have a strong binding for these MHC sequences. So we use this sequence-based predictor to select only the peptides that has higher affinity for the MHCs. Then we model these MHCs, the three-dimensional structure of the MHC, and then we perform a molecular docking approach to put these peptides inside the cleft of the MHCs. And using scoring functions that we develop, we can rank these PMHCs and provide to the user a structure of the peptide and MHC to be analyzed. SARS Arena is a work in progress. We are just finalizing some things on workflow 2. But this pipeline will allow users to search and analyze for conserved residues in SARS-CoV-2 proteins, and the 3D complex can be used in surface analysis to search for shared structure patterns. In the project 2, I'll talk about COVID. This is more related to molecular docking. So this is an ensemble docking approach for SARS-CoV-2 drug screening. So just a little of a background on that. We know that each SARS-CoV-2 protein has a special role in the virus adaptation, so each protein can be used as a target for drug development. And one way to assess this is using molecular docking for drug screening. But one of the things that we are observing is that most studies are not considering the receptor flexibility in the search for these ligands. So this is kind of a problem, because as you can see here, we have in green the main protease, one of the proteins from SARS-CoV-2, and in blue a ligand in the catalytic site of this main protease. This is one structure only. But if you go to the protein data bank and we retrieve all the possible main proteases that were crystallographed, and we observe the catalytic site, we will see that we have a lot of variation on the residues around these binding sites. So our idea with COVID is provide to the user a way to analyze, search for new ligands using the information of these variability conformations right here. So the user can do that, can search for conformations, but this is not an easy task. So the idea of COVID first is to provide a set of pre-computed conformations for different SARS-CoV-2 proteins, and these proteins can be used as input receptor for drug screening. So the user don't have to have expertise on tools used to generate these conformations. We will also select the conformations that are the most variable ones between among all the conformations, using principal component analysis and dimensionality reduction methods. And at the end, we will implement an algorithm for ensemble docking, and we will give to the user a web server where he or she can upload the ligand and search and make its search through a web server. So just about the ensemble generation, how we did that. So first we search for SARS-CoV-2 proteins in protein data bank, And we identified three main targets: the main protease, represented by M-Pro, papain-like protease, the PL-Pro, and the RNA-dependent RNA polymerase, represent for RDRP. I will use just the acronyms from now on. And we have a lot more of structures of main protease, and a little bit, and not so much from these two. And also wanted to highlight here that for main protease, we have two binding sites, one binding site that we call catalytic site and one allosteric site. And for PL-Pro and RDRP, we have only the catalytic site. So we don't want to use all the structures because this is time consuming for the tool. So we need to select the most variable, the conformations with higher variability among these structures. So one of the things that we made was dimensionality reduction and clustering using k-means to select the most variable ones. But we also wanted to analyze these proteins in a context that would be like something that the protein is in vitro. So we wanted to generate more conformations, not only rely on the conformations from protein data banks. So we perform molecular dynamic studies using either charm or Gromos force field. We were able to generate roughly 100,000 conformations. And from these 100,000 conformations, we also select the most variable ones to be included in our ensemble. So at the end, we will have three different ensembles that we call crystal ensemble, charm ensemble, and Gromos ensemble. The crystal ensemble with the conformations coming from the protein data bank, and these two with the conformations coming from molecular dynamics. And as we can see here, at the beginning we had in gray, these are the number of conformations that we had. And after performing the dimensionality reduction, we were able to retrieve less proteins. And this is ideal if you wanted to make a tool that will be accurate and also will not take longer to provide the results for the user. This is just a visualization of one of the ensembles. This is the crystal ensemble for the main protease. So you can see here that we have all the conformations aligned to each other, and we have high variability in the catalytic binding site or in the allosteric binding site. So we observe this variability after performing the search for these conformations. And as I told you before, the COVID is already available as a web server. All the user needs to do is to upload a ligand, a structure of a ligand. He or she can prepare this ligand, then choose the receptor that will be used and the ensemble right here, and then submit the job. And the user can look for this job inside the browser or even download the results to analyze in its own computer. So the COVID offers a ready-to-use solution for researchers to account for protein flexibility while testing compounds against SARS-CoV-2 proteins, and also allows users to run ensemble docking experiments without additional burden of time-consuming simulations required for ensemble generation and docking preparation. So in summary, what I presented to you was the SARS Arena and the COVID, two different projects from our lab. The first one, work in progress. The second one, it is already finalized. If you wanted to use these tools or if you wanted to collaborate with us, if you have any suggestions, please feel free to contact me or contact Dr. Lydia Kavraki. We will happily talk to you about this. So thank you so much for listening today. These are my acknowledgements, and I'm open for questions. Thank you.
Great. Thanks so much, Mauricio. Really nice work. We have a couple questions here. Let's see if we can get to both of them. So first question I'll ask that was from the audience right here. For DINK COVID ensemble docking, how was the data validated?
Yeah, the data was validated using the binding energy from the molecular docking approach. And also we rely on the delta G experimental and IC50 values from in vitro data for different data sets. So we had different data sets of different ligands for each one of these proteins. we had these values, delta G experimental or IC50, and we correlate these values with the binding energy retrieved by for each one of the conformations that we got using our Trian samples. So the validation was basically a correlation between experimental values and the binding energy provided by the program.
Excellent, thanks. And then just a final question here. How many HLA alleles can be used in your workflow 2 that you showed?
As many as you wish, actually. You can use all the, this is more focused for humans, so all the human HLAs are available for use. You just need the sequence, and our program will model the sequence based on crystal structures that are available for the most closer HLAs, for homology ones.
Okay, thanks so much. We'll move on to the next speaker. Great talk again. So let's go to our final speaker in the session.
Thanks so much.
And so Paco, please introduce yourself and take us, wrap up our session for us.
Thank you very much for your invitation. So my name is Francisco Anguita. I am assistant professor in the Faculty of Medicine in Lisbon. Hope that you are okay. You are watching my presentation now. Sorry. As I told you, I'm assistant professor at the Faculty of Medicine in Lisbon, in this building here, and I'm also a senior researcher in the Institute of Molecular Medicine, which is part of the same university, and both institutions are also joined to this big building, which is the main university hospital in Lisbon in Portugal. So during this short presentation, I'm going to tell you something about a specific family of RNAs, which is named the long non-coding RNAs. And this family of non-coding RNAs is originated from a wide range of genes that are distributed along the genome. And as you can see here in the last gene code version, the number of long non-coding RNA genes is very close to the number of protein coding genes. So these long non-coding RNAs, as other non-coding RNAs, are regulators of the genomic output, and they can act either at the nucleus or at the cytoplasmic level. So just for you not to get very bored about, I will summarize this complicated scheme where you see all the mechanisms that these molecules use by telling you that these molecules are mainly skeletons. They are scaffolds. They are scaffolds of molecular complexes, and they are able to interact with many other partners like proteins, other RNAs, and also the chromatin. So in Covert, we analyze many kinds of data, but I was particularly interested in the analysis of a transcriptomic dataset that has been collected by the consortium. And this is roughly composed by 700 samples from patients from nasopharyngeal swabs. And this you can see the distribution of the samples, of the clinical samples. 50% of them are not infected, but along the samples that are infected, among the patients that are infected, we can distinguish different levels of infections with SARS-CoV-2 by using the qPCR data that we had from the diagnosis. And when we analyzed the transcriptomic data of these patients, it was very striking that we saw clear upregulated expression patterns of all the genes. When we analyze in details these families of genes, we conclude that a significant proportion of the genes that were upregulated, especially in those patients with high viral loads, is represented by long non-coding RNAs. So more detailed analysis allow us to determine that these long non-coding RNAs were mainly antisense, but we also had some other families like upstream from protein coding genes and also divergent transcripts. So just for you to realize how striking is the pattern of overexpression of genes, here you can see at the genomic level how it looks like. So you can see in circles the long non-coding RNAs and in squares the coding transcripts. And you can see that the majority of them are upregulated upon SARS-CoV-2 infection. When we analyze specifically the pattern of long non-coding RNA expression, we found a bunch of these transcripts that were correlated with the viral load. And here you can see a heat map representing the expression levels in the different groups of patients. And we also realized that some of them were strongly correlated in their expression levels, forming something that we like to call clusters of these long non-coding RNAs. So, but more interestingly, we can see here that when we compare the upregulated long non-coding RNAs during SARS-CoV-2 infection with the patients that were infected with other respiratory viruses, we can see that there is a clear fingerprint represented by around 100 different long non-coding RNAs that were only detected in the patients infected with SARS-CoV-2. So we decided to move over by using this group of long non-coding RNAs. Here you can see also the pattern of expression of some of them, some of them 6, in fact, of the more important long non-coding RNAs that we detected across the samples. Okay, and recently we also realized that 2 of these long non-coding RNAs, Mrear and BISPR, were also upregulated in the blood of SARS-CoV-2 patients. So this data has been extracted from this data set from the COVID project. And you can see here clearly how these both non-coding RNAs are detected in the blood of the patients, and they also clearly show an upregulation pattern. So the problem of studying this bunch of regulators is to establish their functions. And for this, we decided to construct something that we used to call RNA-centered regulatory networks. For this, we used this database, which is called NCORI. And basically, the dataset is composed by functional data obtained from wet lab experiments, mainly CLIP experiments, that will show you the interactions between RNAs and other partners, namely proteins and also other non-coding RNAs. So we keep in mind that all this data is validated data, so it's experimentally validated data. So we cross the information of the NCORI database with our bunch of upregulated long non-coding RNAs, and we found very nice conclusions. This is the number of RNA binding proteins that are interacting with some of our upregulated long non-coding RNAs, showing that these RNAs could be involved in this kind of interactions. Moreover, if we look the other way around, you can see here this word cloud showing the more prevalent RNA binding proteins that are interacting with these upregulated long non-coding RNAs. And you can see here some of them like FUS, like UPF, like IGF2, BP1, and BP2 that we will talk about later. Okay, so we decided to propose a model about how these long non-coding RNAs can function during SARS-CoV-2 infection. So we used our experimental data, the NCORI dataset, and also all the relevant bibliographic information. We came across with this scheme, which represents basically a regulatory network based on the presence of 6 upregulated long non-coding RNAs that you will see represented by squares, and the interactions with RNA binding proteins. The size of these symbols is proportional to the number of interactions that are established. So we choose these 6 long non-coding RNAs because they showed the higher correlation levels in our dataset. So if you look at the right-hand side of the slide, you can see something represented within a square. And this square has been taken out from this paper from Schmidt and co-workers that appear very recently in Nature Microbiology. And this paper shows how the SARS-CoV-2 RNA plus genome is able to recruit RNA binding proteins. And some of the RNA binding proteins that you can see here in yellow that are recruited by the SARS-CoV-2 genome are also interacting with our long non-coding RNAs. So it is a very complex network that we will need to understand more in detail, but it suggests that we are looking at an interplay between RNA binding proteins, the SARS-CoV-2 genome, and also the long non-coding RNAs. I have to tell you as well that this RNA BISPR and this Mrear have been already described as main regulators of the interferon response. Okay, so in the meantime, actually, this last Monday, Cell published this wonderful paper showing the structure of the SARS-CoV-2 genome, so the 3D structure of the SARS-CoV-2 genome, and the identification of some hotspots for the binding of RNA binding proteins. These authors actually claim that some of the spots were or will be possible to be druggable spots. So, and they found some very interesting spots. I don't have time to comment to you, but they appear either in the 5' UTR of the genome and also across the whole SARS-CoV-2 genome. And they validate some of these binding sites. Some of the binding sites that they found are exactly binding sites for the protein that we pinpoint in the scheme that you saw before. So we want to propose our model, and this is the model that shows the key players, just a few of them, but I feel that they are the more important ones. So where the SARS-CoV-2 injects the genome in the cell, and this genome acts as a scaffold for many RNA binding proteins. These RNA binding proteins are also in connection, are functionally related with long non-coding RNAs, and they can control together the response of the cell to the virus. One of the most important proteins is this one, UPF1, which has been already described to be involved in the regulation of the nonsense mediated decay, so the mRNA surveillance, mainly that the virus repress this protein to actually repress this quality control mechanism of the cell to produce a lot of RNA and a lot of proteins. So this could be a potential druggable target. And also very interesting, the presence of this helicase and other helicases, and this IGF2, BP1 and 2, that if you remember, I will go back, if you remember, was a spot found here in the paper published last Monday, okay, as a druggable spot. So, and these proteins also interact with the long non-coding RNAs that I show you that are involved in the regulation of the interferon response. So, in conclusion, we think that this could be an interesting model to understand at least the difference between the responses in several patients to SARS-CoV-2. And just for keeping on time, I would like to go to the acknowledgement. I would like to acknowledge everybody in the non-coding RNA subgroup of Covert, and of course, to you for your attention. Thank you very much.
That was great. Thanks so much. Really, really wonderful. And it's a beautiful picture there. So we have one question to get started. And given the time limitations, maybe we'll limit to one, but one question I'll ask is, in the case of strong involvement of some of the described link RNAs in the outcome of COVID-19 infection, as you described, do you think it would be better if it would be better to drug the link RNAs or the RNA binding proteins associated with it? So which one do you think is maybe the better strategy?
Yeah, I think the best or the easiest way to drug something is to drug the proteins, of course. But I think that these long non-coding RNAs have a lot of potential in terms of druggability. Maybe you can think also about the interactions between the protein and the long non-coding RNA. Okay, so you can see here in this scheme that there is some sort of equilibrium between some competition between the SARS-CoV-2 genome and the proteins that are bound to the genome and also interacting with the long non-coding RNAs. So I think that the proteins will be a nice spot, but also some of the long non-coding RNAs. But of course, we need still more study to conclude that.
Yep, great. I think that's wonderful. I mean, I think there's a couple other questions I could ask, but just in the interest of time to kind of get us back on schedule here, I think I'll just thank you again for the really nice presentation, and that concludes our first lightning talk session. So I would like to thank all of the speakers, all the 5 speakers. That was a wonderful survey of some of the great research within and exterior to Covert that's going on specific to SARS-CoV-2. And with that, I would like to ask our second keynote speaker to turn on his camera, please, and we'll get started with our second keynote. And for all the other panelists, please turn off your video and we'll get started here. So I'm just going to introduce, feel free Dinesh to share a slide if you'd like. I'll just introduce yourself and introduce you, and then we'll get started. So as for our second keynote, we'll be hearing from Dr. Dinesh Agarwal. And Dr. Agarwal is currently working at COG UK and Public Health England to characterize SARS-CoV-2 importations. He has a special interest in transmission within care homes and universities as part of the Cambridge University screening study. Dr. Agarwal completed his medical degree from UCL and the University of Cambridge in 2013. He is an infectious disease and microbiology specialist, training in the London Deanery, and was awarded a Wellcome clinical PhD fellowship in 2020 to understand the biological basis of Staphylococcus aureus carriage under the supervision of Professor Peacock, Professor Dinesh, and Dr. Harrison at the University of Cambridge and the Wellcome Sanger Institute. The talk title today is on the screen. It's Sequencing to Understand Transmission at a Regional and National Scales in the UK. And I'd like to announce again, thank you so much for presenting to us today due to the slight change in schedule for Professor Peacock. And with that, very excited to hear all about your talk today. So please take it away.
Hi, thanks so much for that, Todd. First of all, I'd like to apologise on behalf of Prof. Peacock, who had to cancel her talk at very short notice. And thanks for welcoming me. As you said, I'm Dinesh, Wellcome Clinical PhD Fellow, and hopefully I can interest you with my understanding of the use of sequencing to understand transmission at regional and national scales in the UK. Work that's been largely facilitated by COG UK and their national surveillance programme, which I know is well publicised globally. So I'll be touching upon 4 important settings I've been involved in: long-term care facilities, hospitals, less so, educational settings, and then the use of sequencing at a national scale to inform our understanding of non-pharmacological interventions. I'll be going into more depth with regard to educational settings and at a national scale, as these are a bit more recent and slightly more hot topics. So with regard to long-term care facilities, this is just a little table at the end of 2020, where we were in terms of studies outlining sequencing in long-term care facilities and our understanding of transmission dynamics within these facilities as a result. 4 of the largest studies were carried out by researchers within COG UK, and the largest study, Hamilton et al., focused on over 1,000 care home residents in a defined region within the UK. This particular study ran very early within the first wave between March and May of 2020, with overall sequence data already available from 60% of samples at the time. They noted 409 distinct viral cultures in 292 long-term care facilities, with multiple potential transmissions between residents and staff using genomic data. Interestingly, which was kind of very useful at the time, they noted multiple clusters within care homes suggesting independent introductions were common. This was particularly important as it would go on to inform public health teams and their management of care homes, particularly how we dealt with closing visitations to care homes. 2 particular long-term care facilities were located within 1 kilometre of each other and had probable inter-long-term care facility transmission with identical clusters through sequencing. And when we investigated them in a bit more detail, we found that the same paramedics were attending both facilities, and they also shared carers within the facility who were from agency staff. And that led us to suppose that perhaps transmission was occurring between long-term care facilities. And obviously, with a very vulnerable population, this was something of concern. 40 of the 694 cases where we were able to link them to hospital data in conjunction with Public Health England, we noted that they were categorized as suspected hospital-acquired infections. So relatively low proportion of only 5.8 cases, once again dispelling some of the suspicions that care home infections themselves were being seeded from hospitals. One study which I've talked about here was carried out in Norfolk, and which is another region in the east of England. And the study itself focused on one particular long-term care facility. The reason why I've included this is it was of particular interest in that the care facility noted that the cases within the residential home were identical to one another. When we put these in the context of the community, we found that they were distinct from the community sequences. And these community sequences were being generated already within April of 2020 by COG UK. This distinction allowed us to focus on hospital settings, hospital cases themselves. We found that the sequences that we were seeing within the hospitals were from individuals of greater than 85 years old. We then linked these 85-year-old cases, these patients back to their residential homes and noted that they were from 6 different long-term care facilities, all of which shared the same agency staff, suggesting that once again, we were confirming the finding of the importance of inter-long-term care facility transmission. Cambridge University hospitals were particularly well placed to understand understand the transmission dynamics between wards, staff, and also their outpatient settings, with the implementation of a screening program very early on in the first wave seen in the UK. We implemented a very rapid genomic surveillance program where we reported upon genomic clusters on a weekly basis to our infection prevention control teams. There were particular clusters where we noted which were of particular interest, where we found that, for example, cases were linked in dialysis units, which were not known to be previously linked at all. These were patients who were being dialyzed on the same day and within beds that were close to one another, being cared for by the same nurses or healthcare assistants within those dialysis units. Genomics provided particularly interesting insight when evaluating possible linked epidemiological clusters. So these patients who spent large amounts of time on the renal wards were investigated, and we found that those clusters which we were seeing within the renal wards were distinct from the dialysis units. That allowed our infection prevention control teams to focus their efforts on IPC measures within the renal wards and allow the dialysis units to maintain a separation and indicate that measures that we were using to implement that separation were working. So what about universities? Universities are ideally suited to mass asymptomatic screening and surveillance. They directly target the key problem, i.e. asymptomatic infection and transmission, and there's a high density of student accommodation, simplifying the logistics of sample collection. Screening programs have the potential to reduce transmission between students, as well as preventing onward transmission to staff members and the public. So now I'll be focusing a bit on the genomic epidemiology of SARS-CoV-2 in the University of Cambridge, where we identified the dynamics of transmission and this is one of the first studies which has reported, or the only study so far, which has reported upon the transmission dynamics within a higher education setting using sequencing in an integrated, comprehensive manner. So just briefly in terms of the testing pathways at the university, as this is all necessary in order to implement a surveillance program. First of all, we have a symptomatic pathway where students and staff are able to access testing, and those samples are then passed on to our sequencing teams. We also have an asymptomatic screening program where students are tested on a weekly basis, and these tests are performed within pools of up to 10 students based on household. These pools, if positive, then result in individual tests where students can then be isolated if individually positive, and contact tracing to commence as per national guidance. Those positive tests are then passed on to our sequencing teams once again in order to generate the sequences for epidemiological analysis. From the first term itself, you can see that 257 students were positive from the asymptomatic screening program and just over 500 from the symptomatic screening program, giving us 788 confirmed cases. We further identified 184 individuals that were being tested through the community testing program within the UK, which we could also include in our dataset, given the surveillance program that COG UK had in place, testing and sequencing community samples. Just to give an idea of the epi curve within the University of Cambridge over the first term, you can see that there was a rise in cases in the first week of arrival of students, followed by another rise just before the implementation of our national lockdown in November. In terms of our samples and sequencing pipeline, we only sequence samples with a CT value of less than 33. That's simply to reduce the likelihood of failure of sequencing, given our experience from previous studies. And we utilize the GridION platform for rapid sequencing. With regard to the Cambridgeshire community, this pipeline utilizes the standard COG UK pipeline, which essentially allows genomes from the local community and hospital cases collected as part of the national SARS-CoV-2 testing to be incorporated into the study. Samples sequenced at 1 of 17 samples. Hello. Sorry, I think somebody might need to mute themselves.
Yep, muted. Sorry about that.
Thanks, sorry about that. Okay, so samples are then sequenced at 1 of 17 COG UK sequencing sites and prepared for sequencing using either the ARTIC or Rna-Seq protocols. These samples are then sequenced using Illumina or Oxford Nanopore platforms, and all samples are uploaded to processed through the COVID client pipeline. The COG UK pipeline itself is a bit more complex than this, but this gives kind of a flavor of how we test and sequence through the community. The genomic data is then filtered to exclude sequences with less than 95% ends, and those with particularly low file sizes, and aligned with Minimap2 with the Wuhan 1 reference genome, and all protocols are available on the GitHub website for COG UK. In total, in this particular university study, we're able to include just over 50% of university cases in the sample set. And here's our maximum likelihood tree, which includes 972 community cases from the surrounding area and 482 university cases. Over the 9-week term itself, we identified 62 global lineages in the combined data set, with 23 global lineages from the university and 57 lineages from the local community. Not completely unexpected, given the size of the local community is far greater than the university itself. And in total, we identified 198 possible transmission clusters defined by Civit. Civit's clustering tool, which was designed by Rambo et al. In the COG UK group to essentially identify phylogenetic neighbors, which would identify possible epidemiological links between individuals that could be then investigated further to look for possible cross-transmission events. We identified 8 clusters within the university that contained 5 or more university cases, representing 91.3% of all university cases. Reassuringly, this suggests to us that most clusters that did form within the university abated as a result of good infection prevention control measures implemented. One of the next steps we wished to do was to identify the major factors determining transmission within the university. The largest transmission cluster, accounting for just under 70% of university sequence cases, was used as a model to look for transmission within the university. This comprised students across 29 out of 31 colleges. Just to give some context, the colleges in the university represented essentially accommodation blocks for students and also social hubs. the cluster itself was found across 28 undergraduate courses and 208 households. The lineage which the cluster identified to is B1.160.7. And as this is a kind of predictable question later, we found no mutations associated with increased transmissibility in this particular cluster to explain its dominance within the university. So first of all, give some context to this particular cluster itself. We built a Beast tree, a Beast timescale tree, which used the priors as mentioned here. And of note, the cluster which is highlighted by the red square, where you can see the university cases themselves highlighted by red, by the red leaves. The time to most common recent ancestor of this particular cluster to any of the community clusters within our data set was back in May, which was around about 130 days before the university itself started. So this suggested to us that this cluster itself was likely introduced into the university from students rather than from the community itself. Furthermore, we used the COG UK dataset, which provides surveillance of all genomes within the UK to understand whether this particular genome was found to have a particularly wide base within the university, within the UK, i.e. was it just multiple students coming across from multiple sites within the UK and all contributed to a large cluster given the limited diversity of SARS-CoV-2 globally. We found that this particular genome was only sampled within the UK on the 4th of October, so only a few days before the university started, which was very lucky for us. And that was only in one particular region. That genome itself was was only sampled in that one particular region for the next couple of weeks before it was found within the university and then sampled in the community in week 3 of the university term when it finally did spread into the community. To further give us some confidence, this particular cluster was made up of one transmission event and a single outbreak. We used another tool developed by COG UK researchers called A to B, which essentially uses timing data along with sequence data to look for transmission between individuals. When ordered in time, you can see here on the bottom left-hand corner, this figure represents consistent transmission between all individuals within the cluster, giving us further confidence that these were unlikely to be separate clusters forming within the university. That gave us the opportunity to understand a bit more about the epidemiology and the transmission dynamics within the university itself, given that this represented 70% of cases. As you can see here, the majority of cases by week 6 were within first-year students and within the undergraduate arts and humanities students. That's consistent with the modelling data from the university screening programme, which looked to see what were the most likely or the most overrepresented student groups within the university for transmission. Further, we wanted to find what was the concerning event that led to the transmission of this particular genome across multiple accommodation sites. And using contact tracing data within Public Health England, we link this to a common exposure event at a social venue that was identified by over 30 individuals in the first 4 weeks of the university term. All sequenced individuals were located on the large cluster. And of note, in the first 2 weeks, the 9 students who were part of this cluster were from multiple colleges and courses and were known to have attended or were a household contact of a positive individual who hadn't been sequenced, but was still on the cluster. We then used this cluster to understand the transmission dynamics within the university and also identify the effectiveness of national policy to limit the transmission within the university. We found that when students arose when we fitted a BD Sky serial model, the effective reproduction number was particularly high, and this could be for multiple reasons. One, students were inclined to socialize when they first arrived, And secondly, students were coming into a particularly naive population, and therefore an unstructured population in which the effective reproduction number could essentially imitate or equal the actual reproduction number for SARS-CoV-2, given that we saw it around about 2.5. The reproduction number was rapidly contained to less than 1 through effective infection prevention control measures and the screening program implemented by the university. However, this seemed to fail just before national lockdown, where students would have been socializing in the expectation of a period of isolation. And this is what we saw. The reproduction number rose back up to 2 before it dramatically reduced to around about 0.3 in the weeks after. The other interesting output from this particular analysis was the infectious period estimated was around about 3.2 days. Now that's particularly low, lower than the infectious period of around about 7 to 8 days noted in other studies. And that might have been secondary to the fact that the university had the asymptomatic screening program, which would have led to earlier case ascertainment and isolation, leading to a lower period of time where students could go on and spread the virus. We noted other patterns of transmission as well within the university outside of this cluster, and the second largest cluster itself involved 30 students who were all within one accommodation block. 50% of these were diagnosed by the asymptomatic screening program, and this, along with other infection prevention control measures, meant that it could be contained within the accommodation. And after week 3, it no longer spread and didn't propagate. This was seen in at least 6 other clusters. Given the importance of household transmission, we looked at the household attack rate by utilizing the ability of genomics to remove individuals or remove households where more than one introduction was likely, and therefore refine a standard Reed-Frost chain binomial model to estimate household attack rates. Using this model, we were able to estimate the transmission probability after being in contact with an infected individual within your household of 7.7%. Once again, this was much lower than reported household household secondary attack rates of around about 12 to 15%. And that may be because student households are particularly disparate and different to usual conventional households, but also secondly, the asymptomatic screening program allowing earlier identification and isolation of infected individuals. That isn't convention within the usual community setting. We also importantly wanted to see if there was any signal for transmission between the university and its local community, given this is an important, this was an important consideration for universities opening and closing in the first instance. Using the 2 distinct phylogenetic approaches, which I demonstrated earlier, with the time-scaled coalescent tree and also the maximum likelihood tree, both demonstrated segregation of the majority of university and community cases, and that was reassuring to us. Further, closer examination of the sequence data did identify some individual cases of potential transmission, but importantly, we found that of the 161, so over 80% of the 190 clusters, including community cases, 161 included no university cases at all. So just over 80% of the community clusters included no university cases, which was reassuring once again that there wasn't significant transmission between the two. Further, the largest university cluster, the one that we've detailed in quite a lot of depth, represented 70% of the cases within the university, but only included just under 2% of the community cases itself, and therefore hadn't spilt over in large amounts. We also wanted to corroborate our findings by linking this with national contact tracing data, and we found that of the, though limited, number of cases who reported common exposure events, i.e. places where they had visited that were in common with other individuals within the university or community, there was only one individual event which was shared between a community individual and a university student. Furthermore, we in fact noted 19 epidemiologically linked clusters through standard epidemiological clustering that were in fact refuted by phylogenetic variation. So what we can find, what conclusions can we draw from the university study itself? First, the majority of university cases were linked to a single genetic cluster present early in the term in students across multiple courses, colleges, and years of study. Epidemiological investigation of this to identify the source of this cluster was made a priority to implement appropriate infection prevention control measures. The social venue that we noted as the source of dispersion of this particular cluster was noted to also be the root of many of the infections seen just prior to national lockdown. And this is now being considered by public health agencies as we start thinking about reopening universities and coming out of a national lockdown in the UK. Outbreaks were restricted to a single accommodation block, and when this was the case, they didn't observe sustained transmission amongst students. A lack of transmission was seen between university-affiliated cases and the local community in any substantial level. And finally, evidence supporting the efficacy of a national lockdown and university measures for dramatically reducing COVID-19 case numbers within the university was provided by genomic sequencing. And now to take a slightly different approach, I'll be talking about the use of SARS-CoV-2 sequencing at a national scale to evaluate the effectiveness of a non-pharmaceutical intervention, particularly looking at the introductions of SARS-CoV-2 into England between June and September of 2020, so summer 2020, and evaluating the efficacy of travel restrictions. This particular study that we conducted was between June and September 2020, overlapping with the introduction of travel corridors within the UK policy. At this point in time, if you were traveling to a country with a closed travel corridor, that meant quarantining at home for 14 days. The data that we acquired was from a combination of the NHS Test and Trace programme, which is the national community testing programme linked through Public Health England, and COG UK providing the genomes for our study. Over 4,000 probable imported SARS-CoV-2 cases were identified, who reported just less than 19,000 contacts. Of these 4,207 probable imported cases, we had 827 genomes available that were of high quality and we could include in our study, and we could compare these to over 130,000 UK sequences completed by that point. The UK sequencing coverage over that period of time was around about 18% of cases. Those were cases which were tested, not necessarily all cases. And this was strictly a combined epidemiological and genomic study, partly to demonstrate corroboration between the two study methodologies. So with regard to the epidemiological findings, we found that 53% of cases were male with a median age of 26 and an ethnic makeup that was largely similar to that of England and Wales. Over 50% of cases reported travel to 3 countries, including Greece, Croatia, and Spain. Importantly, when we fitted a negative binomial regression model to the contacts reported by cases, we found that travel restriction reduced the number of contacts per case by 40%. We also found that of those cases that were returning from countries without travel restriction, the 16 to 20 age group had the highest number of contacts. However, this reduced to a number in line with that of other age groups when they were returning from countries under a travel restriction, demonstrating a level of compliance, as you can see from panel B in the figure below at the bottom of the slide. We then sought to corroborate this information with genomic data. And what we managed to do here was, first of all, identify unique travel-related genomes within our data set. We did this by identifying those genomes that were of a unique UK lineage and had not been sampled within the COG UK dataset for the 4 weeks prior. We also included within these unique genomes that were of at least 4 SNPs different to their closest relative within the COG UK dataset, and those were the samples which were sequenced prior to the travel-related case being sequenced. We then took these unique sequences and evaluated the COG UK dataset and identified genomes which were similar to these, i.e. within 2 SNP differences over the next 4-week period. This would identify possible travel-related clusters that would represent either onward transmission or further introductions of similar genomes. As you can see from the top left-hand figure, the number of genomes which we noted from countries under travel restriction were far fewer than those without a travel restriction, with a rate ratio of 0.17. Of interest to us was whether this was truly representative of onward transmission, and we used our contact tracing data to try to evaluate this by including it in the model. We found that the number of when we included the number of contacts in the model, the rate ratio was only slightly attenuated towards one, giving us some additional certainty that the additional genomes that were being seen in the similar clusters were further importations into the country from countries without a travel restriction. We also used the Polecat clustering tool developed by Andrew Rambo's group in Edinburgh, part of the COG UK research group, to analyze a particularly divergent cluster highlighted in red in the left-hand figure. This was on a particularly long stem with regard to the other UK genomes surveyed through COG UK. We found that this genome was disparate and spread across the entire English population, but in a particularly young age group. When we linked this to contact tracing data, we found that the majority of these cases were travel related, and in particular from the Baltic region, i.e. from mainly Croatia and Hungary within Europe. We evaluated this particular lineage for any mutations that would increase transmission or be linked to an increased transmission, and found none. And reassuringly, we found that when travel restrictions were implemented and travel corridors to these particular countries were closed, the number of genomes being sampled within this cluster were also accordingly reduced. So in conclusion, closing travel corridors significantly reduces, but does not eliminate onward transmission of imported cases. And that's a finding that was corroborated by both epidemiological data and genomic data. We found that the 16 to 20 age group had a higher number of contacts, and this would now be a possible target for public health campaigns, though reassuringly, once again, they did demonstrate compliance when countries were under travel restriction. Travel-related genomes could be identified with the Polecat clustering tool, and after a period of national lockdown, and this will be of importance as we try to identify divergent genomes, particularly those which may be variants of concern that are imported within national boundaries. But ultimately, genomics has a vital role in understanding transmission dynamics, and that's the message that I tried to get across in this talk. Surveillance to boost sample coverage is essential. With low coverage, it's particularly difficult to identify clusters of interest, given the difficulties that we do encounter with SARS-CoV-2 genomics, given its limited diversity. And this requires the correct infrastructure to be put in place early in an investigation, be this locally or nationally through a program such as COG UK. And finally, genomics can be used to evaluate the effectiveness of non-pharmaceutical interventions at a national scale, which once again provides public health bodies another tool at their disposal to evaluate some important socially restrictive measures which are implemented on their populations. I'd like to thank the groups who have been involved in facilitating particularly the university study here and the travel-related study that I've just presented, and to you all for listening. Thank you very much.
Yep, thanks so much, Dinesh. That was great. Thanks for covering all of that wonderful material. Really impressive. For someone that's been following closely all the great work from COG UK network, very nice to have you today, participating, sharing your work, and especially given that we heard from Duncan McCannell earlier in the day about the Spheres network. So one theme that certainly emerged in early were these establishment of these networks of collaboration in sequencing and surveillance, which we direly needed. And I think COG UK has been a beacon for that for sure since the beginning. I have a few questions from the audience that we can ask. So I have a little bit of time. So the first question that I'll ask is, what are the biggest challenges and barriers to carrying out these kinds of large-scale genomic epidemiological studies?
So I guess that's it's a difficult question. The first, the most important point to establish is one that you've just touched upon, which is collaboration between academic institutions. I mentioned in the talk, we have 17 sequencing centres across the UK, with, I guess, the Sanger Institute as the hub for community sequencing. That only came to fruition through large-scale collaboration between academic institutes across the nation to put in the sequencing infrastructure and coordinating sample collection, but also having the computational infrastructure to try to store, analyze, and then be able to provide this kind of data, evaluating things at national scale. But then even at a regional scale, for example, when we look at the university study, one of the reasons why the University of Cambridge was able to conduct this study was implementing the local infrastructure to allow for linking of student samples to sequencing centres and facilitating that transition. And then finally, I think it's impossible to do this. I think, and hopefully I've tried to get this across, it's impossible to find meaningful results without decent epidemiological data and metadata. So having good relations with your public health teams and having a back and forth, so conducting the research is vitally important, but being able to provide them with evidence that they can then inform their public health decisions on is really important. And once you've got those links in place, then that trust evolves, and one can then try to draw meaningful results from these large-scale type studies, be it on the restriction of travel or outbreaks within colleges and hospitals and care homes.
Great, thanks so much. Okay, so the next question we have here, there are multiple studies that refer to the use of SARS-CoV-2 sequencing to understand transmission in various settings. And the question is, has this resulted in routine use of genomics to inform infection prevention, control, / public health measures in the UK?
Yeah, so so ultimately it has. I mean, the genomic cell within Public Health England look at this at a national scale. So first of all, it was whilst cases were on the lower side, local, evaluating local transmission dynamics, and that's actually one of the ways in which the Kent variant was identified. So by Kent variant, I mean B1.1.7, which was the first variant of concern. So it was noted that the cases within that particular region of the UK were rising despite a national lockdown. And that didn't quite make sense, which led to the further more detailed genomic evaluation of those sequences, identifying the kind of concoction of mutations in the spike protein that led researchers to think that this might be something that's more transmissible. but even more locally, looking at hospitals. At Addenbrooke's, which is our local hospital in Cambridge, we have a now weekly integrated epidemiology and epidemiological and genomic infection prevention control meeting, where we present our genomic findings, and then that's kind of integrated into real-time action.
Okay, great. And then I have a question of my own I wanted to throw in here. And you mentioned this for sure, but just looking again, thanks to this, thanks to everyone out there who's been sequencing and depositing data in GISAID in the public repositories, this has enabled just this and unleashed this unprecedented amount of data for this pandemic. And I guess my question would be, given the hundreds of thousands, I don't know if you've exceeded the 1 million genome mark yet, but what type of analyses does this filter? How does this help? And I guess more broadly speaking, through just broadly speaking, COG UK and internationally, how does all this extra data really help us track variants, et cetera? If you give just maybe an explanation of that to maybe those that are not familiar with this topic. So as a layperson looking at all this mountains of data and this great work you've been doing, how does this really enable new analyses?
So I think one of there are kind of multiple core aims for COG UK when it was first established by Prof. Peacock, and one of the aims was to be able to shine a light on clusters that were forming within regions, and then by doing so, directing and facilitating public health contact tracing, but more importantly, and perhaps, maybe more importantly, but at least equally importantly, thinking about vaccines and vaccine escape. And as we saw in December, the UK, I think at that point, had contributed over 50% of the sequences in GISAID, which has thankfully rapidly come down as a proportion because other countries have become so much more alert to this. And could increase the efforts of sequencing, which is really welcome. But essentially, as a result of that, we were able to identify the first variants of concern. For example, the Delta H69 and H70, V70 mutation deletion, identifying that allows us to then carry out the experimental studies that we also then showed that it had an increased risk of antibody escape, and then ultimately incorporate those findings into the next primers, which will result in vaccine development that might be the boosters, which we want to use for future populations to try to mitigate or at least increase the efficacy or maintain the efficacy of the vaccines that we use. So it's all kind of like a race against one another in one way, like an evolutionary arms race of the virus and us, and sequencing has clearly demonstrated that it's a vital component and friend of ours.
Wonderful. And then I guess maybe a final question here, and it's the same question I asked Dr. McCannell. So where do you see, like, what's the roadmap for COG UK 12 months from now? So where, given all the successes, everything you've accomplished, what are kind of some of the big challenges as you see them, or where's COG UK headed in the next 12 months to face some of the challenges that out there? So.
Yeah, so I mean, thankfully I don't actually have to answer that because Sharon had actually recently written a blog on this. But I think, I mean, it was set up initially as a research consortium. I mean, as a result of its fruition, it's brought together a whole country's worth of sequencing efforts. And there are multiple ways in which you'd want to multiple reasons for wanting to maintain that. One is obviously the kind of pressing issue of COVID and wanting to maintain vigilance through sequencing, and also the amazing research that we get to do and see as a result of COVID and within COVID, but also wanting to take this not just keep this to COVID-19, but to maintain this culture in all other pathogen sequencing. The big problems that we'll see, not just in 12 months, but in 10 years, 50 years, 20 years, will be antimicrobial resistance and developing similar networks as COG UK is kind of one of Sharon's next ambitions to kind of ensure that we have a culture of sequencing incorporated in routine pathogen care, really. And then I guess, I mean, what we've been doing for the last year, assisting the ongoing public health response, and that will always be part of kind of the use of COG UK as an institution and the kind of the research that comes out of it in real time.
Yeah, great. Well, I think just to wrap this up, that wonderful, really wonderful presentation and talk. Thanks so much for joining us today. Really appreciate the time and all of the information you provided today. So with that, I would like to thank not only you, but all of our speakers up until now before our first 10-minute break here. Wonderful way to start this third symposium off. And thanks again, Dinesh. Really a great pleasure to have you here today with us. And now we're going to pause for about 10 minutes. We're going to pick it up in 9 minutes at 1 p.m. Central, and we'll get started with our second lightning talk session. So thanks, everyone. We'll turn the lights back on in about 9 minutes. Thanks, Dinesh.
Hi, everyone. Taking over for Todd for the afternoon, moderating. We have about a minute before 1 p.m. Central, but I thought I'll start a little earlier just to give some time for people. But so I'd like to introduce the next session, which Kyle Hernandez from the University of Chicago is moderating, and Kyle is in charge of he's basically the lead of our modeling subgroup. As we mentioned earlier, we have many different subgroups, and he's one that's running the show for the modeling subgroup, which you'll hear about in a second. One of the people in there about their work. And then there's a couple of, as Kyle introduced, a few people talking about our tools that in collaboration with Real Networks with Reza Razul for the Mass-Check app, and then Brian Schmoller, who's the vice president of Signature Science, has developed a very nice exposure assessment tool, COVID exposure. And then this whole lightning talk session too with Michael Joachim talking about microbial effects and synergy between the virus. So no further ado, Kyle, I'll let you take it away.
Thanks, Afshin. So as Afshin mentioned, our first talk in this session will be from Catherine Conley from NASA Ames Research Center. Go ahead and take it away, Cassie.
Thanks very much. I'm just now sharing my screen. It will actually work. Can everybody see this?
Yes.
Great. Okay, so I am going to be talking about work that the modeling group has been doing pretty much since the last, for about a year now, more or less since we started the Covert. In particular, this work I initiated it because I was concerned about some of the models not showing accurately, not being very accurate in terms of representing deaths that were happening from COVID in subsequent weeks. So this is definitely a collaborative work. There have been a lot of suggestions from many people. We submitted preprints, manuscripts with a variety of people from the group. As more people join the modeling group, more people have been contributing to this work. So it's definitely a collaborative effort, and it's very nice to have such a interested and active group of people supporting any kind of research, these kinds of research questions. So the fundamental problem is that when you're trying to model something, you need good data in order to feed your models so that the model outputs will be accurate. That old garbage in, garbage out is absolutely correct. I will note that my background is in astrobiology, so I'm normally looking for signatures of biological activity in extraterrestrial material. And of course, we haven't found any yet, but that means that I have a lot of awareness of the need to be able to use different data streams in order to assess whether what I'm seeing in one type of data is consistent with what I'm seeing in other types of data, to be able to have a comparative approach to evaluate the quality of any of data that I'm using to make any kind of interpretation. So if we have highly variable data streams, then our interpretations are going to be somewhat dubious. And if you have really what looks like confident data in one data stream, you need still to be able to check that against other data streams, because there may be some sort of systematic or systemic errors in your instruments or in your detections mechanisms that are going to render the interpretations you make of that single data stream dubious. So common metrics we use in astrobiology are to compare data streams from different detectors. You compare recently obtained data with historical data. And then importantly, in order to compare data from different locations, as well as from different detectors, you need to have some sort of way to normalize the data so that you can compare apples to apples across different data streams or different sources of data. And when you're trying to characterize the same thing, the same object or the same situation, you need to be able to normalize across different detectors to make sure that you understand, that to facilitate your ability to understand what's going on. So early in the pandemic, there were many models being reported on. There were questions raised in the media about the quality of the data that were being used to inform those models, the quality of some of the models. And if there had been data quality metrics, then that could have distinguished locations where the data were reliable from data, from locations where the data being produced were perhaps less reliable. And this is important, of course, all the way along in producing models, but most particularly now, it's absolutely essential to have good data in order to understand the efficacy of the vaccination programs. Because if you have a location that is never reporting high amounts of COVID, you will not see that the vaccine had any impact there, although perhaps it does. Then in contrast, if you have locations that previously were reporting high levels of COVID and subsequently are reporting low levels of COVID, regardless of whether there's a vaccine being rolled out in that area, again, you're not going to know whether the vaccination programs are effective if the quality of the data you're using to evaluate them are not very high. So in the case of these analyses, what I'm reporting here are data streams specifically only from the CDC, but from two different units of the CDC that I'm considering two different detector systems. So we have the Case Surveillance Task Force that reports on daily data. We have them that are from the public health surveillance activities. We also have the National Center for Health Statistics that reports on death certificates filed. And there is a delay in the death certificate and filing death certificates. That delay is different between different states. It is also different, can change depending on how overloaded the public health system is in the particular location reporting. So each of these data sets reports incomplete data in different ways. So rather than trying to do a regression to estimate the final, what the reporting eventually will be, instead I'm using the raw numbers and normalizing those raw numbers, recognizing that there will be some level of incompleteness in those data, but using them as observations of a phenomenon that I'm trying to understand. In addition, we have historical data available from the CDC Wonder database, as well as being reported out in some of the data sets that the Center for Health Statistics has put out on COVID. So those historical data are a good way to, a good potential way to normalize the data consistent with what we do at NASA in terms of normalizing against various historical data sets. In addition, the CDC does provide regression analyses in their excess deaths data set. They report both the average expected causes, all causes, deaths from all causes based on a regression analysis of historical data. They also report on an upper bound above which they would consider deaths excess. And I'll describe a little bit more about that later in the talk. So these are the detector data streams that I'm using to do the analyses. Initially, just looking at raw numbers for the entire United States, in red is shown the weekly case surveillance deaths. So these are daily reports, and I've just summed up 7 days to produce the weekly data so that I can compare it with the weekly report from the National Center for Health Statistics on death certificates with COVID as the underlying cause of death. So in blue is COVID, is death certificates. In red is the weekly case surveillance data. And these two data sets are slightly out of phase. Death certificates filed eventually, and mind you, there's a longer lag in the reporting of death certificates, but 4 weeks early in the pandemic, when these data are relatively complete, the CDC says that death certificates about 80% complete, even in the slower locations, after 2 or 3 months. So data older than 2 or 3 months should be substantially complete. So in these older months of data, death certificates, when there is an increase in COVID, actually precede the surveillance, the reports of surveillance deaths. So these surveillance deaths are reported immediately and can be slightly revised later, but the eventual number of death certificates reported over when looked at over the entire US, the death certificates increase earlier than the daily surveillance data, even though the lag in death certificates means you don't see those deaths until after the death certificates have been filed. So in increases, death certificates increase, in decreases, death certificates go down earlier also. So there is this phase lag between surveillance data and underlying COVID deaths. But for the whole US, the magnitude is about right. It's mostly that there's a phase lag. And then you have these oddities in the weekly reporting that get evened out in the death certificates. However, when you go to look at my third detector, which is all-cause deaths in 2020 and 2021, above the average expected all-cause deaths that were reported by the CDC's excess deaths data set, all causes above average is anomalous, is different than either the case surveillance or the underlying COVID death certificate deaths. So that line is shown in the bottom panel here in green. And all causes above average increases before the death certificates and before the case surveillance data for the whole US. It gets to a higher peak in April, then it drops down to be fairly similar to the death certificates and the surveillance data in May and June. But in July, you start to see this dramatic increase in all causes above average expected, which is greater than the increase in either case surveillance or underlying COVID. So there is a difference in how all causes above average are being reported that changes over time relative to case surveillance and underlying COVID deaths. So again, these differences are going to be potentially problematic in understanding how the vaccination rollout is affecting deaths from all causes compared to deaths from COVID, and also going to be affecting the models we're trying to predict the case surveillance data. If the case surveillance data are not accurate relative to other data streams, that means that the models perhaps, there might be a way to identify locations where the models could be less or more accurate, depending on whether those 3 data streams are showing similarities or differences. It could also be that those differences at US level are consistent from state to state, but in fact, they are not. So here's 4 example states that show differences in the, that give examples of the different kind of differences that you see. In California, the green all causes above average data stream is fairly similar to the COVID data sets, either surveillance data deaths or underlying COVID deaths for up through July, and you start then seeing a dramatic increase in deaths from all causes and death certificates from COVID after about November that are not reflected in the case surveillance data. And remember, after January 21, these death certificates are probably, this peak here may be partly an incomplete reporting issue. But California shows variability in the death certificates in the data streams late in the pandemic. In contrast, Massachusetts has the greatest peak early in the pandemic, but then after that, the case surveillance data actually are reporting more death certificates than, the case surveillance data are reporting more deaths from COVID than have ever been reported on death certificates since July. And the difference in all cause above average is actually lower than the number of death certificates from COVID. That's a very unusual pattern, but it suggests there's the way that Massachusetts is reporting deaths is very different than the way that other states are reporting deaths. In Florida, the case surveillance data and the all causes from COVID on death certificates are fairly similar, but again, out of phase. In contrast, the deaths above average from all causes are dramatically greater starting in June through the peak in July, and then have come back down after September back to closer, so that all 3 data streams are fairly similar. This large peak in all causes above average deaths was just reported in regression, a paper that was published a few days ago and has been starting to attract attention in the media, that there's this large inconsistency in deaths above average from all causes and deaths specifically from COVID. So that was something that was coming out very obviously, even as these deaths were happening by comparing these raw number data streams. Now looking at the raw numbers, the last example is something where things look very anomalous, in that in North Carolina, you see up through end of September, beginning of October, end of September, that the all causes above average are somewhat above the 2 data streams from death certificates and surveillance data, but these are consistent with what other states see. Starting in the fall, the all causes above average number drops considerably, such that by November, you're starting to see thousands of deaths below average, which means that North Carolina just has stopped reporting death certificates. This started dropping in the beginning of October and has gotten pretty dramatically significant here. So this is a very strange situation where when you subtract average numbers from all causes in 2020 and 2021, you get really large negative numbers.
Yes, there's about 1 more minute left, just FYI.
Oh my goodness, okay. So the, thank you very much. So the normalization, normalizing over average expected rather than all causes, you start seeing more consistent data sets. I wish I had a clock up because I have lost all my time. But so 10% COVID by all causes, 1% COVID by underlying case surveillance data over average is greater than 20%. Can look at all states by flipping the graph sideways, and different states show very different numbers. Again, North Carolina shows 10% COVID by average by 2020, whereas many less deaths by all causes. Looking at states from above or below average by county, lots of different states show different, show high variabilities, both counties above average as well as counties below average. And again, the COVID percent COVID of 2020 numbers don't correlate with COVID above average numbers. If you look at county level discrepancies in Florida, the largest counties are showing relatively similar, relatively appropriate numbers, whereas it's smaller counties that are showing the largest divergences. Whereas in North Carolina, even the large counties are showing many deaths above average, yet a few counties are still reporting significantly higher than average numbers of COVID deaths. Of deaths above average that are not COVID. And it's very important to look at the data that are not known as well as data that are known. So there are a large number of small counties, over 1,000 counties at the end of December, that still haven't reported more than 10 deaths from COVID. That means the county level data are not available for all-cause deaths. But looking at average annual deaths, those counties would be expected to have on average about 20,000 deaths. So if you have whatever fraction of deaths are happening from COVID, those are deaths that are potentially being not reported, at least as county level. They may be rolled up into the state data as reported by the CDC. But different counties, different states have very different variable numbers of counties that haven't yet reported data. So larger counties are in certain states. Texas and Florida have quite a few counties that are not reporting data. So trying to sum up, develop metrics that are able to show these data for all states in a very efficient way. If the surveillance data were being reported consistently with the all causes above average, that is the data from all states were being reported consistently, all the states should fall on a single line. In fact, the regression for surveillance cases and all causes above average R is only 0.75. There is a moderate correlation, but it's not nearly as large as it would be expected to be if all the states were reporting their data consistently. So some states in the Northeast are reporting actually more surveillance data relative to the increases above average, whereas most states are reporting pretty close to, pretty consistently amongst all the states. But looking at the divergent states, again, North Carolina is very, very low, although it is reporting fairly high amounts of COVID. The total all above average is quite a bit lower than one would want to see for consistent data sets. And then other states are reporting lower surveillance cases and lower all cause above average cases. So these discrepancies are still visible. Then so COVID has caused large increases above average, but the fraction of the increase that's attributed to COVID on a state level and on a county level is highly variable. And some examples of those variations, a few states that attribute low percentages of weekly deaths to COVID also report very low numbers of average expected deaths, although those by 3 months prior to the report date, there should be greater than 70% of those deaths should be reported if things are going normally. So there's some very strange anomalous actions in the reporting of all cause deaths on death certificates in these states. There are many states that are reporting low numbers of all cause deaths above average, and yet very high numbers, very high fractions of 2020 deaths being from COVID or all cause deaths being from COVID. So this inconsistency in reporting of COVID deaths relative to reporting of all causes deaths is sort of endemic and potentially a systemic error, or it could be it could be a result of reporting delays. There could be other contributing factors to that, especially when you look over time. And final conclusion is that using appropriate data quality metrics is going to be very important to understand the efficacy of the vaccine models, because if there's a state or counties that are just not reporting deaths as being from COVID, if you only look at COVID numbers to assess vaccine efficacy, you're not going to see that the vaccine is effective in those states. So thanks very much. And sorry for rushing through everything. I didn't realize how long it was taking because I don't have a clock on my computer.
Thanks, Cassie. I think we'll have you answer the questions in the box for the sake of time, in the Q&A box, if you don't mind. Appreciate it.
I can do that. And I need to stop sharing.
All right. Our next speaker in the session will be Brian Schimler from Signature Science.
Hello, can you guys see my screen?
Yes.
Great. I appreciate the opportunity to share this work with you guys. This was a homegrown COVID project, and that we worked on collaboratively with several other members, including Molly Isbell from Signature Science, Sam Dutta from Utah State University, Afshin, Cecil Alex, a student at the University of South Florida, Shirag Goel, a student at Northwestern University, and Tenley Archer from Algin Biotechnologies. So the objectives that we set out to do on this was to come up with a way of understanding the exposure and the risk, therefore, that groups working in workplaces have from COVID. And when we look at the variety of different qualitative and quantitative guidance that's given from a large number of sources, government and otherwise, industrial hygienist or a health and safety manager or an operational manager is left with lots to wade through. And there's a variety of different factors, both mechanistic and behavior-based and sort of population-based, that have to be considered. So that's what we attempted to build into our model. And the idea was to give this tool to the health and safety manager or the industrial hygienist so they can advise their organization on best strategies for mitigation. So in the end, the tool at its current status, it looks like this. It actually lives within a PDF, and it's a JavaScript-based model that runs sort of in the background of the PDF. And I'll just give you a quick tour of the 10 steps that it goes through. The first is you assess your subpopulation of the group that you're going to be engaging with or that you're looking to manage. In terms of what you know about them, in terms of their exposure or risk to exposure to COVID. Then you add the number of people, and you have two scenarios that you can run side by side. And then the next step is to look at distancing options, ranging between 0.5 meter up to 4.5 meters. And we've done this for both sort of SI units, the metric system, and for US customary units in feet. So there's a version that's in those units as well. So then the next step is looking at the mask usage, ranging from fitted N95s all the way to no mask, and then how much compliance you have with that. We look at vocalization behavior, how much people are talking and the different activities they're engaged with, as well as your inhalation, and then the duration of the activity, and then whether it's indoor or outdoors. If it's outdoors, what's the wind speed? If it's indoors, what's the ventilation rate? And what's the filtration scenarios, whether HEPA filtration or no filtration. And we also build a ventilation estimation tool that can be used by if you have a CO2 monitor and you place in the room and you let people spend some time in your room, you note what the CO2 was at the starting time and what at the end time, you can estimate the ventilation rate. And then then finally the room dimensions. And then we have the local conditions, and we look at the prevalence of cases in the community, the undiagnosed factor the estimated undiagnosed factor of COVID in the community at that time, and then the variant prevalence and immunity prevalence, so the most recent thing we've added to the model. And then finally, results are presented, and then you can see a sort of a thermometer here of comparing the two scenarios. So that is the model as it currently stands. And we'll come back to this in a few minutes. So some background on the model. It is really an exposure dose model where you're calculating the exposure from the concentration times the inhalation rate times the exposure times the number of people in the group. And we're looking at group exposure, not individual exposure. And that's because we're thinking that people are, the industrial hygienists and health and safety managers are interested in looking at the total exposure of a group. So the guts of the model, we employ a near field, far field box model approach. This is commonly used in industrial hygiene field. It was originally developed for chemistry exposure, and it's been adopted and adapted to bioaerosol exposures by us and others. And essentially the box within a box model looks at the room as one box, and then sort of has a virtual box to account for the higher concentration in close proximity to the source. So in this case, one person is a source and the other person is a receptor. So what about multiple people? Well, here's how the model would look. You actually have to apply what's known as the principle of superposition, which is essentially adding the effect of one person on top of another person on top of another person on top of another person. You have to apply this principle in your model, and the multiple box approach. If you're using a rectangular grid space, it would look like this, and it gets kind of messy visually and mathematically. So we were able to find a simpler way of doing it using a triangular grid of the world, using triangular prisms that are formed from that. And then that way, you basically, if you're saying everybody's going to be 3 feet apart or 3 meters apart, they actually are in this model. So this was one of the innovations we brought to this approach. So that results in you summing up lots of terms from the near field and the far field, and calculating a worst case exposure for the person at the center. And what we've done is, in our model, conservatively, we use that worst case exposure as our result, as opposed to sort of calculating the value for everybody in the model. We use the worst case person as representative of everybody. So there's obviously some conservatism there, but it's also simplifies the model, makes it more practical to use. So the other aspect of the model is that it is a comparative exposure model, and we're comparing it to a baseline scenario. That is a scenario that it would be deemed high risk by OSHA and by the CDC. And we're using that scenario as to compare all the other scenarios too. So if that scenario in the model is equal to a 1.0 result, and everything higher than that is higher than 1, and everything with a lower exposure is less than 1. So the assumptions are shown here for the baseline scenario. So the resulting exposure dose equation simplified version actually is shown here, where what I'm not showing is all the action that's happening inside these summations. So as you thinking back to the first slide second slide where I showed the model steps, well, each one of these represents a step in the model. This is the first step, looking at the subpopulations. This is the ventilation step. This is the distancing step, the mask wearing step. This is the ventilation, the vocalization step, the inhalation step. This deals with the building dynamics, and then the exposure. The number of people exposed is built into step 2, and so on. So now we're actually also make some further adjustments, and these are the more recent things we've added for the prevalence of immunity and variance in the population. So those we've done based on the fact that some of the variants are more potent. So we come up with a higher equivalent exposure value for the more potent variants, and potency meaning more transmissive. So for B117, we're using a 50% assumption of increased transmissivity. So in case of the immunities, that we're looking at that functioning in two ways. First, we're including the vaccination-based immunity and recovery-based immunities. And then a recent Israeli paper on the Pfizer vaccine showed a three-time reduction in shedding rates based on vaccination. And then this is a little bit perhaps controversial, this aspect of what we're doing. We're applying a sort of the immunity as an effective barrier, and based on this sort of respiratory exposure dose model. I'm sure there would be questions on this, but I'll move on in the interest of time to some.
3 minutes, Brian.
Okay, great. So here's a couple of examples. This is the often used Skagit choir example of March 2020, and with 61 people present, and I'm modeling here both the A and B scenario, A being the actual event, and B, what if they did this outdoors, and what would the results be? So modeling the various parameters that we can collect on that event, we show that was indeed a very high scenario with value up near 100. And then if we moved it outdoors, we would have seen quite a bit lower exposure rates, right? So we know that, organizations like choirs did indeed do that over the past year for their groups. So here's another example of modeling in a company in-person training. This is something, we looked at in our own company using the model, where we are looking at, hey, we have to train 10 people, and our room doesn't seem like it's big enough, right? So first we model what the scenario was in where we were first going to be having the training, and then we modeled sort of the mitigation strategy and compared the two. And in this case, we were able to set, and I hadn't mentioned this before, a CO2 action level for the room, and we actually have CO2 monitors in the room. So we also had a number of our members of the group do some real world scenarios. So Chirag Goel from Northwestern University has looked at using the model in a variety of university settings, comparing the prevalence on the campus based on actual data from the health office in the university, and he's built to a database of the results, and we're going to be using that in an upcoming paper. Another example is Cecil Alex, another student member of our group, University of South Florida, had modeled a super spreader event that happened in his community. And he then did a bunch of what ifs of what if it was in different communities, and what if it was outdoors versus indoors, and so on. And then the other thing that is interesting we can do with the model now that we added the vaccination parameters, what's the end game scenarios? When is life going to be safe enough to do things like we used to do, right? So we plug that same scenario that I showed you earlier in there, but in this case, instead of being at 20 average cases, daily cases per 100,000, we have this at 0.1, and then we have the vaccination rates going all the way up to 80%, with the recovered at 35, and these are not. There's a union of these that there are people, of course, that are vaccinated and recovered, so we're taking account for that. And then we use a 90% vaccination immunity effectiveness for that, as I mentioned before, with the effective barrier. So that is showing us at a low end of a medium risk in the model based on those scenarios. So next steps, we're looking to do some validation of the model using real world data and computational fluid dynamics scenarios. We're looking to expand it to looking to expand to transmission using the dose response approach, because our model right now doesn't quite go all the way to transmission, where it's still an exposure dose model. Looking for additional pathogens, we're going to migrate it to an Android or an iOS platform, and the other thing we can do with the model with the same math is to allow people to place people wherever they want in a room, not at sort of set, spaces. You could sort of model a very specific scenario, and then if we have a variant specific and vaccine specific data, right, where let's say there's a variant that is not effective against a variant, we can add that to the model, and then looking to sort of build out the model in different types of computational platforms to run various scenarios and sort of have this be a front end to other types of analyses. So that's all I got. So
Thank you, Brian. I appreciate it. If you wouldn't mind, there's 2 questions in the Q&A that maybe you could answer by typing in the answers there, so we can go ahead and move to next. I appreciate it. Thank you. Our next speaker in this session will be Riza Rasool from Real Networks.
Hi, everybody. Let me share my screen. So a camera can look at a passing face and instantly estimate whether it's wearing a mask, measure its temperature, estimate age, gender, sentiment, and even match it to a database of millions. Automated facial recognition abdicates a very human function with all of its subjectivity, foibles, and partiality to a machine. We want machine-based facial recognition to be fairer, to be more objective, not hardwired with the very fallibility, unfairness, and prejudice prevalent in society. Modeling AI for low bias is possible, and here's how. So I'm going to share my deck and go through the slides as quickly as I can. In the interest of time, let's go into presentation mode. There we go, it's coming along. And I'm gonna click through. So, hello, I'm Riza Rasool, CTO of Real Networks. Many of you may know Real Networks as the inventor of streaming media. You may have used the Real Player. One of my priorities since joining has been to promote AI in all of the product portfolios, especially the Safer Computer Vision program. This presentation summarizes my paper, How to Engineer Ethics into AI, Combating Bias in Facial Recognition. It's based on the learning on the SAFER program, but let's unpack this dense 4-page paper in 10 minutes and see what we can do. So a deep neural networks 101 would say that it's really a mechanism of asking questions of data. You train a model from historical data with historical answers that we call the ground truth. The model is then used to make inferences from new data and provide new answers. You can see the model is analogous to the structures of biological circuits in the brain. Weights, operators, and nodes are analogous to dendrites, neurons, and axons. The more nodes, the more the complexity, more computation required to deliver an answer. So to bring it, make it more relevant to this audience, here's an example of a cancer diagnosis model trained on historical biopsy data with the doctor's diagnosis as the ground truth, and the inference allows for automated analysis of new biopsy data with an automated diagnosis made by the machine. Sounds wonderful. So a simple binary classifier and more complex vector classifiers can be cascaded together for useful applications. Here we see face detection being followed by a number of other models that can enable either media and entertainment applications or, for this audience, public health applications. As the pandemic took hold, it became clear that computer vision could help in touchless authentication, access control, occupancy counting, mask compliance, and even fever screening. I was introduced to Afshin and Todd, and as he mentioned, Covert and Real Networks released the Mask-Check mobile kiosk app that monitors mask compliance. You can get it from the Covert site. It's free of charge. We should, why don't we just give I'm going to stop sharing and in fact share the app screen quickly. Let's see the beauty of there we go. We're going to share from, I was hoping to be able to share from my mobile phone, but maybe not. Okay, so let's instead share what the output of the Mask-Check app could give you. And you could, the output of the Mask-Check app is in fact a map. The output from thousands of kiosks placed at buildings, at offices, at shops that report on mask compliance can be aggregated to provide a heat map across the country. And here in Seattle, where it's been deployed most, you can see the data is all aggregated, anonymized, and viewed at a county by county level. Researchers could look at it at a more detailed level if they wish. So let's get back to the deck. The map sounds useful. Mask-Check, let me stress, does not use facial recognition, but I want to talk about the elephant in the room. So Joy Buolamwini and other researchers have shone a light upon the matter of biased AI. Popular facial recognition systems underserve Black females despite their fame and celebrity. Here, a number of them identify famous females as male. Just for illustration, I use my software to in fact show that it's possible to make that determination correctly. Isn't it just a problem of lighting, optics, and the laws of physics? Well, no, it's not. It's not just the design of the optical path that's at fault. The increasing logic and automation built into cameras is prone to bias. Cameras are proliferating, becoming smarter, and even autonomous. The problem will only get worse. So what is bias? NIST measured how facial recognition accuracy varies across demographic groups for about 103 algorithms. Each algorithm was calibrated for white males. So here's the purple line showing the calibration point. And then their accuracy was measured, and the dots represent the other measurements. The accuracy was measured for the other demographic groups. In their 2019 report, NIST chose to highlight the trends in algorithms from China versus the rest of the world. So the bottom two quadrants are from China. The top two quadrants are the male and female tests for the rest of the world. I originally discounted this as pandering to the prevailing administration, but changed my mind after that. Well, we were glad that the Real Networks algorithm achieved the lowest bias, but I was interested in how other researchers address the problem of classifying humanity. So how did we do it? Well, our recommendation and our how-to guide is first develop a code of conduct. You're not going to stumble upon a low biased approach by accident. Develop a code of conduct, a set of guiding principles on how you're going to conduct an AI program. This is very important. Next point, more practical, source a diverse representative training set with consent. Researchers are hungry for data sets. Don't accept metadata uncritically. Think about what questions you're going to ask of the data. The AI will always give you an answer. Be careful what you ask for. So pseudoscientific classifications of humanity, though debunked in Victorian times, still persist. Sussman tackles the problem in his book, The Myth of Race, and the troubling persistence of an unscientific idea. This has relevance to public health, epidemiology, and the attribution of certain pathologies to population groups. Correlation is not causation. So what can you ask about a person just by looking at their face? Can you determine their zodiac sign? Now that sounds unscientific. What about their credit score? What about their religion? What about criminality? What about sexual orientation? Would you believe it? There are AI programs that actually aim to do that. AI will always give you an answer. Be careful what you ask for. My preoccupation in classification stems in part from my South African roots. I was born at the height of apartheid, a regime under which our rights and level of citizenship was based upon racial classification. I grew up in London and now live in Los Angeles. I guess I'm the embodiment of the South African UK variant of humanity, now at large in California.
3-minute warning.
Okay, so can an AI model be racist? Yes. How can we tell? Is there a simple metric that's short of the exhaustive NIST test? Yes, I propose that there is. I looked at the speed versus size of models. It's a simple test that anyone can run. In the cohort of the most accurate algorithms, there's some outliers that seem to run much faster than their size would predict. I suspected that not all of the model's nodes and weights were being used to compute the inference. So I hypothesized that there is a metric, the metric I call sparsity. It's the product of size and speed. The sparsity in a model indicates that there's an artificial superneural logic being imposed upon the network, resulting in a collection of demographically compartmentalized submodels. In the case of FR, this would suggest that a collection of demographically specific models are being aggregated together, but with a switch in the front, which selects which submodel to use. The switch actually embodies the very sexist, ageist, and racist logic that we were trying to eliminate from this objective AI model. And the number of submodels could be large. Imagine if you had, let's say, 2 genders, 3 age groups, and let's say 4 race groups. That could end up with 24 submodels. This is obviously a ridiculous solution. I plotted the data points using the bias rank that NIST provided and my new sparsity rank. Of course, to fit inside a camera, the models need to be compact. To operate on live video, they need to be fast. To be socially responsible, the algorithms need to be fair. So we want to be in the bottom left-hand corner of this quadrant. It's possible to be fast, small, and fair. So the good news is that NTech Lab here shows that a collection of biased models in aggregate can achieve low bias, albeit at 80 times the size of an unbiased approach. Wouldn't it have been easier to treat humanity as a single homogeneous data set and you get a more efficient result? So I'm going to leave this slide up by way of conclusion, perhaps a bit provocative for the rocket scientists in the audience, but I contend that the Wernher von Braun defense is at best cowardly. As engineers, scientists, program managers, we can do better. We must do better now more than ever. I'd be happy to take questions before I dash out to get my first vaccination. Thank you.
Thank you, Riza. Very timely, relevant topic. If you wouldn't mind, there's 3 questions, it looks like, in the Q&A box that you could type an answer into so we can move on to the next speaker, if you don't mind.
Let's
Appreciate it. Yes. Okay, our last speaker for this session will be Michael Yoakum from Baylor College of Medicine.
Hello, my name is Michael Yoakum from Baylor College of Medicine, and thank you for allowing me to present our ongoing research on behalf of the Covert microbial subgroup. I have no conflicts of interest or financial disclosures. So, stand by. So in order to better understand the potential relationship between COVID-19 and disease severity, and the community dynamics and functional profiles from a microbial and microbiome standpoint, we conducted an analysis using human bronchoalveolar lavage fluid metatranscriptome samples that were sourced from 8 different publications and made available through public repositories. These samples comprise 3 main cohorts consisting of an unaffected control cohort, a community-acquired pneumonia or CAP patient cohort, or COVID-19 positive patients, with a secondary analysis of disease severity outcome amongst a subset of COVID-19 cohorts broken down by survival outcome, deceased or survived. The objective of this study was to compare the COVID cohort amongst the uninfected and the community-acquired pneumonia patient ball samples and identify any changes in microbial-derived community dynamics or gene ontologies associated with COVID-19 and predict outcomes of COVID-19 based on the metatranscriptome profiling, with the overarching hypothesis that there's some potential informative relationship between the microbiome and the severity of the COVID-19 disease onset and progression. So raw sequences were downloaded from SRA or CRA, followed by read processing and adapter trimming, QA/QC, and filtering of human and low complexity reads using Trimomatic, FastQC, and FastP, followed by taxonomic classification using Kraken-2, and functional annotation using SeqScreen. The taxonomic classifications were then decontaminated against negative controls when applicable using the library decontamin R, followed by statistical analysis and visualization using the bioinformatic software packages Phylogenetic Vegan Metacoder. The functional annotation gene ontology counts derived from SeqScreen outputs were parent propagated using the Covert micro conda package generated by Mike Lee at NASA. Parent propagated gene ontology term counts were then subjected to Dirichlet mixture modeling or DMM clustering and comparative analysis using multivariable associations with linear models or Maslin-2 from the Huttenhauer lab in R. All scripts, intermediate files, and results can be found and are open sourced at our OSF website and can also be found in the Covert micro GitHub repos or the Conda package. So after read filtering and batch effect sample removal, our sample cohorts consisted of 29 uninfected patients samples, 25 community-acquired pneumonia samples, and 32 COVID-19 samples, bringing the total N in this analysis to 86. Amongst the COVID-19 cohort with known survival outcomes, 10 samples were from the deceased cohort and 15 were from the survived cohort. So results from the Maslin-2 analysis revealed that 30 out of a total of 13,534 gene ontology tags were associated with COVID-19 when compared to the community-acquired pneumonia state patient cohort or the uninfected patient cohort, using a p-value cutoff of 0.05 with Benjamini-Hochberg multiple test correction and controlling for the random effects associated with publication and patient. The significant gene ontologies were comprised of 6 depth 1 parents that are involving catalytic activity, binding, metabolic and cellular processes, biological replication, regulation, and interspecies interaction between organisms. The significant terms of interest associated with COVID-19 include hydrolase and transferase activity, transferring phosphorus, and nucleotidal transferase, and ion binding. Additionally, results from the Dirichlet multinomial mixture clustering analysis using all 13,534 gene ontologies resulted in a best model fit using 3 distinct DMM clusters that were significantly associated with each case type. The taxonomic analysis revealed a statistically significant decrease in the log2 median ratio of several species belonging to the genus Sphingomonas when compared to both the uninfected and CAP cohorts. These findings support previous reports regarding the association of the healthcare-associated pneumonia genus Sphingomonas and significant gene ontologies derived from the Sphingomonas proteins in the COVID-19 samples were hydrogen peroxide, catabolic processes, responses to oxidative stress, catalase activity, heme binding, and metal ion binding. The catalase protein decomposes hydrogen peroxide into water and oxygen and serves to protect cells and toxic effects. So as a discussion point, perhaps the Sphingomonas is responding to the COVID-19 conditions in the patient by expressing genes to help it survive well under the COVID-19 disease. We then conducted a subsequent Maslin-2 analysis amongst the COVID-19 samples with known survival outcomes, revealing notable functional profiles associated with phosphate, phosphorylation, metal ion binding, nucleotide terms, and lytic activity, or hydrolase, endopeptidase, etc. These findings were similar in nature to the gene ontology terms associated with COVID-19 versus the uninfected and community-acquired pneumonia patient cohorts. So collectively, while this data does not speak to causality or directionality of the association, it does demonstrate a significant relationship between the human microbiome and the severity of COVID-19, rendering a further testable hypothesis that warrants further investigation. So, in conclusion, we observed unique taxonomic and functional discriminant features in the bronchial alveolar lavage metatranscriptomes associated with COVID-19 and deaths, with taxa of interest, including the genera from the Sphingomonidaceae class, and functional annotated gene ontologies of interest, including phosphate, phosphorylation, metal ion binding, nucleotide terms, and lytic activity. So with that, I'd like to thank the Covert Microbial Subgroup team members and give special acknowledgement to John Fauner and the Texas Advanced Computing Center at the University of Texas at Austin for providing the high-performance computing resources that have contributed to the research results that were reported. I'd also like to thank my PI, Dr. Kirsty Agard at Baylor College of Medicine and Texas Children's, Mike Lee at NASA, Victoria Zakas at the University of Chicago, Kristen Curie and Todd, and the entirety of the Triagen Lab at Rice, and our fearless leader, Krista Turnus at Signature Science. So with that, I'd like to thank you all for listening, and I will be available to take any questions.
Thank you, Michael. Do we have any questions, Cecil? We have a question of what are your next steps?
Yeah, so next steps are to bridge the gap between the taxonomic associations and the gene ontologies with the uniprot protein ID counts and really take it to the next level beyond just an association and draw a real relationship.
Great, thank you.
Okay, thank you very much.
Afshin?
Yeah, Thank you, Kyle. And that concludes that session of great talks from Michael and Riza and Brian and Cassie. I want to say that so far I think there's been a lot of great ways of showing how Covert has been doing such great work. And I also want to point out that, Covert's about open science, right? So a lot of this stuff majority of stuff has not been published yet and is in working manuscripts that are going to be submitted in the next, few weeks or month. So that's the goal, is to not only be able to share everything, but be able to share with the public even before it's published. So if anyone wants to ever join Covert or collaborate with these various groups that you've seen and we'll see in the rest of the afternoon, contact us, and you could, Covert's open for anyone to join. Okay, so without any further ado, I'm going to share my screen. So just to briefly go over what's left of today and the next lightning talk session. I'm moderating this next session. It was about therapeutics, drug targets, risk prediction, and long haulers. And so I'm going to start actually giving a talk about microRNAs, and then Elena Casaravi from University of Milan will talk about some nice AI features relating to how to make diagnostic for the clinic. Dr. Robert Miller will also be talking about some ways of sequencing data and looking at COVID RNA sequencing data and different ways of how that is impacted by impacting patients and different pathways being regulated. And then Sonia Villapol will also finish off by talking about CNS issues or central nervous issues in COVID. So let me stop sharing this and I'll share my presentation. Okay, so here we go. Go to the beginning. Can't reveal my punchline yet. Oops. Revealing my punchline. Need to scroll up. Okay. Okay. So we got to keep track of time and I'll try to be within the time. So as I said, what I'm going to be discussing is a paper that we're hopefully going to be submitting in a week. So the preprint will be available on a bioRxiv or one of those preprint sites. So everything here is unpublished, but again, this is the spirit of open science and what Covert's goals are, to actually show data to the public and hopefully get people interested to work with us and work with everyone here and promote, the quicker way for everyone to get out of this situation, the pandemic. So I'm focusing on microRNAs. Earlier, you heard Paco from University of Lisbon talking about long non-coding RNA. So what are microRNAs is another form of non-coding RNA, meaning RNAs that don't translate to proteins. So microRNAs, why worry about them? Why care about them? Traditional microbiology view was DNA, transcribed to RNA and translated to protein, putting it simply. But around 2000, well, the first time someone discovered a microRNA was 1993, but around 2000, people really started paying attention to it. Before then, people thought these microRNAs are just debris. So that's why it's largely ignored. But then people started discovering these small RNA that are around 22 nucleotides, approximately, depending on the microRNA. They could range from 18 to 25 nucleotides, sometimes a little plus or minus, 5 nucleotides. So they started discovering that these small microRNAs can actually regulate lots of genes or messenger RNAs. Could be like hundreds and hundreds of thousands of genes, depending on the microRNA. And not only do they do that, but they're, as you see, the new understanding is since 2000s is that they're pretty much involved in every step of transcription to translation to even directly impacting DNA, and DNA can impact microRNAs like methylation, for example. So this new understanding and the extreme important regulation of it is actually something that people started paying attention to. And because of their size and stability, that they could actually are floating freely in the body. They could be found in every fluid in the body, from your blood, saliva, tears, urine, so on. And then they're also been known to be involved in every aspect of disease, but there's also good microRNAs that you want to be floating around your body, helping your normal biological functions. And not only are they found in humans and mammals, but in every living thing that you know of, from plants to bacteria, as you heard Michael Yoakum talk about. There's microRNAs involved in there, and even viruses, which I'm going to talk about. And not only that, but they're found in C. elegans, as I said, fruit flies, so on, insects, and they're highly conserved between species. So this comes into play why microRNAs might be important for viruses. So what is the virus relevance of microRNAs? So this is kind of more of the Wild West territory, because 20 years of biology or 20 1 years of biology is you're still, really relative infancy of how you can view or how things have changed. So microRNAs are in that stage. But in the virus microRNA literature in the past decade, people started discovering some interesting things viruses do with microRNAs. For one thing, it's been shown that viruses can integrate microRNA genome into its machinery, actually integrate it. So, and it uses that to actually either for replication or also to evade the immune system. And one way it would evade the immune system, now there's a recognizable human genome integrated in the virus that now, let's say that when the virus, this foreign object comes in your body, the human body might recognize that human component and not think it's a foreign object anymore. So it uses that machinery. It's kind of a clever machinery, uses and a weird one, but it does it. And due to the highly conserved nature between species, microRNAs can also, people have seen in the virus literature that maybe this might be one mechanism it can use to transmit between species. One paper I just want to really highlight is that this microRNA 122 in the hepatitis C virus literature, this is a couple of years ago, they actually found out that indeed this one microRNA is incorporated into the hepatitis C virus and is the true driver of everything that this virus does. So this now leads to, can there be a similar mechanism in SARS-CoV-2 with microRNAs? So just before I start, as I said, Covert is a really highly collaborative group, people from all disciplines, and I just want to show, this is a large group working together for a common goal, and every of these subgroups have this. So these are all the people involved, and I hope I didn't leave anyone out from the paper that's going to be published. But as you can see, I think this is the way science should be done. Everyone working together, large groups, and not just one lab isolated and focused together. So this is like a general overview of the story I'm about to tell you. So I identified this one microRNA that early on through transcriptomic data that seems to be driven or from the transcriptomic data in the host seems to be driving a lot of the responses. But as we started looking at it more and more, we found this whole story that's here and I'm about to tell you. So early on, when there wasn't as much sequencing data publicly available, me and Rob Miller, I'm here just to put out, who's involved with every slide. I put pictures of people that are helping out for the slides so you could actually know who did what. But as you know, early on in March, that there's a much publicly available sequencing data, but there was a few out there, and Rob found this bronchoalveolar lavage fluid Rna-Seq data set that's from the Wuhan, China population. And so we started analyzing that, and lo and behold, he ran the Rna-Seq, and I said, oh, let me use my bag of tricks and try to predict what microRNAs might be involved. So reverse engineering from the transcriptomic data, from the gene level. And what we saw, indeed, that I predicted from these microRNAs here, small, but that were probably involved, prediction. From these microRNAs, all of them but one was predicted to be inhibited, and the one that wasn't predicted to be inhibited is MIR2392. As you know, the higher numbers you go, or you might not know, the higher numbers you go for microRNAs, the less is known because the numbers sequentially. So, but running prediction tools, again, I see that this microRNA is involved with heavy mitochondrial suppression. Doug Wallace is later on in another session going to talk about mitochondria and SARS-CoV-2, so stay tuned for that. And also then downstream of that, all these different pathways and different effects that we know, the virus causes in the body. So, this led me down to further look to see if this microRNA is actually integrated into the virus. So with Todd and his very talented grad student, Nick, they said, all right, can you guys map this microRNA genome onto the SARS-CoV-2 genome? So that's exactly what Nick did. So Nick then took that genome and then looked at all the different variants. You see the ones that Duncan McConnell talked about and all the various other people earlier this morning talked about, from the P1 to the 117 to 135. And indeed, what happened was, in microRNAs, there's a thing called the seeding region. It's an 8-base pair seeding region. And that's the most important part of the microRNA, where it uses to bind to genes, to silence them or promote them and cause the havoc it needs to do to, the microRNA to do to actually then, to cause all, let's say, the pathway regulations and so on. I'm going to show you. So in the microRNA gene virus literature, that's the part that's should be, if it's integrated in that genome of the microRNA, should be integrated in the virus. And indeed, when Nick mapped it to all these different variants, starting from the reference variant from Wuhan, so we saw that indeed there's multiple regions, 3 top heads he saw, but multiple regions, 8-base pair perfect mapping of that sequence. And one of them is the NSP2 region. And talking to various other Covert members like Joe Guarineri and other colleagues, they informed me that, yeah, the NSP2 region is not as much paid attention to, but it's people have said it's involved with replication. And as if you remember earlier, SARS, and it said one mechanism that viruses use to incorporate the microRNA is for replication. So that made perfect sense why that's there. And then there's other regions like NSP3 and NSP2, and that's at 8 base per region. So having all these multiple regions mapping so well to SARS-CoV-2 and it's variant independent, it actually, it's not random. And if you let's say go to the 7 nucleotide as opposed to 8 nucleotide, then you see mapping onto the spike region and other regions. Now, is it conserved between species as microRNA? So this is now just hypothesis driven, looking at the sequence and mapping. And indeed, you see that, with primates is highly conserved. What's interesting with mice, it's not. And if you know about the mice model and SARS-CoV-2, with mice, it goes directly through the mice and doesn't impact them at all, doesn't phase them. So that could be maybe an explanation why. With bats, there's partial conservation, which could mean the bat is a carrier because they don't get sick, but they carry it and they pass it on to humans, which is what the theory is here. I won't get into this for the sake of time. So I'm going to go on, I'm doing a big data dump on everyone. So, but just bear with me because I go really quick to the story. So we only have 15 minutes of lightning talk. So what we see here now is just looking at downstream targets from Chris Mason's group. He sequenced lots of data from the New York population, so nasal swabs from different viral loads. And then more recently, a paper that he submitted available on bioRxiv. He had sequenced patient autopsy data from COVID patients. So we got heart, kidney, lung, liver, lymph nodes. And what I did here was we took that process sequencing data and I mapped it to MIR2392 gene targets. And the long story short here is basically we see overall pathways and genes being regulated in different organs and the nasal swabs that correlates well with SARS-CoV-2 pathology. That was really cool. So this is one of these experiments that now functionally want to show. I think this experiment is one of the highlights of the paper before we get to the patient data. And this was done by Rob Miller and Eliza at Morehouse School of Medicine. And then Eve and her Ermi, they actually had analyzed some publicly available monocyte data, which I incorporate here. So what they did was they took MIR2392, overexpressed it in a human cell line, a neuronal cell line. And the goal is if this microRNA is truly driving everything that I'm saying in a host and causing all these downstream effects, if you just overexpress this in a cell, can you reproduce the same exact things as you would do without a virus? And this is what we did here, or I say we, meaning Rob and Eliza. And then they sequenced it, and then I tackled this analysis. So what we saw here, this is a data set that was from a publicly available sequence data set that someone had done on a cell line, A549, overexpressed, they infected the cells with SARS-CoV-2. And just pay attention to the top row. This is the MIR2392 experiment overexpressed in these cells compared to the controls, meaning the cells in the normal conditions or a vehicle controlled condition, whereas a vehicle where they've been infected by just some random thing that's not MIR2392. And what we see here is that indeed this is the fold change values in the genomics, transcriptomics, that we see a positive correlation, it's significant positive correlation, which is what this is a correlation plot of, with all these different conditions that the cell was infected with virus. This is the pathway version of that. And indeed, like there's when you look at patient samples, this is the autopsy data and nasal swab data, looking at viral load versus negative, you see a positive correlation from the patients themselves, just by having this virus being not virus, but just having this microRNA being overexpressed in the cells. When you do downstream pathway analysis further, you see that indeed SARS-CoV-2 pathways are being actually positively up. This pathway is saying this is upregulated in these cell lines. So the pathways that are supposedly related to SARS-CoV-2 infection are up, which means that if I gave this sequence data to someone and I didn't tell them that it was not infected with the virus, someone would tell me, once they say you do downstream analysis on this, someone would think these cells had been infected with the SARS-CoV-2 virus, while all we did was overexpress MIR2392, which might lead to the fact that when you do predicted diseases for this, you see this microRNA is related to nothing good. If it's overexpressed in the body, and the downstream gene targets, you see that it's related to all these different types of diseases, which actually, when you do literature searches, that we see are symptoms in patients that are infected with SARS-CoV-2 and have even long-term COVID effects, from cardiovascular effects and things we know to actually like male infertility. If you actually do a literature search, you see that some COVID patients have reported that, to deafness, even to vision problems, and so on. So again, I'm running close to the end of the time, but I'm going to get to the climax of this story right now. So this is now going to patient. So if this microRNA is truly what I'm saying is it doing, is it circulating in the body? So with collaboration with Mohammed, he was really key clinician at University of Maryland, working with Matt and his team, Marissa, and his collaborator Anthony there at University of Maryland to actually get me a lot of patient samples from the serum and the nasal swabs. And then a collaboration with Jonathan and Matt and their Matt's lab person, Susan, they were able to get lots of urine samples to see if this microRNA is actually floating around the body. And then with collaborating with Chuck and Broad and me, we quantify this using droplet digital PCR technology. So what we saw indeed in the serum, this is just the overall comparison, not looking at specific details, but looking at positive or negative. We see that indeed in the serum and in the urine, they're positively, if a patient has SARS-CoV-2, this microRNA is positive or significantly increased compared to the negative. What we see in the nasal swab samples is something different, which might indicate, that's the microRNA doesn't really get expressed until the virus has taken hold, basically, in the lungs and is circulating, and it's going to a higher viral, or it's not necessarily there until the virus is having the full effect it does in the body. At least that's a hypothesis. This is an ROC curve looking at like patient survival and sensitivity, and you can indeed see that that's very significant with more severe cases of SARS-CoV-2 in the patient. And this is just kind of a breakdown. So in the serum, you had patients that were intubated, patients that went home, and this is negative. Same with the urine. This is the true negative population, healthy, nothing. Outpatient of SARS-CoV-2, and then inpatient with a negative population, inpatient with a positive population of the urine. So it's significant all around, except for the nasal swab. When we compare to other coronaviruses, it doesn't have any significance there. And it's more the level of the copies of microRNAs that are floating around are more level of your serum. I'm getting close to my end of the time. So this is now the real climax. Can you use this for therapeutics? And indeed, you can. So this is where collaboration of Nushri and Matt and Big Boom. She really quickly, Nushri has this platform called FAST, which is if you give her a antagonist to a sequence, you can actually she can make a inhibitor for it or a delivery system for it. So we gave the antagonist this MIR2392. And then we worked with Matt Freeman at University of Maryland. And what they did was, they did the viral screening with A549 cells in the dish. And what we see, this is the dream curve of a pharmacologist that they're like. 100% of the inhibition of the virus was 0% cytotoxicity. And these are 3 different replicates, biological replicates. So indeed, that you can get pure, inhibition of the virus in a dish. Now we run a pilot hamster experiment. So what we did here really briefly, we did intranasal application and then an IP intraperitoneal application. And this is 1 day before viral inoculation is the red, and then 1 day before, 1 day after viral inoculation. So this is our first time doing it. Same with the IP, and this is just the PBS control with like nothing, no inhibitor in it. And indeed, what happened was day 1, we see significant reduction. This is an oral plaque forming assay for the monovirus in the hamsters. We see significant reduction of this virus when we apply this inhibitor. So this is our first time playing around with concentrations. We didn't give it every day, but as you see the second day, the intranasal application is still, there's some residual inhibition of it, but day 3, we still didn't give it. So that's now resolving back to normal. So now we're working on experiments to start giving it daily dosages and at a higher concentration and so on. So we got to play around with the dose, and we're doing all the studies to actually try to figure out the optimal dose we do in an animal to then translate to human, as opposed to in vitro studies. We nailed it there. So what is next? So, we're working on the investigation of new drug studies, enabling studies to actually then be approved by the FDA so we can do clinical trials. So this involves animal experiments. But the main thing that I want to get across before I stop is that if anyone's interested in helping with funds or collaborating this to push this clinical trials, I think this is really important. And this could be maybe a novel way to inhibit SARS-CoV-2. So that's really the gist of it, and quick data dump on everyone here. And then I have to probably stop and I'll answer your questions in the chat because I went over by 3 minutes. But this is the team, of course, as I want to highlight them because none of this could be done without the team. And the papers hopefully be submitted in a week so everyone could read the real nitty-gritty details. And of course, all these people involved, all these nice entities were involved with helping with some funding, and then these other people had individual funding that helped out too. So that's a really quick synopsis of microRNA, or this microRNA involvement. So let me stop sharing. Let me see if there's one question. I'll answer one question that has been upvoted. Did you try to map miR2 to other known viruses like hepatitis in the sequence? I have not, but that's a good idea. So, that's the one thing that's, there's a lot of work to see if this microRNA is specific for SARS-CoV-2 or if it's, can it work for other types of viruses too? But I'll answer other questions in the chat as then I'll type in. But before I stop, I want to introduce Elena Kazaravi. And she's from University of Milan. Are you there, Elena? Yes. So if you want
Yes. Hi.
Very good. So if you want to start sharing the screen, introduce yourself very briefly and take it away. And I'll give you like a 3-minute warning.
Okay. Thank you, Afshin. Thank you, Afshin and Todd, for inviting us. And we are the Anacleto Lab from University of Milan. We are, sorry, the paper went on. We are the polyclinical hospital from Milan, the emergency department and the radiological department. We are the BBOP from the Berkeley laboratories and the Robinson lab from the Jackson lab. We are here to present our research, which was aimed at developing an interpretable machine learning classifier for COVID-19 risk prediction in the emergency department. The work was specifically requested by clinicians from Polyclinical Hospital in Milan, and it was meant to optimize the patient flow, improve the patient treatment, and reduce the waiting times in emergency department. To develop the model, we integrated all the data they provided us, which consisted in clinical, laboratory, biochemical, and radiological data. For radiological data, I mean the radiographic images which are preferred, at least when there is a big wave in the emergency department. Our aim was to develop, and what they asked for was the development of a method which was interpretable, which means that its prediction should have been explainable and easily deployable in the emergency department. We did all our best to follow all the main steps of our algorithm, to compose them so as to provide a robust and exhaustively tested model. In details, if we go through the steps of our model, also considering the problems we have to cope with, which often affect medical data set regard. We need some missing data imputation algorithm to input the missing values, because often we have missing values in our data set, and this happened also in our data set. We then pursued a robust feature selection technique by experimenting different models. We went through different prediction models, and then we discarded those that achieved the lowest relevance. And we tried to avoid all the unbiasing, which were highlighted by Winans and others in their paper, which is assigned in the bottom of the slide, and which mainly regard the data acquisition problem, which is often not coherent, which is often collected in a time which is not coherent with intended time of usage of the system. A missing follow-up, which doesn't guarantee that the classification of the patients are stable, are sure. A missing description of the hyperparameter setting or no hyperparameter setting for the models, and a biased model training and testing. This is, in a nutshell, the pipeline of our model, which was developed after a dataset acquisition and cleaning, a step of missing data imputation, and then 3 steps: feature selection, model induction, and model explanation, which were operated through tenfold cross-validation to obtain an unbiased estimation. So, in brief, our dataset, it was collected in the period of in the peak period of the first wave in March, from the 7th of March until the 10th of April 2020. Patients whose data was collected were positive to COVID-19, and they were followed for 5 months. After collecting the dataset, we discarded all those variables for which 50% of the data were missing or who had negligible variance. Overall, we collected 301 patients. The variable with a mean age of 61 years, 7 days of symptoms, mean 7 days of symptoms, and from those patients we collected laboratory measurement, which you see, some of which you see in the following in the slide, and some Boolean variables which describe symptoms, comorbidities, and some counts, which we collected, and then 4 radiological variables. On these radiological variables, I have to say something. 2 of them are from humans. Other 2 are from an automatic method. We decided to compare human score to automatic scores computed by a deep neural model to check if a coherent automatic system could be not better, but could be used instead of a time expensive radiological evaluation. So the first 2 scores were evaluating the severity of covert pattern and the extent, and the same from the automatic score, which were derived from the application of COVIDNet, which is a deep neural network which was applied after transfer learning on our images. After composing the dataset, we had to investigate. We had to go through some issue which regard the missing data imputation. When we have to deal with missing data imputation, the first thing to do is to understand the missing data pattern, which may be, I will go briefly on this, but the missing data pattern can be of 3 kinds. Missing not at random, which means you have a reason for the missing data mechanism. And you can understand this by applying simple tests on a data set composed of Boolean variables, where the true values correspond to the missing values. The same applies for missing at random and missing completely at random. That is, we have also statistical tests to check the difference between missing at random, which means that the probability of me being missing is the same only within groups defined by the observed data. For missing completely at random, which is the easiest to define, this means that all the data have the same probability of being missing. After performing our test, we understood that our data were missing completely at random. So we could experiment 4 different algorithms that perform missing data imputation. One of them it's really easy. Picks the nearest neighbor of each point with missing value and samples one neighbor and use the value of the missing neighbor as there is a chart which I don't see, and picks the nearest neighbor and use the data from the nearest neighbor. Other 2 methods exploit random forest. The random forests are trained on the observed data to predict the missing data, and the other one is based on a maximum likelihood estimator plus random error. So when we have different models, how do we test a different imputation model? The base of the imputation model is that none works, essentially. So when you apply an imputation model, you iterate the imputation model, since all the recent imputation model contains some randomness. Every time you will have a different value, inputted value. So you can compare essentially the between imputation variance from subsequent imputation. We applied this, we computed this between imputation variance, and we could understand that the model based on maximum likelihood obtained a very, very high compared to the other between imputation variance. You see the scale in the image. You see that this 3 is almost even 50 times predictive mean matching and the other random forest based method, and what we call mice PMM, which I didn't describe for lack of space, are even ages less stable than Miss Forest. So we use as master imputation model Miss Forest. Then we went through feature selection, which was similar and not so easy. So after testing different models, Boruta feature selection method resulted as the most stable and the most effective. Boruta feature selection essentially play cards because it takes the original feature set, it shuffles it and produces a shadow set, joins the two sets and trains any classifier that can provide a feature importance with a new set which contains the shadow set. After the training, each original variable obtains an importance which is compared to the importances of the most important shadow feature. Those variables, original variables that get an importance which is higher than the most important shadow feature win. Those that get a lower importance lose. After repeating this several times and applying a binomial test which checks for a significant number of wins and losses, we can detect those features that must be removed because they lost a significant number of times. So they are at all not informative at all, so we discard them. Those that won for a significant number of time are important. Those that are still under consideration iterate through the through the process. The problem is that when you repeat Boruta several times, at the end you have to stop. And at the end you have always some tentative features which won but not so much, essentially. To solve this tentative drama, we operated also Boruta through the full cross-validation, and we selected those variables at the end which were important enough, that is, that were selected as important or tentative at least half of the time. We're almost finished. After this, we can go with our classifier. So in each iteration of the tenfold cross-validation, we proceed with the feature selection on the training set, and then with the selector feature set, we train on the training set random forest whose parameters were chosen by grid search, which is a greedy search algorithm which is the most feasible when we have a limited number of samples, as in our case, and whose other parameters were chosen either based on clinicians' advice or based on experience. And we applied a kind of random forest which is imbalance aware. The random forest, as well as the following classifier we tested, which is an associative trees, which can combine, which is derived from the random forest with the aim of making the random forest more explainable, can both provide a feature importance. So
Later, you have 3 minutes.
Yes.
Sorry.
This is the schema of the associative trees, which essentially is a really clever system to make random forest more explainable. Random forests are forests of trees. Trees are explainable decision trees. They provide the rules. But when the trees are many, the explainability of random forest become a bit limited. So we try to experiment also these associative trees, which are obtained by unrolling random forest and then pruning the rules, and then apply a greedy algorithm to discard those rules that are repeated or not important. All these models were compared to something you should well know, which are generalized linear model, which I don't describe because they are simply the extended version of logistic regression. And at the end, we tried different combination of Miss Forest, which was the best imputation algorithm, and a prediction model, and other combination of mice random forest, which is a sort of Miss Forest operated multiple times by adding more randomness, and our prediction models. The most stable and most efficient classifier was the one using mixed forest plus random forest. And this is proved by the area under the curve and by also the F1 score and sensitivity and specificity, which are quite balanced. And this is important, especially in our data set, which was highly unbalanced. It had an important unbalancement. What especially clinicians wanted was a feature relevance. So they said us, which is the feature I have to account more when the patients come to the ER? So we also provided them with this feature plot, which is quite stable, and it's currently applied in the emergency department to select most patients at highest risk. We also provided them with some cut points, which are simply found by bootstrap sampling, by applying some bootstrap sampling and finding the best cutoff, and then computing the mean of the cutoff over multiple sampling. If you want to check on the code, we have the code, but it's at your own risk because it's not committed, but by tomorrow we will start to do that, and we will provide a very nice implementation with comments. And we are beautiful, and this is us. And if you have any questions, here I am. So questions for Elena. Afshin, do I have time?
Maybe one really quick question. If you can answer it quickly, I'll maybe you can answer the top one here.
Did you try any other approaches to feature selection? Yes, we tried recursive feature elimination, and we tried also univariate analysis. Univariate analysis, both of them obtained results which were not consistent with the random forest, and obtained the result that also clinicians suggested as being too restrictive. The other question.
I think that's it for the questions. Can you type in your answers to the rest of the questions?
Okay. Yes.
Perfect. Yeah. Thanks. No, wonderful talk.
Thank you.
And I know it's getting late there for you in Milan, so.
Yes.
So,
okay.
Thanks for sticking along. Yeah. Okay. No, great talk. Great talk. Thank you. And then next up is Rob Miller from Morehouse. You already saw his picture, but now you can see his face and hear him talk. So I'll let you take it away, Rob. Thanks.
Trying to get my screen to share. It's not doing it. One moment. It worked earlier.
Yeah, you did test it earlier. Let's see. Special. I don't think you have any. Is it giving you errors on sharing?
Oh, that's interesting. Do you want to have the next speaker speak? For some reason, my display is not sharing, and I'll relog in.
Oh, okay, so yeah, let's. I don't know if Sonia, are you ready? Then we could swap between Sonia and Rob. That's probably a good idea. Sonia here. Yeah, Sonia, you're here. Would it be okay if you go next?
Yeah.
Okay, yeah. Great.
Yeah, can go.
Perfect. That way we'll save on time. Yeah. So Sonia Velapool is up next. She has a great paper she put out recently. So I think you'll be talking about that and CNS issues and other things. Okay, Sonia, thanks.
Yeah, let me see. PowerPoint. Sorry, I was waiting for Rod, but 1 second.
Yeah, we put you on the spot.
Yeah, it's fine. Already.
Sorry.
Can you allow me to share? Yeah.
Yeah.
Oh yeah, I can.
Yeah, you should have privileges. Yeah, you should be good.
Oh, it's here. Okay, good. Okay, so I'm going to talk about the long COVID.
Good, perfect.
Yeah, is that okay?
Yep, perfect.
Yeah, so I'm going to show briefly our work on long COVID that we're studying. So I'm collaborated with several people here covered in the neurology subgroup, but and also related with microbiome projects. But we find out that we saw a lot of patients that they here at my hospital in different other places, also and other collaborators that we have. So they show some neurological problems. So we performed this meta-analysis to look at the long-term symptoms and effects of the disease. And that is called, is the new name that the NIH established a few weeks ago in February, end of the February, that they call like post-acute sequel or the SARS-CoV-2 infection or past. That is the same that the long COVID, but they adopt this new definition because long COVID doesn't mean post-COVID because it's still the sequels of the COVID. So I want to show briefly what is that and why that is really important. So as you know, a lot of patients that who have they suffer like a mild or moderate or severe COVID, they have a lot of symptoms weeks after or months later, and all these symptoms that are defined as the long COVID. So the most important the most important is the fatigue. Most of the studies they show that, and then they call the brain fog or the sleep disorders, etc., fever, gastrointestinal problems, anxiety, a lot of neurological problems. That is the thing that call our attention. So we don't know yet the magnitude all these symptoms, and we want to do a study, a meta-analysis on the literature, what was described so far. But we think that probably, of course, that will be a huge public health impact on the population because maybe it's can affect more than we expect. So we know that most of the patients, they full recovery after the COVID, the acute disease, in 4 to 6 weeks. But some studies, they show that they consider that this long COVID after 2 weeks when they passed the diagnostic because they were tested negative, but after 2 weeks even they show some symptoms. So the timing is a little bit is not very well adjusted, but the concepts say that is after 6 weeks that we can consider like long COVID, 8 weeks and 12 weeks. So this data is pretty new. Like there are the major studies that show that 7 in 10 people that they were in the hospital, they were not full recovery after 5 months. We are extending this to 5 months post-COVID, and the factors that are associated with this more severe long COVID is there are the gender. So there are female, there are the middle age, the white ethnicity as well is associated. 2 or more comorbidities that was important to development severe long COVID, and then that more the severity of the disease also is the more relevant feature to development this. We published this paper, well, it's in Bioarca, in the end of the January. That got a huge impact, even still is in the review. But we want to show a meta-analysis that was presented in the literature during in the last year. Like we did all this, the recompilation of these works until January of this year. So it's more or less a few months since that we know about this disease. Right? So it's not completely everything. So far during this month, we identify around 18,000 works that they were reporting some kind of the long COVID effects or symptoms. And then of those, we exclude some of them because they were smaller studies. More of the studies, they were less than 100 people. So we had to discard that were not enough significant as well. We discard reviews, letters, comments, etc. So in the end, we select a total of 15. And these 15 studies of this huge meta-analysis. So we identify 15 long-term COVID-19 effects. and the people, the total participants, or the people, patients and participants, because they were people that were not in the hospital, some of the studies. So in general, were around 47,000. So we did this meta-analysis, and we identify, for example, using the PRIS guidelines, symptoms that they were very common, like fatigue, and that's the most common, the itch, hair loss, dyspnea, depression, anxiety, a lot of neurological problems, memory loss, and hearing loss, or another similar related, effects related with the audition, and we classify these symptoms and with several statistical programs, and we conclude that, for example, around 8 or 10 people, they show at least one symptom after recovery. So 80% of the total of these participants, they show some kind of the problem. Also, we found some signs or some biomarkers that were described in these studies, like, for example, the C-reactive protein that corresponds to 8% of the patients, the serum ferritin as well, the D-dimer. So could be indicators as a biomarkers of this long COVID, but it's not like a spectrum. It's not very well to identify, because the percentage as well that are low. That means that there are some symptoms that are transitory and they are not permanent, or they are not related with chronic inflammation or systemic inflammation overall. So and the most important as well, so is the abnormal chest, that is in the 54% of the cases. So that most of the patients, they suffer months after respiratory problems. When we look at in general all these symptoms, this is a schematic of the results. So we see that the fatigue, and that was in all the studies, all the studies that we are analyzing, the fatigue is the most common, and that also is the one that lasts more. So it's chronic even until 3 months, that it was the extension of their studies, but there are studies after that they show until 9 months, and the people still had this fatigue. And then the head on the attention disorder specific, and that was really relevant in kids as well. The hair loss and other symptoms that, cough, etc., nauseous, gastrointestinal problems, that can affect different systems. And this schematic is more, can represent most the different systems on the body. So something that paid our attention was the percentage of the numerous symptoms that are related with the CNS, right, with the brain. So since anxiety, depression, that can be because the effects of the inflammation post-COVID, or even all these neuropsychiatric problems that there are because the pandemic and all these COVID effects that is not related because they passed the COVID, not is related with the situation, of course. So it's kind of difficult say what is the reason or the cause. But it's true that there was an increase as well the stroke, and that is something a little problematic. So people were recovery. 3% of the population they suffer ictus or some cardiovascular problems post-COVID, and that is irrelevant because they don't have any kind of the comorbidities that they are predisposed to suffer these cardiovascular problems. So that was another thing that was important. Also the post-traumatic stress disorder and as well. So cardiovascular problems, the cardiovascular system was really affected, and we identify as well joint pain and all these digestive problems that could be related as well with the change in microbiome over time, diabetes, and for example, some that is very curious that we didn't expect that, but for example, the 1% of the patients, they developing a new hypertension months after past the COVID. So that was something that the people should check, because the hypertension is difficult to detect, and we don't have any kind of the predisposition. So that was the original idea to generate this analysis to like a call out to the clinicians to identify all the symptoms that the people didn't suspect or didn't suggest that they have. Curiously as well, there are a persistent neurological system. This is another paper that was published a couple of weeks ago. So they're related with cognitive dysfunction, but were patients that they passed the disease with mild or moderate symptoms. They didn't have to be in the hospital, but months later, they developed some neurological problems, like, for example, the cognitive dysfunction. They are confused, they cannot focus on anything, blurred vision, and the dizziness that is pretty common, that is associated to the fatigue. So that is most than 0.5% of the percentage that this thing can happen, and that is something that is feeling all these problems in the hospitals with the units and the people. They need to return to the hospital, need to be hospitalized after COVID when they didn't have to be in the hospital when they passed the acute COVID. So that it's a huge problem that that the health system should be prepared. So that is something important to think about. So as you know, the Congress provide a lot of money to investigate all these causes and to investigate more about the long COVID and the post-acute COVID, and they assigned on the first round 1.15 billion of funding for the next 4 years, and then this is the first set of the money. Then the next cycle will come with more, because it really is a need, and we need to answer some of these questions. So the NIH, they have this spectrum of the recovery, so they don't know exactly how many people will recover after COVID, how many of them, this kind of the percentage that we don't know exactly. So it's necessary to know more statistics about that. How many of these people continue to have symptoms and for how long, and we don't know exactly. As well, we don't have that much idea about the underlying biological cause of these prolonged symptoms. We don't know exactly if there are a genetic cause or something like that. So we don't know the people that is vulnerable of that. We know that the women, for example, they get more affected in the middle age, but we don't know exactly, we don't have statistics about this. And then we don't know if the SARS-2 infection titer change in the body that can increase the risk to other conditions. And that is related, for example, with the neurodegeneration and neurological problems, that if there are this inflammation, it can persist over the months, can develop some neurological conditions that related with neurodegeneration that can be, for example, Alzheimer's disease or Parkinson's disease, that we know that happen in other viral infections in the past. So all these questions need to be some answer. Yeah, I know. So I just want to thanks to this work has a huge impact, especially in this Hispanic network, like in Latin America and Spain, and in everyone, even the clinicians and then the public was very interested about all these symptoms and all these effects. So I want to thanks to all the authors of these papers and all these publications, and of course the neurological group here and the COVID-19 International Share team. And that's it. I have 1 minute left for some questions.
Keeping track of the time too. Okay, so there's a question here in the Q&A. Says, do we know the proportion of COVID-19 patients experience long-term symptoms, or what proportion, I should say, of COVID-19?
Yes, it depends what kind of the symptoms, but the more, for example, the fatigue is 60% of the patients that recover, but it's a lot. That means it's a less 80%. 8 patients or 8 people that recover from COVID, on 10. So this is a lot. Someone has some problem over time, like it's not that important or more important, but it's an important number. So it's an important proportion.
Maybe one more question and we move on, but I'll ask Eve's question. Focusing in on children, what proportion have long haul and what symptoms?
Sorry, I didn't understand the first.
So Eve says, focusing in on children, what proportion have long haul and symptoms?
Oh yeah, that is a very good question as well. So that children pass the COVID like with my symptoms or asymptomatics, or unfortunately that is like that. But curiously, the teachers in the schools there find that these children, they have some neurological problems as well. They lost attention, and they identify now these children like with problems that they pass the COVID. Nobody even realized they have the COVID, some of the cases. So that is around 58%. There are a couple of studies. It's not a meta-analysis, it's another studies, but 58% of the children, they have some long COVID problems. So that is another call that we need to pay attention to that. Yeah, it's very important.
Great. Can you answer the rest of there's a couple more questions. I don't know if you want to type it in, and then we'll move on to Rob. But thanks, Sonia. Great talk.
Okay. Thank you.
Rob, can you share your screen? Oh, yeah, now we see it. Perfect.
Now we see it.
I'll give you a 2-3-minute warning too. So
okay, great. So I'm Robert Meller. I'm an associate professor at the Morehouse School of Medicine in Atlanta, Georgia. And what I'm going to talk about is host transcriptome responses in COVID-19 patients. And in particular, we're interested in how the host transcriptome response can actually be used to identify patients who actually passed a PCR test. So this data is all from Chris Mason's data that was published in Nature. And this was a study of around 800 plus samples that were subjected to RNA sequencing. And in them, there was about 700 samples, which were human nasal swab samples that were sequenced. And we were able to look at both the SARS-CoV-2 aligned reads and also the human data. And actually, when we do that, we find something quite interesting. So these patients in blue, they were all designated as being PCR positive for SARS-CoV-2. So they would have been quarantined and subject to normal restrictions. Whereas these patients in red were those who would have received notification that SARS was not detected in their test sample. But what we did is we then aligned the PCR data to the sequencing data. And surprisingly, what we actually found is there were a number of reads, about 10% of the PCR negative reads, actually we found trace or number of reads aligning to the SARS-CoV-2 virus genome. So we split this data down a little bit more. You can see that the PCR positive samples were allocated high, medium, and low categories. This is based on the CT count. So the high data was typically less than 18 counts. The medium was 18 to 28, and then the low was greater than the CT value of 28. So if we look at this data further, we can see there's a large proportion of patients who were negative, but then we also see there's this proportion here, which were PCR negative, but we could detect SARS via the virus, via sequencing. We also found that there were a number of patients who actually had additional respiratory virus infections, and we actually subtracted this group from our subsequent investigations. So what we did is we reanalyzed the Mason data following realignment to the human reference genome and the SARS-CoV-2 genome. And this was performed on the NASA High Performance Compute Cluster, and Amanda Severia was the lead in that program. What we did is we then looked at the expression of the various SARS-CoV-2 genes in these samples, and then also analyzed host transcriptome responses using LEMA to determine whether or not there was a similarity in the pattern of gene expression in these PCR negative but sequencing positive samples. So first of all, just looking at the expression and distribution of reads in these samples, what we see is we see reasonable coverage of most of the SARS-CoV-2 genes. So we can see here, this is a schematic of what the SARS-CoV-2 genome looks like. There are various different size genes. This is the spike protein. And what we can see is there's reasonably consistent alignment of reads to various different genes. So this is total counts, and it's on a logarithmic scale, just so it would actually squish onto the diagram together. And one thing that you have to remember is that this doesn't take into account the depth of sequencing. Some samples would have more reads than others, and other samples, other genes, obviously they're larger than others, so you would typically see slight variation in the number of genes we'd expect to see aligned. But we see reasonably good coverage across many of the different genes of the SARS-CoV-2 genome. And so our conclusion from this would be that actually this would suggest it's not just a mere sequencing artifact where we're just having a single read or amplicon that seems to just be been incorrectly aligned to the SARS-CoV-2 genome. So then what we did is we used differential gene expression analysis using LEMA. We used an R package implementation of it to look at the expression of various different genes in patient nasal swab samples subjected to various loads of the virus. And what we see is actually this strange biphasic effect. So in the high viral load patients, we see predominantly increased gene expression. This reduces in the medium load, and now we start seeing an increase in repression of gene expression. And then in the low viral titer groups, these have more predominantly downregulation of gene expression versus upregulation. We can perform gene ontology network analysis of these data. So this is using the TopGo package. What we see is that there are various clusters of associated functions that are identified, especially those associated with smell, chromatin remodeling, and metabolism. Interestingly, in the medium viral dosing patients, we also see an additional cluster that really seems to be associated with repression of gene expression and RNA silencing. Then in the low viral load population, we still see this cluster here associated with sensory and perception of smell and pathways associated with chemical sensing. But then we have a few others associated with clustering with biological process of metabolism. And what's interesting is that we don't see a huge overlap really in terms of the absolute numbers of genes differentially expressed in these different groups, maybe with the exception of the medium group. But when we look at the gene ontology, we do see that the majority, so these in the top 30 terms, the majority of terms actually are common in all three subsets. For those who are interested, the other respiratory virus data group also shows a predominantly large increased gene expression. We actually look at the actual gene expression profiles compared to the COVID infected patients. We see it's approximately shared. So 50% of the gene expression pattern from COVID is similar to that of viral, the other respiratory virus infections, and 50% is unique to COVID, and same for the virus. Again, similar pathways though in terms of the virus infection compared to COVID, especially the high. So then we look at these PCR negative sequencing positive samples. When we look at differential gene expression versus controls, what we see is a pattern that looks very much like the low viral gene load patients. That is, it's predominantly a downregulation, a decreased gene expression pattern. We see this either using the volcano plot or Lema's MA plots. If we then look at differential gene expression between this low viral load and the PCR negative sequencing positive samples, we see basically only one gene is differentially expressed. And so this really suggests it's like the least difference group in this data set. But then what happens when we look at the biological processes? At the top, this is the medium data set, and this at the bottom is the negative PCR samples. And so what it actually looks like is it's very similar in structure to that of the medium viral load patient data sets. And actually when we perform Venn clustering of the Go terms, what we actually see is that there's more similarity between the negative and the medium than there is between the negative and low dose patients, low viral load patients, COVID load patients. We've also done this using a more sophisticated pathway analysis using GSEA. This is actually Ashen's favorite tool. And so again, though, what we see is we see greater similarity in actually the medium dose COVID patients to this PCR negative patients than to the low dose COVID affected patients. And we also see this as well in the curated pathways as well. So the curated pathways is a slightly different data set, and it looks at actually pathways associated with diseases and disorders, as well as some KEGG data sets. So taking this all together, we can conclude that PCR assays, especially those based on a single gene, may not accurately reflect an individual SARS-CoV-2 status. We actually have had some interesting data since we performed this analysis. We were doing some validation studies for a testing lab, and actually we received samples from a lab that used a single gene for their PCR testing. And lo and behold, we actually found 10% of them, which were deemed negative samples, we actually found using a 3-gene testing protocol actually came out as positive, which actually would suggest that maybe this is a common issue, and especially when we're using PCR tests that rely on a single gene rather than a panel of genes. What's interesting though is that the transcriptome responses therefore could be used to identify severity of infection. Actually, it's suggestive that there's maybe unique profiles of gene expression in patients depending on what different pathogens actually are affecting the patient. So just finally, I really want to acknowledge the assistance and the collaborative nature of the members of the Covert Consortium, especially Chris Mason, who very early on, before data was even published, was willing to share their sequencing data with all Covert members, and has worked very hard with us, and his team as well have also been very responsive when we've asked them questions about the data. Amanda, I've already mentioned, who is the lead of the realignment at NASA. Jonathan, Afshin, and Deanne, who've been wonderful in helping with working out this analysis. Fritz helped with some of the pipeline work, Eve, Francisco, and basically members of the pipeline microRNA communities as well. And finally, I really want to acknowledge all members of Covert for creating this wonderful organization that really gave me something better to do than trying to source toilet paper when the SARS pandemic first started. So it was a wonderful distraction, and I'm glad to have played a small part in it. Glad to take any questions.
Thanks, Rob. Yeah, good. I think that was all of us in Covert. We wanted to do something instead of going crazy. But anyway, yeah. So I think you do one question. You're even under time, so this is good. So let me ask a more generalized question, and I don't know if you could type in the rest of the answers. But the one question maybe more generalized is what's what is it? Oh, it just went away. Hey, here we go. I got upvoted. What lessons learned here should we apply to the next pandemic?
I think one of the key things for me is not to rely on a single gene for doing anything, especially diagnosing a disease. It seems like multi-panel PCR really is the best approach, though it should be noted that even that's not infallible. This has especially been noted with the Thermo Fisher TAPMAN assay, because they've actually discovered that the S gene probes that they're using for one of the 3 genes for that actually seems to drop out when you have the variants that were originally described in the UK. So we just have to be careful about understanding the limitations of any diagnostic approach that we use. I think also we should maybe not always say that PCR is the gold standard, and maybe sequencing is the gold standard, but I might be biased in that opinion since I like sequencing.
Sounds good. Yeah, a good answer. So that's one thing Covert wants to do for the audience is prepare for the next pandemic, because there probably will be one, unfortunately. But okay, so Rob, I don't know if you could handle the typing in the rest of the questions. Of course. Yeah, okay. And then I guess Todd, I'll give it for introducing Keynote. Thanks for everything for the session. Thanks, Todd.
Yep, sure thing. And I'm just on stage right now just to introduce Dr. Angela Wilkins, the Executive Director of the Ken Kennedy Institute at Rice University. Angela, thanks for chairing the next session, and please take it away.
Thank you so much for having me. Our next keynote will be discussing the Decode Effort Initiative at MD Anderson. David and Andy, if you could pop up. Along with founding and leading the Decode Initiative, Dr. Andy Futrell is the chair of the Department of Genomics Medicine at MD Anderson, and he focuses on the translation of genomic techniques into therapeutic applications. And then we also have Dr. David Joffrey. He's the first ever Chief Technology and Digital Officer at MD Anderson. David oversees MD Anderson's data governance and integration across the organization's campus, and also he's a professor who still practices science when he has time. Thank you guys both so much for being here, and I'm looking forward to hearing more.
Our pleasure.
Pleasure to be here. I'll bring the screen up here in a second. Tell me if you can see it. Can you see the screen?
Mm-hmm.
Okay, Andy, take it away.
So what we're going to go through is a bit of a tag team approach here to give you an update about where we've gotten to with an effort that started almost a year to the day ago. And it was born out of this idea that there was a wave of viral infection coming into a highly immunocompromised set of patients that we look after on a daily basis, and what do we need to be thinking about to prepare ourselves and also, you know, try to get out ahead and provide some useful information to those elsewhere who could potentially improve their ability to manage patients who both had cancer with a COVID infection. Can I have the next slide, please? And the original sort of alarm bells, if you will, began to go off fairly early in the process. What you see here is original data from China around sort of Valentine's Day in February, where it showed a high mortality in patients who had, again, a diagnosis of cancer and a COVID infection. And this began to get us thinking that, well, if the numbers looked like they were going to look, we had to begin to prepare to at least understand how we could implement a research framework so we could learn as fast as possible from infected patients as they began to emerge. Now the next slide, please. And this began to sort of meet itself out over several additional publications, both from China and data from Memorial Sloan Kettering and other places, which again sort of suggested that this disease was playing out differently in cancer patient populations. And what you see here is a Kaplan-Meier curve showing the sort of the 14-day mortality if the anti-tumor treatment was within 14 days or not within 14 days, even the sort of sequelae of severe events relative to COVID infection seemed to be different. And so not only was the sort of the absolute presence or absence of a cancer diagnosis or an active cancer in a patient, but also the treatment and the timescales in which that treatment was being given. And that began to formulate this idea that, these were things we had to understand because some of these things are modulatable. When you give treatment, if you have to delay treatment, what agents you might use or how you monitor on the backside become things where you can intervene and potentially bend curves on the outcomes. And so this was the grist to the mill, if you will, that began to let us form the decode protocol. The next slide. And so what we decided to do in really early March or mid-March probably was think about what we needed to put into place. And we immediately gravitated towards that sort of coin of the realm, which is an IRB protocol that would allow us to do research. And the protocol was written as broadly as we could possibly write it with really quite intense involvement with our IRB folks at the time. And so it was written sort of in tandem, developed in tandem to let them know this is what we're thinking about doing. And the goals of it were pretty straightforward: to source, aggregate, and integrate and analyze all data. And the all is important here, from patients who are tested, symptomatic, or at risk for COVID-19, because we want to understand potential prognostic, predictive in the context of drugs that might be used, and outcome correlates that will empower better management of patients who have, again, a cancer and a COVID coexisting sort of set of diagnoses. And so the approach was an IRB approved protocol. It was a waiver of consent based protocol, which, again, was an extensive discussion with our IRB folks and the compliance folks. Was this an appropriate approach to take? And eventually aggregating all of that data into an institutional data lake architecture, which David will describe in a few slides. But it sort of brings together 4 quadrants of activity. So one is patient care data, which is the data that lives in the medical record, the EPIC, and also external treatment data that's brought in by the patient when they arrive. It also involves, non-standard care data collection, thinking about sort of biosensors, wearables, and in our case, really extensive exploration of the patient-reported outcome space, both in the context of COVID, but also now moving into the context of vaccination, it has to be said. External data, we weren't the only ones who were alarmed or, and began to spin up things to not only make ourselves not go crazy, but also begin to sort of think about global aggregation of data in this space. So the ability to bring in external data was explicitly called out in the protocol. And also we tried to think just a little bit ahead of the bend of the curve, and been having conversations with our colleagues in Memorial Sloan Kettering who had been literally hammered by the first wave, and thinking about things that they weren't able to set up in time. What could we do differently? And one was tissue collection. And so how do we begin to modify the protocols that we had to begin to collect tissues on patients in fairly challenging environments? So certainly at the beginning, where they were in complete isolation, there was pass-throughs which had to be designed from the clinical side of the house to the research side of the house. We had a complete moat around the hospital, so no one crossed the barrier. And so these were all logistical things, but to get the protocol basis was incredibly important. So we modified longitudinal sample collection protocols that we already had to move into this space as well. So this was sort of the paradigm shift that we had, both around leverage everything you can, but also instill this team data science and this team approach, which, as you can see from the sort of the circle in the middle, requires all these pieces to work together and actually learn on the fly how to integrate them. So one of the silver linings, if one can describe a global pandemic as having one, was the ability to advance this culture of data science driven off the horrific situation of a global pandemic. And I like to refer to it as a bit of a crucible that allowed us to do things faster in ways that, you know, even shocked our institutional colleagues. I mean, the DCODE protocol was written in a week and approved in a day. You can say those numbers and name things in front of any MD Anderson clinician, and they will sort of, their eyes will bug out of their head because that's unheard of. But it shows, you know, when the chips are down, so to speak, we can move quite quickly. But also, I think showing them an aggregated view and an organized view of what we were intending to do early on in the process helped grease the wheels of that. But it has changed the way we think about aggregating data. It's changed the way we think about interacting. So it has outcomes which I think will yield benefits for cancer patients writ large, in our institution anyway, for years to come. Now the next slide, please. And so we keep track of the kinds of data you might think, and this is sort of the global view of our positive patients on a daily versus cumulative basis. And you can see clear evidence of the two waves, sort of in the June timeframe, and again in the sort of the late 2020, early 2021 timeframe, and how we've all sort of reverted back to essentially midsummer numbers, late summer numbers for MD Anderson. Cumulative, I can't read these numbers, but my guess is it's over 3,000 patients at this point that we've seen with a COVID and cancer diagnosis. So we're beginning to get into numbers where one can think about doing, you know, not only sort of observational, looking for the, you know, the large effect size signal, but actually some fairly detailed investigations, which we'll hit on a couple of those in sort of overview form as we move through the presentation. Here's the next slide, please. What's shown here is sort of a living diagram, which is ever expanding of the disciplines in terms of, you know, clinical specialty or research specialty, area of the hospital, silo of activity. We're actually working on the problem of COVID and cancer from various perspectives, but also more importantly on a common data set generated from those 3,000 patients. And it ranges from everything in sort of medical oncology with various specialties, derm, interventional radiology, endoscopy, a large presence of surgical oncology. We've got nursing studies going on, pharmacy, supportive care. I mean, it is really, you know, we get used to looking at this, but this is an incredibly broad array of individuals working on a single problem. I personally, and I've been at Anderson 9+ years now, can't think of another topic that has this broad a buy-in in terms of research interest as this has been able to generate. And of course, what this does, this not only gets people interested, but what it also does is gets people knowing who everyone else is, which in a place the size of MD Anderson actually has huge value, because you get this cross-fertilization sort of environment where people begin to think, well, I can work with people in benign hematology if I'm a neurologist, and that becomes a new axis of activity, which again, I think can only sort of empower our future research in cancer as we move forward. Now the next slide, please. And what this shows is just as you look and do a word cloudy kind of thing in the various sort of protocols and the various sub-protocols that were written, sort of what comes up, and it gravitates around the central idea about what does this mean to an individual patient. And we'll show you a few examples of the various angles and tactics and sort of vectors that can take, but it is remarkably consistent that, you know, it has been very much trying to understand, how do we understand the play of this disease in our patients from various angles to try to improve or militate against bad outcomes in the context of you know, a fairly serious disease anyway. Now the next slide, please. And again, this is sort of those I think we're north of 60 or 70 different projects now, but they're sort of laid out from left to right in sort of age of maturity or evolution of maturity. We have ones which are pre-engagement, where we sort of sit down and we say, okay, what is the study you'd like to do? What is the question you're trying to ask? Try to get a feel for, you know, the data, large data type areas that might be involved. Do we already have them? How fast could it go? Do we need to go think about new things? So we have two that are just come online from melanoma surgery and a nursing study as well. We have active engagement, which is beginning to sort of dig in and say, okay, let's see what it's going to take to get this thing done. And there's certainly those which are now in full flight, where we're actively engaging and pursuing the research space. And again, everything from imaging physics to leukemia to intensive care and plastic surgery are in this sort of bucket at the moment. And then we have the support phase, where there's been a maturation of the interaction of sort of the data science team, if you will, and the investigators within the clinical or support departments. And there's champions have been identified, and they're kind of self-actuating at this point. I mean, they're running, they've learned how to use the platforms we have. There's still a lot of interaction, but they are pretty much moving the research along on their own, or first approximation of their own, to the point of actually publications emerging, presentations at ASCO and other in meetings as we move along. So the outputs are beginning to emerge, and that's quite gratifying. And they're important because they are informing us and shedding light on the things we need to think about in terms of cancer patient management. The next slide, I think, is yours, David.
Thanks, Andy. As Andy highlighted, one of the things that's happened over the past year is Decode has been the crucible, as Andy describes it, and really brought culture, process, and technology together. You think about lighting up 70 projects across an institution. You could imagine that if we didn't think about organizing this a bit at the beginning, every one of those projects would have been tapping the same person on the shoulder and asking for the data. And we can imagine there'd be quite a divergence. So one of the things we really realized that emerged out of this activity is that we have these research questions and we want to think about a common set of data, these over 3,000 patients now we want to draw on and learn from and use data science methods. We really need to sort of come up with a team data science paradigm. And this really emerged because in this context, everybody was poring over the same data, and improvements on that data would benefit others. And so a real teaming paradigm, and the work that was assembled to get the data to flow by whether it's the IT teams, the researchers, the curation teams, the analytics groups was quite remarkable. And early on, it was recognized that a lot of people were putting their energy into this, but it wasn't necessarily going to affect their research project themselves, but it would be very much more a team sport. And so early on, back in May, we said, if we're going to work like this together, what is the team's principles? And so we drafted these 4 principles, and they've actually survived quite nicely, and they've started to inform the way we work. We use them to describe them to people who are joining the team to participate. And it also helps the technology teams put the right kinds of tools in place. And the first one is that the data that we want to collect, we want to really make sure we capture all the metadata on it. So that's the observations are in context. So we can really have a good understanding of where that data came from. And having, in the context of COVID, we realized right away, what kind of test was this? Do we trust this test? Is this a reliable test? So really early on, the questions were, where are these observations coming from? Can we trust them? And then that whole ethos really baked in, because we realized if we can keep track of that all the way through, we can decide what to include or not to include in subsequent analysis. And that basically led to sort of second principle, which lets us contemplate the question of quality. Just because we have something in front of us doesn't mean we can trust it. But what else around it could we use to develop some confidence around that measurement? And that's this concept of using the dynamic assessment of quality if we have the data in context. Of course, realizing that during the pandemic, new kinds of information, vaccination status, for example, is now coming on. How do we know that? Did it come out of the record? Is it a pre-recorded note from another clinician, or did we capture it directly within the EPIC system? And then if we start to do that, we can start to say, well, the conclusions in these papers, which we want to publish and are publishing, they can be proven to be robust because we can tie them back to the actual observation. So link the insights back to the observation makes it a powerful fabric. And it's really remarkable to see the connection now in the conversations around when you drew that conclusion, what data did it come from? And finally, from that team perspective, understanding who participated, who helped collect this. And in fact, one of the things that came out of the initiative was to recognize the Decode team as a collective author on the publications that come out of the work that's come through this activity. So really changing the way the team gets together and being together Monday, Wednesdays, and Fridays for an hour from 7 to 8 for a year now has been remarkable, as Andy suggested, in driving this team forward. So the principles have been very helpful. What the principles helped us do is kind of think about the way we think about the digital architecture at MD Anderson. And I'm not going to be able to do justice to the scale of detail in this, or we're going to be able to do justice to the scale of detail in the projects. But I'll just give you a bit of a hint. We drafted an architecture called the context engine. It's called the context engine because we want to keep the data in context, just like the principles describe. We worked with some industry partners to help build this, but it's produced a really consistent way of the teams thinking about data and keeping it in context so that we can leverage it as much as possible. And so this was a bit of a rallying call for the IT teams to come together as we rapidly assembled a number of different components early on in the project to let the data start to flow, because we wanted everything from vaccination status to test result all the way to ventilators and anesthesia machines because of the potential value of that information for the work. And I'm not going to go into technical details, but give you a bit of a feel for all the different components. Our entire EPIC framework was drawn in terms of its data, all the data in our healthcare data model, which goes way back in the late 40s in terms of data, and our Hadoop data lake, which has very complex and diverse data sets, are all now drawn into this common framework, and you could tap into it through a common analytics engine that we've provided and recently adopted within the organization. And that interface was supported through something called the data office, and I'll get to that in a second. And it's been very powerful for convening these conversations across these over 70 projects. So building the context engine is a technologically interesting conversation unto its own. Running the context engine, that is, how do we kind of make sure these principles are supported, has been another development. And we've actually built something called a Data Governance and Provenance Office that stood it up in a year, led by 2 individuals shaping this. And within that, we have an ontology team that thinks about how things are named, a quality team that looks at how the data quality is maintained, but also a compliance team to make sure we're satisfying all the rules that we have to follow in healthcare, while we're working as fast as possible to try to extract insights that are important to our patients as they deal with COVID. And so connecting researchers to data sources, from data sources on the right all the way to a data office on the left through a framework. And this framework will persist. As Andy pointed out, it's been something we've put up to realize that we need to learn fast and have high quality data, and that needs to be managed. It's illustrated here in another graphic. This is just a visualization, but the point being that the rich data sets we have on the right in the context of COVID and cancer are important, but for us moving forward in the context of cancer, and the consumption of data on the left, you can see there the Decode data office. That's the data office where all those projects went and said, hey, I have a question, I have a research question. Does it fit within the Decode protocol? What data is available? And that conversation says, well, this is what we've got so far. And making sure that that data group is fed by the data sources on the right in a structure that's managed, but also technologically supported, is what the so-called data management system we've now stood up and reinforcing moving forward. And it does sort of links the architecture and the technology back to a process for governance. And this has turned out to be extremely helpful for us. And we're now looking to scale this across the organization to more than just COVID-19 and cancer, but cancer in general. So I'm going to pass it back to Andy. We're just going to go through a couple examples. We have like 4, I think, projects, 2 kind of more in the bio space, 2 more in the imaging space, or 3 in the bio space and 2 in the imaging space. Andy's going to take the first 3 of them here, and then I'll take the next 2. Andy?
Yeah, I mean, again, just to give you a flavor, I think, of what kind of data and the projects and the questions that were getting asked by folks, you know, who had never actually probably even been in the same room with each other before. And the first one is a project from benign hematology, led by Christian Rojas Hernandez and the Decode team on the issue of, you know, characteristics and clinical outcomes of coagulopathy in patients, again, with a cancer diagnosis and the COVID-19 positivity. Because one of the early observations that emerged, again, from the world experience was this apparent increased incidence of diffuse disseminated intravascular coagulation in patients who have COVID-19 illness when they're admitted at MD Anderson. So how do we begin to investigate that? And, you know, it was assembling data that, you know, we don't normally go worry about, which is sort of the benign heme data, which is, you know, fairly routinely collected on cancer patients, but it wasn't something that had entered into the realm of what we actually worried about yet. So there was a understanding phase that had to go on with, you know, okay, where do these data live? What do they mean? Where's the best source to gather these in terms of, you know, the thrombotic and hemorrhagic complications? What are the strategies that are being deployed in these patients? What are the outcomes? And, you know, what are the impacts when you begin to measure these things? Can you begin to think about, you know, risk markers for those patients who will end up with, with problems in terms of their intravascular coagulopathies when these 2 sort of disparate disease states converge in a single patient? So this is, again, an ongoing project that has been run over a period of months, going on for probably 8 months. I think this was one of the early ones, but it was, again, born out of the clinical observation that we were seeing in certain patients, and had been reported in the literature, really serious sort of coagulation in the peripheral circulation. How do we begin to understand this in terms of our cancer patients? Now the next slide. And sort of following along the same sort of cardiovascular line, another project, again, which was led by Nick Palaskis and Anita DeWall and the team, was asking very specific questions about our patient population as to patients with cancer and pre-existing cardiovascular disease, which isn't such an uncommon phenomenon in cancer patient populations. If you understand the biggest risk for cancer is an elderly age, and that goes along with other comorbidities, which frequently include cardiovascular disease, then it's not an uncommon phenomenon to have cancer in a pre-existing cardiovascular disease. But what is the issue of complications in terms of a COVID diagnosis? Do they have a higher incidence of myocardial injury? A very specific question. Do they have increased cardiovascular events compared to those patients, COVID patients without cancer? And again, Nick became an absolute master of the platform. He's a cardiologist, but he had the fire, and he became an absolute master on the platform. And he sort of drove this project to really begin to explore this space again in an everyday management problem that exists in our patients anyway. But now, what wrinkles does COVID throw in it that we have to be really keenly aware of if indeed the risks of any of these events is severely increased or significantly increased? Can I have the next slide, please? And sort of again, moving sort of on another tangent, it was this notional idea of, since we adopted relatively early on in the process universal asymptomatic testing for any patient going to surgery, what is the number of patients and summarize their testing results? That is an early dip into the water of what the disease was actually doing in our population, almost from a hospital epidemiological standpoint, if you will. What is that preoperative testing rate in our cancer patient population, sampling those patients going to surgery? How does that compare to the local testing rate, which is beginning to emerge at the same time? What do we know about the community versus the community of cancer patients, and specifically those going to surgery? Can you ask questions about short-term outcomes for patients who underwent surgery relative to those patients who underwent surgery without COVID? And how the practice and the outcomes in surgical oncology, how they're affected. And one of the early questions was, everything shut down essentially in the hospital, and there was inevitable delays due to a host of factors one can imagine: the inability of patients to travel, the reticence to be, in a group, all these things. If you can recall the state we were in back in March, April, May, June of last year, what were delays in treatment going to do to outcomes? In terms of potential disease progression, in terms of even the extreme example of taking what was an operative condition in May becomes an inoperable condition by June because of the tumor growth. These were all questions that begin to be really dug into to try to understand the impact. And if you can frame the impact, then you can begin to sort of think about the risk you're willing to undergo to sort of intervene if the impacts are actually substantially negative. So I think it's a framework for understanding, as was said in the prior talk and after, what happens in the next pandemic in terms of understanding how we manage and triage and understand the treatment of our cancer patients. The next slide, I think, goes back to David on the imaging.
It goes back to me, Andy. One of the things I wanted just to add is that, it wasn't like we had an n equals calculation at the start of this. We just knew we were going into something. And I think one of the most interesting things was that as the ends, after the first peak, everybody was like, wow, we built this whole infrastructure, but we're not going to have enough data to learn from it necessarily. But then it kept going, and the questions change dynamically as the end goes. And this feedback between the research teams and the data is really dynamic. And I've learned a lot from that process as well.
I mean, it's a fair point. I mean, there was this time, have we built all this for quote unquote nothing? And all of us were quite hoping, to be honest, that we had. Other than the things that David showed prior. But it turns out, you have a wave and the numbers increase, you have a wave and numbers increase, and now you have the advent of vaccination on this now. So now the questions begin to shift and turn over time, but some of these are still fundamental in terms of delay of care and outcomes from that delay, or different types of care. And then the immunotherapy question there, I think, is huge as well, which we're just beginning to sort of dig into.
Yeah, it's very interesting, dynamic, agile year. This effort was very interesting. The teams at MD Anderson are very interested in segmenting lungs, obviously, because it helps us in our design of radiotherapy treatments and other assessments. And so the team, this is headed up by Christy Brock, she's a physicist. She very quickly realized there's an opportunity to use some of those tools to help us auto-segment lungs within cancer, COVID patients. And also, could we do it in lung cancer patients with COVID? And so this is just sort of some work on using some machine learning based approaches that she had developed, and realizing that they don't necessarily port across identically, because the presence of COVID substantially changes the texture within the lung. Of course, you can see it quite visibly in the middle example of a heavy COVID patient with a lot of lung congestion visible in those images, and that it changed the performance of that activity. So some pretty straightforward work that would then position us to better be able to classify based on CT imaging. Another example was some work done by a new recruit that joined us in the middle of the year, actually. He just started, I think, in May last year, and he landed in the decode space, which I think was fantastic because it gave him a place to get going, Jia Wu. And he has a paradigm where he looks at habitat in lung cancer. And he said, could we use these tools to start to classify different subregions within the lung and adapt this sort of habitat imaging concept to a COVID-19 problem in terms of tissue classification. So pulling the data and the teams together and the expertise together, here we have an imaging physicist, a radiation oncologist, and a medical oncologist who focuses on immunotherapy, all working together based on the data sets coming from this platform. So I'll just quickly summarize. I mean, as highlighted in Illustrated, Decode was built to accelerate the pace of discovery and understanding the interplay of COVID-19 and cancer. And it was really built in 10 days and lit up. And then, as Andy highlighted, it drove together a very strong community of like-minded individuals to answer the important research questions. And it really accelerate the mission through strengthening the coordination of data management capabilities we're seeing now. So that it not only was a really exciting time, but in many ways it sort of creates this exciting future for us because we've learned to work differently together under this team data science approach. And that framework we see moving forward, all those projects came to the data office team, and they've really built a dialogue between the researchers and the back-end data sources, which I think has been fantastic. And there's a wealth of research projects, and these are turning into grant proposals as we try to respond to NIH opportunities and others. And the teams have really been strengthened. So really nice to just share the experience with you guys and give an example of how the pandemic has changed things. Not only our understanding of the impact of COVID on cancer, of course, but I mean, it's changed the way we're working within the organization. Andy, do you want to add any other closing comment?
No, I mean, I think you hit upon the highlights. I mean, again, if one is allowed to claim a silver lining in this kind of situation, I think it has been a shift in our thinking about how we can work together, and the almost sort of like the art of the possible, to sort of beat on that overused term. And even now we're seeing things emerge. I mean, just to give you a quick example, I mean, the idea of nodal flare post-vaccination in patients who are going for progression scans, that's an issue now. Yeah. That issue we never had that issue before, but now it is a very real issue. So there's constant sort of things emerging, and the framework to handle them now, I think, is what everyone has confidence in. I think, the ability to now apply that more broadly in the cancer context, sans COVID, hopefully, at some point, will be quite interesting and hopefully useful for our patients.
Happy to take questions. Angela, I presume you're going to chair the session.
Yeah, I'm back. So one of the questions, you mentioned publications and scientific presentations are beginning to emerge from your group. Is there a website or place that we can see all the research outcomes?
We don't have them on a website at this point in time. PubMed is starting to show up there. It takes a little bit of time, of course. And I know there's been a number of submissions to ASCO. Send us a note. I mean, it's probably a good idea to create more of an external.
We should do that. Yeah, that's a good idea.
One of the things that you guys are coming up with also is all the best practices around this, which aren't necessarily the most exciting paper to put in PubMed, but it really seems important. I mean, you guys are constantly talking to lots of people in the community, but Are you going to put out some white papers talking about like how we should do team data science?
Yeah, there's some works in progress. Actually, we have drafted them, and I think we're looking for the right place. It's kind of nice to show some results when you put those out.
Yeah.
So it is a works in progress, but yeah, I think we're doing some very interesting work here. I think, and even the ontology team and the quality team and the way the data office works, there's some learnings to be shared for sure. We will share those.
Your context engine that we were building with Microsoft and Epic, I mean, the thought is that this is going to be a system that a lot of people could potentially be using the next decade.
Yeah, that platform right now, we've drawn together a number of different components, and we have some industry partners who are really helping out a lot on that front. We have had great conversations with Microsoft and others around taking the principles and making sure the technology can support the principles. And I think there's a lot of opportunity there. And so we'll continue to describe it as it evolves. If anybody's interested in the ideas or the concepts, I'm happy to chat with them about it.
We received a question about, let me see, have other infectious diseases shown an association with cancer? And I think, the answer is going to be yes, but I think one of the things that made me think about this, I mean, we have, a case where about 100 million people in the world have COVID now. Are you guys thinking about how you're going to examine what, I don't know, the repercussions from this many people having this disease?
I'll start with a simple view. I mean, vaccinations and cancer are obviously related. I'll let Andy talk about HPV and things like that. But one of the things that we've really realized is that we need to understand the vaccination history for COVID patients for sure. And this has become a big topic because it was kind of like, you're vaccinated, yes, but it wasn't really relevant potentially to cancer care. And now that we have so many patients who've been affected by COVID, but then also those who've been vaccinated. And as Andy pointed out, some of these patients, when they get vaccinated, they get a nodal response that can be mistaken as an issue in terms of recurrence for cancer. They're being monitored. So all this starts to play into the equation. So one of the things we really realized is we need to really upgrade our tracking of the vaccination status and previous exposure status of patients. And whether it's patients who have had no symptoms from it at all, or some of these kind of long haulers who are experiencing longer response time. So from a process perspective, Andy, I don't know if you want to talk about vaccination in general and cancer.
Yeah, I think there's a group of us, and it's not a small group, who think for the foreseeable future, have you had COVID, have you been vaccinated, will be a first question, or one of the first questions that get asked, because we have to begin to put, particularly immune response, in the context of a cancer diagnosis with agents that are designed to tweak T-cell responses, by and large cell-based therapies, checkpoint blockade inhibitors. What is the nature of, to be able to mount a proper tumor response in the context of what your viral response looked like to the vaccination? Are these two things related? Are there things we can learn about sort of overall immune tone, if you will, in cancer patient populations with different diseases, or with prior histories of treatments that are myeloablative, for instance, that sort of whack the bone marrow that never quite came back, and now you've got reduced T-cell repertoires, you've got different immune tones. So I think all these things are on the table as points of, I think, true concern for particularly the medical oncology community, where we probably give more checkpoint blockade than any other place in the United States, and we have to begin to understand what the interplay of a prior COVID exposure or not, prior vaccination or not, play in sort of the choice of agent and the efficacy of those particular agents. Now, the two may fly completely different paths, but we don't know that. We have to understand it, I think.
Yeah, it's a whole new real world data measure that has to be tracked now, and there's a lot of variability out there, and even the time space in between two injections. I mean, it's a ton of variability. So, yeah, great question. Whoever put that one up there.
Yeah. And I just wanted to jump in to say thank you to you both, David and Andy, for wonderful. I learned a lot here, and what an amazing initiative. And all, of course, we know at Covert, also standing up a lot in a short time, but definitely learned a lot. Angela, I think we fortunately have to move on, but thanks so much for chairing the session. And thanks so much. We're going to transition to our last lightning talk session here. So I'll hand it right over to Eve. So Eve, take it away.
Okay. Oops. Shoot. Okay, so I'll be talking about orphan genes in COVID, and
Sorry,
yeah.
you might want to make it into a presenter
Thank
mode.
you. Yes.
Yeah, perfect. Thank you. Thank you.
Orphans are products of what we call disruptive gene evolution. So genes can evolve gradually by duplication and modification. And they also can evolve disruptively by formation of novel protein coding sequence. And orphans, is somebody moving that little, somebody's moving that little spotlight thing, and it's not me. I'm not touching anything. So ignore it. So orphans are genes that have protein coding sequences with amino acids that are not similar to that of any other species. So they're unique to a species. And there's about 8.7 billion extant, assuming a really conservative figure of 1,000 orphan genes per eukaryotic species, and what's thought to be about 8.7 million species on Earth of eukaryotes. Yeah, okay. So the characteristics, there's a lot of characteristics that are different. And if you look here, this is a graph of genes of Arabidopsis, and they're separated according to gene age or phylostrata. And so the species-specific ones, the orphans, which are at the top, if we look at their structure, the predicted protein structure, it's almost exclusively undefined. And as you go to older and older, more ancient genes, you find that eukaryotic and prokaryotic genes have a much higher amount of defined structure. And there's other characteristics as well. And so these undefined structure, which are disordered structure, tend to be sticky proteins. And so they interact with other proteins externally and internally. And they're critical to speciation. And so they're a key part of ecological warfare. There's tens of thousands of stinging toxins in jellyfish, sea anemones, wasps. And these are, each species tends to have its own cadre of orphan genes that encode these stinging toxins. And also there's pathogen-resistant genes that interact externally in Arabidopsis and humans and other species. They respond to abiotic challenges, antifreeze proteins, self-incompatibility proteins, and they integrate into existing internal networks involved in metabolic regulation and disease. So one such is the QQS protein from Arabidopsis. And that protein is it's an exemplar of a protein that can function in a different species. So they're unique to individual species, but because they're sticky and they interact with existing proteins, can be introduced into other species. So in this case, soybean, Arabidopsis to soybean. And if you just look at the black lines, those are controls, whoops. And you can see that the transgenic soybean have more resistance to aphids, more resistance to bean mottle virus, more resistance to cis nematode, and quite a lot more resistance to bacterial sudden death syndrome. So this complicated slide, but I just want to show you the middle of it, really. So there's a tremendous amount of transcribed sequence that we don't know what it is. It's not annotated. Some of it's probably noise, but some of it's expressed to high levels. And of the orphan genes, they're hard to recognize. And we believe that there's quite a number of orphan genes that are still within this dark transcriptome. They have not yet been identified. And we call these unannotated orphan transcripts evidence-based orphans. So there's a bunch of orphans, some are evidence-based, but they haven't been annotated, and some are annotated in the human genome. I want to show you that there are population-specific orphan genes, and we only have this right now for Zea mays. If you look at this slide, basically, this is a whole lot of Rna-Seq samples. This is the level of expression. We've filtered out all the low expression evidence-based orphans. And you can see that there's a lot of very highly expressed orphans over many, many conditions. And also, this is a specific line of ZMAs. You can see that there's a smaller number, but a significant number of very highly expressed orphan genes that are unique to a particular line. We've seen this for 25 different lines of maize. So there are population-specific lines. So we have a great Covert team here, and the inspiration and the experimentation of all these people has really helped to drive all this work. So our questions are, we want to know what are the baseline differences in orphan and EB orphan expression across populations? Are COVID-19 relevant processes impacted across populations? Can orphans provide sensitive molecular signatures for COVID-19? And do orphans play a role in COVID-19? So we're just started this about, well, almost a year ago. Our pipeline involves, we're taking from NCBI SRA database, somewhere like 40,000 different samples of COVID exposed and other types of cancer and non-disease tissues. We're putting them through a specially designed pipeline. We are identifying the novel genes and the annotated genes de novo from the raw data. We're inferring the phylostrata, so we know we can identify which ones are orphans and which are not. And then we're subjecting it to exploratory analysis. And so this is with meta-ohm-graph, and we basically can analyze in R, we can analyze in meta-ohm-graph tools, and we can visualize the data. So this large bit of tens of thousands of different RNA-seq samples are then analyzed in meta-ohm-graph. So in our recently published paper, we have at the baseline, this is undiseased data from GTEx, we find that in a wide variety of tissues, the most highly enriched processes in African Americans compared to European Americans are to do with infection and immunity, and particularly this cytokine receptor interaction pathway. So there are differences in pathways. There's also differences in the cell-specific expression of genes. So these are esophagus genes. What we've done is overlaid the data from a Rna-Seq data set by Madison of esophageal single-cell data with the expression data that we have for esophageal cell from GTEx. And you can see that there are certain cell types, particularly dendritic monocytes, epithelial glands, and specifically duct and mucal cells that are very highly expressed or very highly represented in African American individuals compared to European individuals. And the same is true for the lung genes. We notice that there's a certain number of genes that are very highly differently expressed. For example, this one is 41-fold greater expressed in African Americans compared to European Americans in a wide variety of tissues. And we've identified now EB orphans that are differentially expressed in African Americans compared to European Americans. And this particular is from the cancer TCGA data set. And so you can see these are three. You can barely see the expression of the genes in European Americans, but in African Americans, there's quite a lot of expression of both of these, all of these genes here. European Americans, and this is where it's separated out into different organs, and you can see that African Americans of this particular EB gene have a lot more expression than the European Americans. So we think there's a lot of genes that may be getting ignored because they're not expressed highly in European Americans, and they may be very relevant genes to cancers and other things. We also see that novel genes are expressed in a cell-specific manner, and this is testes cells, and so this is the data of Guo et al., and they separated the data according to cell types, which I've marked here, and the overlay is we reprocessed their data to look at the EB genes And so we've clustered individual EB genes here, and you can see that here's one particular EB gene here that is preferential to this peritubular myoid, kind of smooth muffly type of cell type. Here's another that's very highly expressed in the Sertoli cells, and you notice there's 2 clusters of Sertoli cells, and it's only highly expressed in one of the clusters. So it may distinguish between them. And then here are 2 that are highly expressed in spermatids, and again, they're in parts of the spermatids, parts of the sort of a subcluster of the spermatids. So it may further distinguish between cell types of spermatids. And our preliminary data shows that we have upregulation of some genes and in European American populations and upregulations of others in African American populations. And this is from nasal swab data from the Mason group. And I won't discuss this because it's pretty preliminary right now, but we're starting to find this. So I want to mention that any gene expression data is a combination of genetic factors and socio-environmental factors in humans, that would be. And so it's a very complex phenotype. It's a phenotype, and it's reliant on, to dissect really what causes it is reliant on metadata for these, both socio-environmental aspects and genetic aspects. And if we had more metadata, it would be, we as a scientific community, it'd be much easier to start to be able to dissect these things out. So in summary, I've showed you processes, key cell types that are key to COVID infection are differently active in African Americans and European Americans. The EB orphans are differentially expressed between populations and cell specifically expressed, and they seem to be DE expressed in COVID and among populations. And we're ongoing processing of the RNA-seq data, particularly related to COVID as it's deposited, so we can look at this further. And we're cataloging the EB data, the EB genes, and we're releasing all the data, all the metadata, as soon as we are able to do so. And we also have reproducible pipelines, all the codes available on GitHub, and we'll have a new workshop, we hope in the autumn, on using the tools. So that is it. And are there any questions?
Sorry. Yeah, there are a few questions. Sorry, Eva.
Okay. Well, my problem is I lost my Zoom because it went to my slides. I don't know why it's doing that.
Let's.
I don't know why I've completely. Let's see if I. Okay.
I can ask a question while you.
Okay. Yeah.
So there's a question here that have you looked at orphan genes in immunocompromised individuals or patients with long COVID?
I would love to. We need the data, and we would be really interesting. So as soon as we get that data, there's really a limited amount of public COVID data, RNA-seq data available right now. But as soon as it's available, we certainly want to do that. Great question.
And with that, I think I'll just hand it back to you. Okay.
Okay. All right. And then I see another question here. Did you look at these orphan gene? Oh no, that's the same one. How common do you think population-specific evidence-based transcripts are? Well, we still don't know that. I'm going to guess that there's at least hundreds, but we'll have to see. And I'll do one more question. I have another minute.
Sorry to interrupt. I think we should move on because of the sake of time. We're over. So this.
Okay, I mean, all right, I can go on then. I'll answer it some other way and pass it on to Peter Robinson from Jackson Labs.
You need to.
Hi, everybody. So Guy Karlovac from my group is going to be talking. Guy, are you here?
Yeah, we see him. We see him. He's here. He's on mute.
Afshin, were you referring to me now? Because there were like a few minutes where I couldn't really get any input from the Zoom.
Yeah, you're here now. We can hear you.
Okay, so it's my turn.
It's your turn.
Okay. I hope the connection remains stable during the talk. Okay. So I'm going to give a talk about a project that we're doing in the Robinson Lab. We investigate alternative splicing patterns that are specific to beta coronaviruses. Received help with, from a lot of people in Covert. So I would like to mention in advance that we are very grateful for all the input that we received, both in data and in consultation. So what is alternative splicing? What are we looking at and examining here? So we have a gene, and the gene has various products. In this example here, you can see that there is a darker shaded exon in the middle, and it can be spliced in and result in this inclusion isoform, but it can also be spliced out and result in an exclusion isoform. And there is, of course, the transcript continues downstream and upstream of this event, but there will be a different transcript whether this exon is included or excluded. This figure is from a paper where they studied dengue virus and its effect on splicing. And as you can see here on the right side, there's a difference in the abundance of transcript of these two genes, depending on whether the cells are infected or not infected with dengue. And this is something that has been investigated in various viruses. And so we are interested whether we see an important impact on splicing, in particular in coronaviruses, beta coronaviruses. In order to study that, we use high throughput data, both from the Mason Lab and external data sets. Previous knowledge about the effects of SARS-CoV-2 on transcription, translation, and basically all this chain in the host is that it's known that NSP16 interacts with components of the spliceosome, and it suppresses host splicing. There was a recent paper about that. And NSP1 contributes to inhibition of host protein translation. Also NSP8 and 9 are known to interfere with trafficking to the cell membrane of proteins, and have been shown to suppress the interferon response. To follow up and to study splicing in more detail, we developed a model that includes both changes in expression and changes in splicing. So if you look at this cartoon at the top line, there are two samples, sample A and sample B, and the expression is represented by this bar of stacked colors, and it's higher in sample A on the left side. And so we'll have a covariate beta that says expression in one sample is, let's say, twice as high as it is in the other sample, in sample B. And in order to model splicing, we need something that is not just continuous, because splicing, the proportions of isoforms always have to sum to 1. So instead of just adding this beta, which represents a multiplicative factor of changing transcriptional event, we are using the HSON perturbation, which is given on the right. Basically, if PI tag is the proportion of isoform I in cases and PI is in controls, it is mapped using this vector covariate alpha. PI is mapped to PI tag, and we can calculate this mapping using this operation here, which is also illustrated for a vector of 2 transcripts that are exactly half of the total transcription event in cases. And then by using a certain values for the covariate alpha, we map them using the HSON perturbation to different proportions in the other group. So once we fit this model, we get posterior distributions for alpha and beta. And these will tell us whether we observe, whether we believe we observe a differential expression and splicing. So in order to translate these posteriors to probabilities, we need to somehow summarize the posterior distribution. And in order to do that, we use the region of practical equivalence to illustrate using the beta, which is the simpler covariate. If we know that beta is 0 or a very small value around 0, we would say there is no difference, practically no difference in cases and controls. And so we sum the posterior outside this region, and that would be the probability of differential expression. For slicing, it's a little different because, as we said, it's not just addition or a multiplicative factor. So in order to say that there is no difference between cases and controls, we have to say that pi tag is pi. And in order for that to be true, alpha I, which is the ith entry of the covariate alpha vector, has to be equal to this sum. And so in order to say no difference, we have to look around the region that in which alpha I, the entry I of the covariate of splicing, is equal to this sum. And we do exactly the same thing as I described for beta, and we get a probability of differential splicing. We do have to adjust for multiple testing, and so we use a spike and slab prior. And the spike component is concentrated within the region of practical equivalence, and the slab is flat, more or less flat, or has a high variance. And then in order to find the weights, we just compute for all the genes together with the same prior, the posterior mode, and take the weights that correspond to that. And then we just fit the model for each gene individually to get full posterior. And this gives us corrected probabilities. And so the false discovery rate is just the sum of probabilities in a set of isoforms that there is no effect. So this is the methodology for identifying differential splicing. And so we applied it to a set of pathogenic data sets. Some of them are beta coronaviruses, and some of them are other viruses, and even one bacterium. We created a pipeline using SnakeMake. So we start from the SRA numbers, and the pipeline downloads and runs everything through the same processing, and runs RSEM to generate the counts of different isoforms, the expected counts. And then we get a matrix, and we apply the method that I described. As you can see, there is some difference in cell types and number of samples. There are other differences in library size and so on. So we will use methods that somehow can see beyond that and compare the data sets using those methods. The Mason data was processed using a separate pipeline on DNA Nexus. So we use those counts as given. And so the first thing that we looked at is Go enrichment. So we can say for each gene, if it has a differentially spliced isoform, we consider it a differentially spliced gene. And then look at Go enrichment. We used Ontologizer, which is a tool for computing Go enrichment. And as you can see, these are three beta coronavirus data sets for which we looked at various categories. This is a sample of the categories. And the response was very similar. They're all enriched. There is a fold change in the proportion of genes that belong to the category, and it's statistically significant compared to dengue, which has different terms, not the same as the beta coronaviruses. Other things that we looked at, we looked at the proportion of intron retention isoform. So as we said, the virus interferes with host processes. So we expected to see various structural properties changing in transcripts. And if you look at the right, on the top, there is the fraction of retained intron isoforms from the differentially spliced isoforms. And in the beta coronaviruses, this fraction is higher, and it's statistically significant, the difference. As you see, on the bottom, it's the number of exons, the mean number of exons, and there's also a difference. So we attribute that to the interference of the viral proteins with the host splicing machinery. Another interesting thing that we observed is a difference in the number of ribosomal genes that are affected by differential expression and differential splicing. So here we can see splicing on the top and expression on the bottom right of the figure. And we can see that there is a difference in the fraction of the total genes that are affected by these changes. And so this suggests that there is, and it has been also displayed, shown before, that there is an interaction with the ribosome, and therefore more ribosomal genes are affected by both in both respects. So these are things that the virus does to the sort of mechanistically, or with the interfere with the processes that affect transcription in general. But how does it affect the host? So one thing that we looked at, I will show you here, is we took cardiomyocytes that were generated from from induced pluripotent stem cells, and we looked at high viral load and low viral load, and we saw that there is enrichment in the high viral load for various categories that are related to the heart and could be associated with abnormalities. And so we interpret that as the effects that the virus has on the phenotype itself of the host. And not necessarily, could be probably a side effect and not necessarily something that is related to the propagation and transmission of the virus. And so this is an example of one of the genes that was affected by differential splicing. You can see here that there are various isoforms that are expressed in patients. This is from the Mason data. The gene is overexpressed in patients, and the direction of change is green if it's higher in cases, and red if it's higher in controls. And the two isoforms, these two isoforms in the middle that are significant, that have a high probability of being differentially spliced, differ by one domain of double-stranded RNA binding. So one of them has one copy of the domain, and one of them has two copies. And so this could be interpreted as having more binding domains in patients means that either the virus uses ADAR more intensively to promote its proliferation, and it could be an effect of the host. The host is generating more of this isoform that, in order to be able to affect the antiviral response more. So for ADR, both antiviral and proviral effects have been shown. So we don't know exactly what this would cause in COVID, why we see that.
Guy, you need to wrap it up. Sorry.
Okay, sorry. I see that I have 40 seconds left. So I'll skip the summary. I'll just say thank you to many people that contributed. I hope I didn't forget anyone, but we got really great feedback, really great discussions, and data, of course. Nobody, I mean, there was no delay when we asked for advice or for data. Everything worked really quickly, and as you can see, we collaborate with people from many different faraway places. So it was great to be able to do that through Covert. And that's it. Thank you for your time.
Okay, thank you very much. That was great. So we'll move on. We're going to answer questions. So answer questions in the chat, guy, and we'll move on to Deanne at CHOPS.
All right, so let me share my screen and go to presentation mode. You can see my screen, right?
Yeah.
Okay, great. So I'm going to talk a little bit about the inflammation group and update on what we're working on. This is one of the many subgroups that you can see here in Covert. And it's a group of people that are all interested in inflammation as well as in immunology. We also have some cancer people working in the group, as well as myself. I'm a biophysicist / bioinformatician, and I'm the director of bioinformatics at the Children's Hospital of Philadelphia. I'm also a research assistant professor at Perelman School of Medicine. So I'd like to talk a little bit about inflammation pathways involved with COVID, inflammation and immune response pathways. So this is an image from Invivogen. I am not related to Invivogen, but it's a cool image, so I stole it. And we work with this image a lot in the group because it's a nice little mnemonic for the kind of things we're looking at. But so we have kind of a threefold set of pathways or functions we're interested in this group. One is inflammatory response, another is antiviral response, and another is adaptive immune response. You can see they're all coupled together in various ways. There's also additional pathways and responses and functions outside of this that we're not going to talk about today, but will be talked about by other people. But the inflammatory response it has a response to the presence of the virus. And some of them are what we think of as classical pathways for caspases and other responses of the cell. And then we have the complement and the innate antiviral response through the innate immunity response, including double-stranded RNA responses, and then also the reaction of the signaling of the cell to the outside world to the adaptive immune response. So we're interested in basically this whole picture. And we started off, like a lot of people did, with the Mason data. There we go. So as nasal pharyngeal swab data from Weill Cornell from the Chris Mason's lab, he had about 670 at the time, 670 patients, well, not necessarily patients, but subjects. A lot of them appeared in the emergency room sometime last year. And we recently got permission to use death or no death data from them. Unfortunately, we don't have much in the way yet of permitted for use of permitted clinical data for this project because of privacy concerns. But we do have things that we can work with. We can work with RNA-Seq count data, which includes human, viral, and other microorganisms, because since it's total RNA-Seq, we can get a picture of every organism's RNA that's in there, and then use various genome resources to distribute it through a pipeline and try to figure out what we're looking at. We also have the viral load status, which is both a PCR viral load status, which is the typical one gene, I believe the one gene spike protein kind of analysis that was done at Weill Cornell in 2020, or it wasn't just Weill Cornell, but a couple of other different hospitals last year. But it also gives us a high, medium, or low, which is based on cutoffs in the clinical test. There's a none, and there's also something called other viral, which I'll explain in a second. But the viral load status is obtained both in clinical PCR results, but also we can kind of ascertain what that is from the amount of SARS-CoV RNA-Seq reads that appear in the data post-alignment. We also have detections of other kind of viral infections from RNA-Seq data using a program called Kraken, which will distribute the data across different microorganisms and give us an idea of what other kinds of infections these patients or subjects may have as they come in. Like they may think they have SARS-CoV, but it just may be a case of the flu, or they may have both SARS-CoV and the flu. So there are a couple of cases of that. So these are our numbers at the bottom. You'll notice that we have for the high group, this is high viral load according to the PCR analysis, is about 64 people, medium, low, none, and other viral. And I'm sure it's going to come up as a question, how does the PCR results relate to the sequencing data? And I'm not going to show a slide on that, but I will tell you it's more or less aligned, although we have our suspicions that some of the people that are none have some kind of COVID infection that's not being detected either by PCR, like Rob Miller just said, it's about a 10% rate for some of the false negatives that came up. There may also be some non-people that actually have some kind of infection. It's either SARS-CoV or some other infection that gives them some kind of interferon signature, which you'll see in a minute. But these are the numbers that we worked with from the PCR data at first. So the Rna-Seq data analysis for the geeks in the room, which I am one, data is processed in a star-2-pass pipeline. It was done at NASA's HPC group with Amanda. It was kind of a covert. We also have a covert pipeline group, and the covert pipeline group kind of aggregates all the different types of pipelines we could run, and we try to standardize and harmonize our efforts under that group. We do batch correction based on, I do batch correction based on the sequencing plate ID from Cornell using SVA or Combat-seq, depending on what approach I'm using. Metadata filters, we try to take only those samples that have human counts of greater than 5 million, which is actually a little generous. Some of our group like to use 10 million. We try to also are a little bit more stringent. We look for average gene counts greater than 100, at least in my analysis. I know that might disappoint some people. I'd like to go a little lower, and that might be really useful for looking, for example, for transcription factors. But for this, there was so much data. We did this cutoff, and we can go back later. But when you do these kinds of filters, we end up with from the 670 subjects we had, it reduced to 444. So we lost about a third of the subject, a third of the samples. And you know, when we do take log, we're going to lose any zero counts of SARS-CoV RNA-seq counts. So I jittered them. There was a study done in Covert that was looking at, you know, the difference between PCR reads and in real time or in different, what would be the model for PCR reads or PCR cycles versus the actual SARS-CoV counts. And it turns out that a level about 100 is kind of possibly background. It seems to be a decent cutoff for some controlled experiments. So what happens is I take the 0 and I jitter it somewhere under 100 as kind of a noise factor. That way I'm not taking the log of 0. And then there's also this additional post-pipeline secondary analysis with DEC2 and R. We do a 2-group analysis, which is like what we like to think of as case control, just like high versus low, et cetera, and permutations of all those different combinations. We can also do a regression analysis where we're looking at the quantitative SARS-CoV RNA-seq counts and use that as the variable to test each gene against, and also individual sample enrichment analysis. And I'll talk about this briefly. So these are the 3 analysis types. The regression, the 2-group analysis typical, you know, like a case control, high versus none, you know, t-test, these kind of people think of a 2-group analysis. DE-seq will do this for you. And also, it will also do regression for you. Although a lot of people don't use the regression function in DE-seq2. You just have to put the model in. I know this is not inflammation, but this is geeking, but trust me. But regression is a gene's expression here in the figure. A gene expression, as a gene expression goes up, you could also look to see how it relates to the number of SARS-CoV RNA-seq counts that are determined from alignment. And then you get a nice curve that gives you a value that's kind of a unit change in SARS-CoV counts versus unit change in gene expression. That's kind of like a full change. And then we also do an individual Z-score analysis where we take the baseline of the negative people, the people with no SARS-CoV load, and they're considered PCR negative and have no evidence of having SARS-CoV. And then we take a gene and then we look at it basically kind of like a normalized standard deviation from that baseline and say that particular gene in that particular person. And then you do this for every gene in that person. Now you have a list of Z-scores of that person versus all of the genes in the baseline. And you can use that to do pathway analysis per person, which is actually really interesting. So our results for just a regular positive versus negative comparison, this is not looking at overall load, but just whether or not someone's been detected with SARS-CoV at all. If you're used to looking at volcano plots, you'll notice that it's p to the negative 40, which is a pretty healthy p-value. But what you're looking at here is the log fold change, which is cut off at 0.048, which is about a 1.5 fold change or so. Some people encourage me to go lower, and we can go lower. And then this is the log p, and this is the p-value of the change. So a lot of the genes, if you're familiar with inflammation and immunity, you'll notice a lot of these genes are very familiar. Some of them have to do with transcription. Some of them have to do with double-stranded RNA response, nonsense mediated decay, and interferon responses. And some of them are actually unknown. Like we didn't really know. Some of them are really well annotated in cancer, for example, but really didn't understand what they do in terms of immunity, or at least what they're doing in terms of SARS-CoV-2. And our group is actively working on a paper now to describe that. And this was the PCR positive versus PCR negative on the lower right-hand side. It was a healthy number of people. But even so, the results are, in all of these plots, are so high, it's almost like a gong going off in terms of gene expression. This is a very responsive gene expression plot. And so when you do pathway MCDB, I think Brad mentioned it's one of Afshin's favorites, but I like it too. You do an MCDB enrichment pathway analysis on the genes that are with the highest p-values, the first 200. I always take the first 200 ranked by p-value. We get a really obvious response here: interferon alpha, interferon gamma, interferon signaling, cytokine signaling, antiviral mechanisms, etc. So you know, if you go further down, you start recognizing even more pathways having to do with nonsense mediated decay, control of transcription, etc. So this was interesting. This is just positive versus negative, but can we do better? So let's start looking at high viral load versus none. So when you start going to high viral load versus none, now we're taking the two extremes, right? We're taking the highest COVID load versus no COVID load at all. And we did this, and the ends are 44 versus 226. So I have a lot more controls than I have cases. But 44 isn't a very big number. But even with 44 people, this is also, again, nasal swab data. So it's not necessarily the best data, but it's still, I mean, it's still an amazing signal, 10 to the negative 60 for some of these genes. And this, I mean, we've gone and looked at this in detail, and this is a very strong signal. When you actually look at the plots of the gene expression itself, you can actually see the signal itself. So some of these genes, for example, ZNFX1 is a mitochondrial localized function as a double-stranded RNA sensor to initiate antiviral responses. And then we have some other genes through here, like interferon responsive, again, the same group of figures, but instead of just doing positive versus negative, we're taking the extremes of those. And we did see a lot of interesting things in terms of inflammatory signatures in the rest of the profile. And then when you do, again, the typical FGC analysis, you're going to see the same players that we saw before, only even stronger. Some of the, now we have some very strong FDR p-values for some of these pathways, where we're getting a lot more genes involved in these pathways because we took the extremes of the distribution in terms of the two group comparison. So regression analysis on all the COVID-19 tested individuals. Now I'm taking the actual level of the SARS-CoV reads. How much viral load do I see? And so each sample is going to have a viral load, and it has gene expression. Now I have to ask, what genes seem to change the most with viral expression, you know, with the viral load? What gene expression changes the most with viral load? So now you're doing a typical kind of regression analysis with DE-seq. And we again see some even more outrageous p-values because we're starting to get even more genes populated because they have a very high response to viral load, which is not surprising because a lot of these are responding to viral load. That's their job. And when you again look at the MCDB enrichment analysis with this, it's again the same old players. Now you're starting to see a little bit more in terms of adaptive immune system coming in. And class 1 MHC needed antigen processing. So now we're seeing a little bit more signal instead of just complement and just within cell responses. We're starting to see things like the adaptive immune system come in when we start looking at the response to virus, which is interesting if you think about it, because, you know, on either side, we didn't really see that in these pathways as well. It's lower down, but it doesn't have as much significance. But as the viral load increases, we're starting to see more and more response to the adaptive immune system. So this is the analysis when we take individual people. So how am I doing for time, Eve?
30 seconds.
I have 30 seconds. Okay, great. So let me just show you one plot, and then I'll go to the end. So we have here a plot where we're looking at each dot is a person, and I've ranked their gene expression by Z-score, as I mentioned earlier. Each gene gets its own Z-score, and then I rank those Z-scores, and I can treat each person like a sample. And so now I can look at to see how the gene expression, how the enrichment analysis performs based on their status of whether they're high, medium, dark, low, or none, or other viral. And you can see here, the normalized enrichment score in GC means how enriched is this pathway for the genes that I gave it. And you can really see a very strong signal here of most of the COVID groups, especially the highs here in the bright red, are pushed towards a very high enrichment score. Each person is for the interferon alpha response. But then you see some people down here that don't, that actually are kind of on the opposite side, meaning they actually have a down regulation of those immune responses. We don't know why. But this is something we'd like to look at. And on the right-hand side, you can also see the same thing. Black is negative and red is positive. So I'm gonna skip through all the interesting things that I don't have time to show you, but I will say that that this is one of Paco's last slides that he showed during his talk for nonsense media decay. And we actually do see, we also do see a decrease in NMD, or maybe more of a control in NMD on average in the people with high or low COVID-19. It's just barely significant. But we also see a decrease in ribosomal proteins as well. So there's something going on with the transcription, and which has been published by other people. But of course, we're using RNA-seq data, so we can only tell you what's happening in terms of transcription.
Yeah, I'm sorry.
No, I'm going. This is it. So I'd like to thank everyone here, and any questions I can answer them typing.
Okay, thank you, Deanne. Yeah, so we have next and last but not least is Doug Wallace, also from CHOPS. He's got some really cool ideas to show us. Doug, are you? Is Doug there? Does anybody know?
I'm here.
Oh, great. Okay.
I'm here, and I'm trying to find my slides up. So I don't know if you're seeing anything.
We're seeing you.
Oh, okay. Well, that's not help. Sure. Let me start again. I am sorry I had this problem before. Share. Oh, damn. Sorry, folks, I don't see my talk. I don't know where it is.
You're clicking the little green share screen and you don't see anything?
Yes. Well, I get a list of things, but without showing my presentation. Why would it do that?
Doug, if it might help, you could email the slides to me and I could show it and you could just tell me to advance.
Yeah, why don't I do that? I'll email the slides to you.
Email to my Broad email. That'd be, emails come quicker that way.
Yeah.
Thanks, Epstein.
Yeah, no problem.
Sorry.
And while we're waiting, maybe we can take some questions. Is there a question, any questions that we want to maybe go back to and answer while we're sure?
Let's see. So, Guy, I had a question. Are you there, Guy?
Yes. I'm here.
Oh, yeah. So I was curious about, in terms of this alternative splicing, are there lots of conditions in which there's like massive alternative splicing, or is that particularly noticeable with viruses?
Well, we see that in, for example, differences between different tissues that are not affected by disease at all. So it's a very important mechanism. I wouldn't rate it less important than differential expression, for example. But we do see it having a big impact on it by viral infection, and it was long before the COVID pandemic. Shown for other viruses that they affect splicing, they interfere with the machinery. There is sort of a co-evolution between host and virus, so they probably try to take advantage of some of it, try to disrupt the antiviral aspect of differential splicing.
Eve, you're muted, I think.
It's not something that's stress related. It's actually specifically the viral, something to do with the viral genome and what it's doing. Is that your hypothesis?
Yes, for example, for dengue, the first slide, there was a paper there about dengue. NS5 is a viral protein that interferes with the spliceosome. They did a lot of experiments to show mechanistically how it interferes with splicing. Since it goes to the machinery, we expect to see a lot of changes. And yes, it's not just the host responding, it's from both sides.
Thanks.
Sure.
I sent that. I sent that to your ACR. I've got to find your other address.
Okay. Unless we want to move on to the Don Mellon, and then Doug, you could talk after Don.
Yeah.
I mean, what do you think? Is that easier?
Why don't you do that? Yeah.
Okay, let's go ahead and
I'll figure out what's going on.
Yep,
Yeah.
no worries. Let's go ahead and do that then. And our final keynote speaker here is on the panel, so I think we can go ahead and get started. So Don, you can feel free to turn your camera on. I will go ahead and introduce you now. Okay, so thanks so much for joining us. And so I have the pleasure of introducing Dr. Don Milton, who is a professor of environmental and occupational health at the University of Maryland School of Public Health. And Don is with us today as a renowned expert in aerosols, infectious disease, aerobiology. He's a very close collaborator of mine, and very excited to have him here to end our symposium with this keynote. He has served on the editorial boards of Applied Environmental Microbiology, Indoor Air, BMC Public Health, NIOSH NORA Indoor Environment Team, and chaired bioaerosols committees. His work is it's too long to list out all of the amazing accomplishments, but I will highlight just the impact he's had in the last year on the onset of the pandemic, and has been leading the way with a lot of the aerosols and infectious disease aerobiology. So with that, without further ado, I will let Don tell us about understanding the control of respiratory virus transmission. So Don, please take it away, and thank you so much for joining us.
Hi, thank you. It's a pleasure to be here. I guess this is a bit of a change of pace from what you've been talking about with transcription, translations, switching, and so forth. I'm not going to talk about any molecular biology. I'm all going to be about much more gross level aerobiology. And I'm going to only show one slide that I think you will have seen before, and that is probably something version of this one, but I've tweaked it a little bit. Talking about how do respiratory viruses transmit. And one of the big problems we're having today is the conversations about this are like a Tower of Babel. It's as though people were speaking different languages, using the same words to mean different things. And I've been ragging on this theme for a while now. I published a paper last summer called The Rosetta Stone for Understanding Drops and Aerosols, because people use terms in such vague ways. Basically, we've got spray-borne drops, or you can call them droplets, but they are ballistic. They are propelled from the source to the target. You have touch, where people touch contaminated things or touch infected people and transfer the virus directly to their mucous membranes or indirectly through their fingers. And then there's inhalation, where we can inhale aerosol particles that can be pretty large, such that they only deposit in the upper respiratory tract or much smaller and penetrate deeply. And I talked to you all about these ideas before. When we look at what was being said about how to control transmission back with the Hospital Infection Control Practices Advisory Committee in 1996, which was building on all that had been happening with HIV and hepatitis B up to that point. And they talked about being concerned about contact transmission, droplet transmission, and airborne transmission. And when they said contact, one of the problems with that term in my book is that contact is used in infectious disease modeling as sort of a catch-all term for any way that a virus gets from one person to another, or an agent gets from one person to another. So, but they mean touching. Droplet transmission is not describing a mechanism, but a thing, but they were quite clear that droplets were propelled a short distance through the air with direct hits on the host conjunctiva, nasal mucosa, or mouth. And when they talked about airborne transmission, they said dissemination of airborne droplet nuclei. So this is a term of medicine like it was coined by Richard Riley when he was a medical student working in William Wells's lab in the early part of the 20th century. They were talking about things that were small enough to remain suspended in air for long periods of time and be inhaled, whether in the same room or over longer distances. Now, they also said that these droplet nuclei had to be less than 5 microns, or a dust particle, which they didn't define what that was in terms of physical units. But the idea was there that this was inhalation exposure, and that was distinct from droplets being propelled through the air. And that translated quite directly into things that you would do. Contact transmission meant cleaning surfaces, wearing gloves and gowns, hand washing. Droplets being propelled meant all the above plus face shields and goggles, and maybe face masks. And airborne transmission meant ventilation, filtration, UV air sanitation, respirators. It was quite clear how these definitions translated into control and prevention. Where are we today? Well, the Centers for Disease Control's science brief about SARS-CoV-2 transmission says contact transmission is spread through direct contact with an infectious person, for example, touching during a handshake. So it's basically the same as what we were talking about before. And airborne transmission, they say, is exposure to virus containing respiratory droplets comprised of smaller droplets and particles that can remain suspended in air, usually greater than 6 feet, and time, typically hours. But they reserve the term airborne to describe infections capable of being transmitted in the air over long distances. Within the same room is gone. Whereas droplet transmission now says exposure to respiratory droplets, larger and smaller droplets and particles, exhaled, no longer propelled, by an infectious person. Transmission is most likely to occur when someone is close to the infected person, generally within about 6 feet. So now, distance, not the mode of exposure or the route of transmission, have become the characteristics defining the difference between droplet and airborne. So how does this translate into a control strategy? Well, contact is still contact, and airborne is still sort of airborne. But what is droplet? What are we supposed to do with this category? How do we protect people up close? Now, to be fair, CDC has said all along that healthcare workers should have N95 respirators if possible. But during an emergency, and they haven't relented yet and said the emergency is over, we have supply, even though we do, you can do other things. So where's this confusion coming from? Well, some of it may be political, because the American Hospital Association, in their testimony to Congress earlier this month, still says all we need is more plexiglass barriers, and we shouldn't worry about giving people respirators because they don't want to pay for them. But there's another piece to this, and that comes from the deep history of infection control and thinking about infection control in the infectious disease community, the medical community, and especially this textbook from Charles Chapin, published first in 1910, in which he said, undue emphasis paid on the invisible and therefore terrifying infection in the air seriously interferes with rational measures for the restriction of the disease. And he was talking about tuberculosis, because he was a Victorian. He believed that cleanliness was next to godliness, and if you did anything that dissuaded people from the importance of hand washing and bathing, that was bad. So he also felt that the mere idea of it being in the air was terrifying, because they didn't understand aerosols, they didn't know what to do about aerosols, and therefore it would just scare people to no purpose. But I have heard some of the IPC committee members from WHO make statements that you could just replace measles in these quotes from the 1910 and 1916 editions of Charles Chapin's book with SARS-CoV-2 or COVID, and it would be almost exactly the same statement. That he didn't think that measles was transmitted by aerosols. He felt that close contact was the driving force. And even with TB, we know that, if you are sleeping in the same bed with a TB patient, you're more likely to get infected than if you're sleeping in another bed in the same room, or if you're in the same household, you're more likely to be infected than if you're just out meeting them on the street or in a casual encounter somewhere else. But even so, we see with TB that only about, in places where it's really still endemic, which is a lot of the world, only 20 or 25% of the transmission occurs in the household. Chapin felt that the reasons for belief in airborne transmission was that it was superstition. It was a theory of aerial infection prevailing from remote antiquity. And he's talking about miasmas and pestilential vapors. And so he says, contact is the most certain and obvious mode of infection transmission. You can see it, you can observe people touching things, and so other modes should not be assumed, and the burden of proof rests on those who make the assumption. Well, that's where we've been now for quite some time. And in this near zone, the burden of proof has been on saying, well, if it's up close, then prove to me that it's not airborne. No, prove to me that it is not droplet spray in direct contact is what they're saying. So the words have gotten so confused that it's very hard to say droplet and know what anybody means by droplet, or say airborne and know what people mean by airborne, because some people are saying airborne means that it's not transmission in this room, but to somebody in the next room who never entered the room that I'm in. So we need to change the words. We need to have words that communicate. And so Hugo Lee has proposed that we use a transfer process focused categorization. The transfer processes are that one touches something, one gets sprayed with something, or one inhales something. Now, these are really quite clear categories. Distance makes a difference because you can't touch something you aren't close to. You can't get sprayed if you're not close. You can touch something that somebody else touched and they're now gone. And you can inhale air that they left behind. But distance is no longer the driving force. It's the transfer process that is the driving idea behind the categorization. And these then lead to clear-cut control mechanisms. And we're back to where we started in 1996. Okay, but getting back to the future means having language that means what it's supposed to mean. And so where are we now? Well, we were there in 96. Hopefully this is where we're going with the SIT framework. But I'm afraid officially we're still here stuck in the middle with this mishmash of a definition of droplet transmission. Even in the face of having published, in this slide I used last time, data on the presence of RNA in the air in hospital rooms, of transmission over long distances in a restaurant. This is a slightly updated picture now with the published version here, where he nicely shows through computational fluid dynamic modeling, going from red to blue, the concentration in the air that was being blown around and recirculated in the restaurant. And the Skagit Choir outbreak that probably everybody has heard about, and which has been modeled based on assuming that the index patient is this one person. And that model ends up with saying that the shedding rate was extremely high. But remember, back then, there was no testing of asymptomatic people. And in fact, as you can see here, many of the symptomatic probable people weren't tested. There were 3 cases with onset the next day. They weren't infected by this person, at least not on that day. So there were at least these 3 people, probably some of these 7 or 8 people, and some asymptomatic people, all shedding at this choir practice. There was probably an asymptomatic case back at that earlier choir practice. There were as many as 10 people infectious at this choir practice. And if one assumes that, you can come up with a reasonable number for the amount of virus being shed in the air, which comes out to be a little less than the average measles case, which makes sense. We know that this infection is not as explosively contagious as measles, and that the outlier events probably were driven by people who were as contagious as an average measles case. Most people are much less infectious. And then, of course, we have another restaurant outbreak reported more recently in South Korea, and we know that B and D entered through door 2, whereas C and A entered through door 1. They never got any closer than they were when they were sitting at these tables. Clearly, quite a long distance. Lednicki cultured virus from air samplers in the patient's room with 2 patients. And still, there was a lot of resistance in the medical community to understanding this role. Why? Well, because the burden of proof lies on those who want to say that it's airborne. But the big problem has been that the time from onset of symptoms to transmission, or the toast interval here, has been very short. And this is what has made this so hard to control compared to SARS-1, where that interval was, this whole peak would have been shifted to the right 5 to 10 days. Here, the interval is happening right around the time that symptom onset happens, and much of it beforehand. As a result, it's very hard to control. And it also says something about who in the hospital is at greatest risk. Probably not the people intubating a patient, because by then they've been sick for 10 days and they're probably not contagious much anymore. It's the people in the outpatient clinic, in the ER, and the dermatologist seeing them for some other reason because they're asymptomatic who is at risk, or pre-symptomatic. But I think the tide has changed. Klompus's paper in Annals of Internal Medicine recently showing a big outbreak at Brigham and Women's Hospital with sequencing data to nail the case of who infected whom, clearly showed that people were infecting people who were wearing surgical masks who were in the same room, that people with surgical masks on were infecting other people. This was not spray-borne transmission. This was not touch transmission. This was inhalation transmission. And I think the medical community is going to come around to understand why we called this nearly 20 years ago the elusive pathway, because you don't see it. It's not obvious, as Chapin would have argued, and that's what makes it tough and interesting. So, where are we today? I argue that we are moving on to the SIT framework, that it's spray, it's inhalation, and it's touch. It's transfer process. We need to use these words to communicate. And then we can begin to really think about the hierarchy of controls. This is copied off of NIOSH's website. Well, we can't eliminate people who are infectious. They're around. We can't substitute an innocuous virus for the one they have. So what can we do? We can use engineering controls to isolate people from the hazard. These are automatic things that don't require people's much cooperation. We can do administrative controls. This is a group behavioral intervention, essentially. And then we finally get to the individual behavior interventions. So how does this translate into, say, opening schools? Well, first of all, ventilation is an engineering control. We can get air changes, we can get clean air delivery to prevent transmission. We could also use upper room UV. I'll get to that in a minute. We have contact tracing and isolation and quarantine. These are administrative controls. They require group behavior and some human behavior in terms of cooperation. We have to move meals outdoors. or install really highly effective air sanitation that can get very high rates of air cleaning where people must take their masks off and are together. And I think going forward, we want to think about how can we control transmission in places like restaurants and dining halls on a college campus. And because we see on our campus that transmission is happening where people want to get together and socialize over a meal. That's a very natural human behavior, and we need to make it safe. Finally, we get to the individual human behavioral interventions, masks and respirators, hand washing, and respiratory etiquette. Those are our last line of defense, not the first line. And we need to realize that and be promoting that. I mentioned upper room UV, especially around lunchrooms. It's a little more difficult to install and perhaps more expensive than just putting out air cleaners, but it can achieve much, much higher rates of air sanitation than you can achieve through pretty much any other known mechanism at this point. In the long run, 222 nanometer UV may be the way to go, but right now I think that 254 nanometer UV is much more accessible. But you can see in this experiment we did in a simulated hospital room at Harvard School of Public Health years ago that we were able to achieve under certain conditions the equivalent of 1,000 air changes per hour. You just can't get there with ventilation. In our laboratory at the University of Maryland School of Public Health, we used the data from that Vaccinia trial, estimated the coronavirus sensitivity as being about only 20% as sensitive as Vaccinia based on laboratory experiments that were done by Guanpyo Ko when he was in Houston. But we still estimate that we are getting 480 air changes an hour for coronaviruses with a half-life of a little over 5 seconds. We get complete removal of 3 logs or more in every 37 seconds. And you just can't beat that. We have piloted doing this in classrooms in the past. It's a feasible intervention. And we know we're up against this terrible pandemic with an incredible mortality. We have something going for us in schools because in schools, what happened to my talk? In schools we have, come on. In schools, we have children, and under age 18, the mortality rate is bad. I mean, it's about the same as for a bad flu year. Like last year was a bad flu year, we had almost 200 deaths. Well, we've had over 200 deaths, almost 250 deaths in children from COVID. But it's nothing like the deaths we see over age 65 or even 18 to 64, where we're seeing many thousands of deaths. So I want to thank you. I think that's the end of my presentation.
Yeah, thanks so much, Don. That was a really nice overall description of everything. Really thankful that you were here to present that to us. I'll just take, for the interest of time, I'd love to go and get into a big conversation, but I'll limit it to one question. And the question is, as members of the scientific research community, how can we better help inform public policies and reduce confusion, misinformation about managing infectious diseases?
Well, that's the objective of this new terminology, the spray, inhalation, and touch formulation, is to give us something easily to communicate clearly about. And I think using those terms, touch, spray, and inhalation, is an important place to start. And then promoting the hierarchy of controls within that framework.
Right. So thanks so much. Really wonderful presentation. Thanks for joining us. As always, you've participated in previous events, so we always help raise the profile of our events with your presentations. And thank you so much for going over that so carefully with us today, Don. So thank you.
You're welcome. Thank you for inviting me.
Yeah. And so I think we're going to go back to our previous talk and so take it away. I think the slides are on screen now, which is great. So let's get back to this talk now.
Okay, so I thank you very much. Thank you, Afshin, for showing the slides. I'll tell you when to change. So I've been asked to represent the Covert Committee on Mitochondrial and Glycolytic Changes in COVID-19. And this committee really got started with some seminal work that Robert Miller and Afshin did, where they looked at the Rna-Seq data from bronchoalveolar lavage samples from Wuhan. And when they looked at the host message analysis, what came up is a very strong association with oxidative phosphorylation and mitochondrial functions, as well as ion transport and membrane proteins. So that then led to the realization that maybe mitochondrial function was very important in this disease. May I have the next slide, please? Option. Are you changing? Oh, there we go. Okay, so what we're going to talk about today is how SARS-CoV-2 really inhibits mitochondrial oxidative phosphorylation, increases glycolysis, and activates the integrated stress response. The human cell has two different life forms: a nuclear cytosolic life form that has about 20,000 genes that are transcribed and translated in the cytosol, and then hundreds of mitochondria in your cell, each of which has its own mitochondrial DNA transcribed into ribosomal and transfer RNAs and 13 polypeptides. And those 13 polypeptides are components of the mitochondrial energy generating system, oxidative phosphorylation. So in the lower right-hand corner, you'll see that there are 7 polypeptides of the 45 proteins of complex 1, 1 of the 11 proteins of complex 3, 3 of the 13 proteins of complex 4, and 2 of the 17 proteins of complex 5. And the mitochondrial polypeptides are translated on mitochondrial ribosomes, which are intermediate between the eukaryotic ribosome and the bacterial ribosome, being initiated by N-formyl methionine. Now, if you impair either cytosolic or mitochondrial protein synthesis, you get an imbalance in the polypeptides that are used to assemble mitochondria. And that's because the nucleus codes for between 1 and 2,000 mitochondrial genes that are translated on cytosolic ribosomes and imported into the mitochondria by the import systems, as the TOM and TIM systems, as you can see in the middle of the slide. So basically then the virus, as I hope you'll see, manipulates mitochondrial function as an important component of its life cycle and its propagation. So if you, as I said, cause imbalance in these two systems of making proteins, you'll get the unbalanced protein response in the mitochondria, and that will initiate the integrated stress response. And all of these are features we've seen so far. Next slide, please. So we got involved in this really in February when a preprint was made available from Gordon and et al. Out of UC San Francisco, where they expressed each of the viral proteins in cultured cells and then looked at what polypeptides in the cell those polypeptides from the virus bound to. They concluded that they were confident about 332 interactions. So I, at the time, got interested and went through all those proteins and found that 55 of them were well-known mitochondrial proteins. That is about 17%. And then my colleague, Joe Guarineri, has looked at other papers of similar results and has found that in general, it's between 12 and 18% of all of the host protein interactions are mitochondrial. And that's the overlap shown to the lower left. And there are multiple proteins that are common to multiple mitochondrial proteins that are common to all of these studies. And on the right lower, you can see that the M protein interact with the TARS and NARS proteins, which are tRNA synthetases. The PMP-CA and PMP-CD proteins are important in mitochondrial protein import from the cytosol, as is the TIM70 system. The NSP6 protein interacts with the ATP synthase, that's complex 5. The M protein interacts with coenzyme Q, which is a carrier of ions from or charge from complex 1 to complex 3. And the NSP5 protein binds to the antioxidant enzyme glutathione peroxidase. So it turns out that the virus is impeding a large array of different mitochondrial functions. Next slide, please. So if you look on the left, this is one of the studies where they looked at some of the viral protein inhibition occur, and most of the proteins that they looked at that are involved in mitochondrial energy production, oxidative phosphorylation, were downregulated. In mice that have been infected with SARS-CoV-2 using an introduced ACE receptor, all of the blue lines in the middle indicate downregulation, and the green is complex 1, the yellow is complex 4, and the red blue is complex 5. So you can see there's a stark downregulation in heart, kidney, and spleen. And that can be seen then if you look at the transcript levels, where many of the mitochondrial transcript levels are also downregulated in a viral infection, both in bronchoalveolar lavage fluids and also in the lung. Next slide, please.
There might be a lag once in a while. I already switched.
Can you change the slide?
Yeah, I already did. I changed it when you say it. I think it's
okay. I'll wait. Sorry. Okay, so as Deanne so beautifully mentioned, she and her colleagues have been doing bioinformatic analysis of the Weill Cornell nasopharyngeal swab data, messenger RNA. And this is an analysis that Afshin did looking at mitochondrial oxidative phosphorylation proteins in different viral load levels. And the far left with orange is high, then blue is medium, green is low, and gray is non-viral. And what you can see for complex 1, complex 3, and complex 4, and complex 5, that the high viral load has a lot of blue, and that's strongly downregulated. But what's is that the mitochondrial DNA, which codes for the 13 polypeptides translated on mitochondrial ribosomes, shows exactly the opposite effect. You can see that that on the high viral load, in fact, it is not only either downregulated, but maybe even upregulated. And in the non-viral load, that's in the non-SARS-CoV-2 virus, that's where it's downloaded. So there is a disconnect between the transcriptional regulation of the nuclear encoded mitochondrial OXPHOS genes and the mitochondrial DNA encoded OXPHOS genes. Next slide. So one of the aspects of that, when you have an imbalance between the mitochondrial DNA encoded polypeptides and the nuclear encoded polypeptides, you're going to induce the integrated stress response. And the some of the most important proteins in that integrated stress response are proteins that activate the phosphorylation of the translational factor, elongation factor 2 alpha. And the key kinase that does that is PKR in the endoplasmic reticulum. And you can see that that is very highly activated in the viral affected, high viral infected cells. The other interesting thing is that PGC1 alpha is the main transcriptional activator for all the nuclear encoded mitochondrial genes. And you can see that it is in fact much less strongly activated than is the integrated stress response. So the system is really trying to cope with an imbalance between the mitochondrial DNA translation products and the cytosolic translation products. Next slide.
Should have changed, but give it a little bit of time.
It's okay. So this is a wonderful analysis that Afshin did today, which I really much appreciate. And what we see here is looking at the expression of the mitochondrial proteins, messenger RNAs, in the autism, in the autopsy samples that Weill Cornell of patients infected with SARS-CoV-2 or not infected. If you look in the center, there is at the top a red bar right in the middle of each of these columns, and down from that is a lot of blue. And what that's saying is in the heart of these patients, there is a striking downregulation of all the nuclear encoded mitochondrial oxidative phosphorylation genes. The other thing you see, if you look on the left on complex 1, beside the column of blue to the right is a column of light blue, and that is also in the kidney. So the tissues that have the highest energy demand are the brain, the heart, and the kidney, and those are the ones we're seeing the most effect on SARS-CoV-2 infection. And I'd like to suppose then that the virus is down-regulating mitochondrial function in these high energy tissues. And that's one of the reasons we're getting these complex phenotypes that we see in the long haulers. But I think this is really remarkable coordinate downregulation of a large number of genes that all are involved in the same process. And again, this would then create this unfolded unfolded protein response and create an activation of the integrated stress response. Next slide.
Should have changed. Sorry for the lag.
That's okay. I'm sorry that my computer froze. I don't know why it did that. So this is a wonderful study that was done by Hussein, where he's now looking on the abscissa axis, the messenger RNA levels with the middle line being normal, and on the coordinate axis, the protein levels. And he's combined the data from two very important but different studies. And what you can see that there are certain genes that are very highly transcriptionally activated, even though they aren't necessarily altered too much in protein. And I just chose this example. SCO2 is a key assembly factor for cytochrome C oxidase or respiratory complex 1, and PNPT1 is the polynucleotide polymerase 1, and its key function is to import RNAs into the mitochondria. And you can see that in the presence of inhibition of mitochondrial function, the nucleus has upregulated the transcriptional regulation of these genes to apparently to try to compensate for an energy deficiency that the virus is imposing. Next slide.
Slide has changed, but give it time.
That's all right. So I just want to finish by saying that we now feel that there's compelling evidence that SARS-CoV-2 infection causes a coordinate downregulation of nuclear encoded mitochondrial function genes and proteins, that there is an effort by the host to induce transcriptional expression of these genes to compensate, but the inhibition of the virus of the protein seems to be sufficient that it cannot compensate for that defect. And that leads to the interesting idea that maybe a lot of the symptoms that we see in the long haulers and in the activation of the innate immune system is due to this chronic energy deficiency. So I'd like to just thank, of course, the leadership of Afshin and Sigrid, both are representing here from NASA, and of course, Chris and his colleagues from Weill Cornell that generated all this RNA data. my colleagues from CHOP and Penn, shown in the middle, and I'd like to particularly mention Deanne, who you just heard from, her and her colleagues on the left, and Larry, Joe, and Yentley on the right. Waldemar is our glycolysis expert. Robert Miller has been our leader in this area, and Sonia at the lower left, and Steve, and Jonathan, Eva, Yared, Mark, and Anna have all made important contributions. So thank you, and sorry for the technical problems.
I think I'm trying to turn on my camera. I think what time is it? I think there's some questions, Doug, but I don't know if you could see the Q&A.
So one question is there any overlap?
Sorry to interrupt, but I think would you be able to just type in answers to those questions, Doug, if that's okay?
Sure, I can do that later then, if that's what you'd like.
Yeah,
That'd
yeah,
be fine.
due to the shortage of time. Yeah, but that's
yeah, absolutely.
Thanks, Doug. Yeah. I guess we'll
move thank on. You.
Yeah, thank you. Yeah, so I guess we'll move on. I guess Todd, did you want to, or Krista, Todd, and others for the panel session? Turn on your cameras. I guess Leo, there you go. There he is, our leader here.
Yeah, I think Leo's in charge. So.
Yeah.
All right, yeah, if everyone's ready to go, we'll go ahead and get started. So I'm Dr. Leo Elworth. I'm a postdoctoral scientist in the Triangle lab and have been a Covert member for some time now. And we're just gonna kind of go over things like how Covert got started and the future of Covert, and also hear from some of the subgroup leads on what they think some of the major accomplishments have been for Covert. So why don't I just start by going around and having everybody give a very, very brief introduction of themselves in case anyone needs a refresher on who everyone is here. Maybe they didn't tune in earlier. So I'll just go around real quick and say a person's name and you can just give a quick introduction. So Dr. Eve Wertelli.
Okay, so I'm at Iowa State and I never really know what I am. I started as a biochemist and I've gotten into bioinformatics, and I'm really interested in orphan genes right now. And I'm super involved in Covert and how the orphan genes may relate to that.
Okay, Dr. Afshin Beheshti probably doesn't need an introduction, but we'll do it anyway.
Yeah, so one of the co-leads of Covert, and I'll leave it at that because you've seen my face.
Okay, Dr. Kyle Hernandez.
Hello, I'm a research assistant professor at University of Chicago and a lead of the modeling subgroup here.
Okay, and Dr. Todd Triangle, also probably but we'll do it anyways.
Yep, assistant professor at Rice University, computer science department, and also lead of the viral genome evolution subgroup and co-lead of Covert. Thanks, Leo.
Yeah, no problem. And also Dr. Krista Turnus.
Hello, I am a bioinformatics specialist at Signature Science, which is a company based in Austin, Texas.
All right, and finally for the panel, we have Jim Cosby, who's actually planning on sharing real quick, like 1 or 2 slides. Are you able to share those?
Sure, I am. Hey, everybody, Jim Cosby. I am a Chief Technology Officer with NetApp. It's a data management cloud-based company. Did you want to go ahead and share those now or at the end of the time frame?
Did you want to introduce Peter, Leo, first? Oh, no, wait. I thought Peter has his camera on. I don't know if he was part of the panel.
Well, yeah, thanks for, Peter Robinson from the Jackson Laboratory. So we're interested in COVID genomics. I'm also involved in the National COVID Cohort Collective, N3C, and where we have data on 4.4 million patients, about 1/10 of whom have a COVID diagnosis. And that's a very similar project to this, and being very, very open, but with a focus more on statistics and machine learning analysis of EHR data, in case anybody is interested.
All right, great. Jim, do you want to go ahead and share those couple slides?
Sure, just one second here.
Maybe I could give context while as you're sharing too. I had mentioned earlier, way back when we first started in the morning, that BSA, the Breathing Space Accelerator, is joining Covert, and Jim's part of that initiative. So,
right, you should be able to see them now. I think Breathing Space Accelerator.
Yep.
Perfect, perfect. Okay, so thank you everybody for the time today. I am with the Breathing Space Accelerator Consortium group. We started this effort about a year ago in April of last year, and it's a broad group of healthcare professionals, doctors, scientists, data technologists, and architects such as myself, as well as researchers. And we came together around trying to help folks get breathing space dealing with the COVID pandemic. And it's for the sake of those over 500,000 lives that were lost, since this has started, that we really try hard to come together and work with technology to help, fight the pandemic. And that being said, there's multiple technology partners in the BSA. Two of the main ones that I'll just talk simply about today are Ever Healthcare and a company called NetApp. And Ever Technology and Ever Healthcare is the baseline application that we chose. They're a healthcare platform based out of Thailand, who is expanding into other parts of the globe as well. They're caring for a little over 3 million patients today throughout Thailand, and caring for a majority of the hospital systems with the technology they bring. One of the things they have that is really nice is their blockchain-based health information exchange core application. So it uses blockchain technology to securely let patients, doctors, and facilities share information about each other, as well as revoke sharing that data in a very secure fashion. And then they have various medical systems around that, such as ancillary services integration and various telesuite parts of their system, such as telemedicine, patient health records applications, and chatbots. And they also have an epidemiology suite and alerting system built into their platform as well. So it is also agile developed in something called Kubernetes, which makes it very modern day age and very adaptable. So plug-in and pluggable is one of the technology terms we like to use, which lets it quickly integrate and talk to other systems. So that combined with the data management, as you move to the bottom of this slide for NetApp, is where the efficiency and then the data flexibility comes in with NetApp. So we are a cloud-led data-centric software company that focuses on something we call data fabric, that lets data be used in any location very quickly and very efficiently, be it in the cloud or in a main data center.